Good morning. I'm Dirk Sostman, and I'm very happy to welcome you to the George and Angelina Kostas Research Center for Cardiovascular Nanomedicine International Meeting, which, like so many other things, we're holding virtually this year. The term nano originates from the ancient Greek word nanos, which meant dwarf. And used as a prefix, nano denotes 10 to the minus ninth. So a nanometer is equal to a billionth of a meter, and a single atom is about a tenth of a nanometer in size. Accordingly, the nanosciences deal with very, very tiny phenomena in the order of less than 100 nanometers. And so the word nanotechnology implies a very small thing. But nanotechnology is actually a very big thing. Focus on the nano world began in 1959 with a very provocative speech by Richard Feynman, who, as you all know, was personified provocativeness. And the speech was titled, "There's Plenty of Room at the Bottom." And this new look from such a distinguished figure propelled the scientific community's focus on nanoscale phenomena in general. The field began to emerge in the 1980s with two major breakthroughs. First, the invention of the scanning tunneling microscope in 1981 by Binnig and Rohrer, who received a Nobel Prize in Physics in 1986. Second, fullerenes were discovered in 1985 by Croteau, Smalley, and Curl, who won the 1996 Nobel Prize in Chemistry. The field really took off at the beginning of the 21st century and has continued to grow at an amazing pace. Here at Houston Methodist, we're fortunate to have the Kostas family, whose generosity has made us a leader in the field of nanotechnology. In 2014, a gift from George and Angelina Kostas allowed the Houston Methodist Academic Institute to begin operations of the George and Angelina Kostas Research Center for Cardiovascular Nanomedicine. George Kostas, the son of Greek immigrants, was born in the United States but returned to Greece at a young age. And when he returned as a young teenager, he did not even speak English. And yet, George was a pioneer in his own right and an immensely successful individual in many ways. He valued education, and he earned a chemical engineering degree from Northeastern University. His alma mater awarded him an honorary doctorate in 2008, and he also earned an MBA from Columbia. He had a long, illustrious career in organometallic chemistry. Among other signal accomplishments, he was recruited by the U.S. government to start and operate the first United States synthetic rubber manufacturing plant, which was already being built in Baytown. In 1946, he was appointed a member of the U.S. R&D Committee and served in this capacity until 1955. George Kostas was a man of science, and his wife, Angelina, was a woman of faith. Together, they believed and personified giving back to the community. We're so pleased and grateful that the funding provided by the Kostas family has galvanized interactions between the departments of cardiovascular sciences and nanomedicine. And we're really delighted that a contact with Northeastern University remains, and we're very grateful to Dr. Lutzi for being part of the executive committee for this meeting. So these collaborations and this funding has led to robust interactions, particularly in the generation of RNA therapeutics. They've laid the strong foundation of knowledge and expertise which built our new Center for RNA Therapeutics at Houston Methodist. And the director of that center, Dr. Cooke, is going to be moderating the beginning of this meeting. The collaboration between our RNA and nanomedicine scientists is critical, as RNA drugs need to be encapsulated in nanoparticles. A perfect and indeed world-changing example being the messenger RNA vaccines against COVID-19. Since its inception and under Dr. Cooke's leadership, the scientists in the Center for RNA Therapeutics have generated more than 100 different RNA drugs for researchers around the world. And this center is one of the premier academic units for developing RNA drugs. The Kostas family has played an important role in catalyzing this exciting development at Houston Methodist, and we are immeasurably grateful to the Kostas family. And we thank you all for joining us here today. I believe that the time you devote to attending this meeting will be amply rewarded with interest and knowledge and inspiration. And with that, I'll turn it over to Dr. John Cooke to make the introductions.
Thanks, Dr. Salzman. Thanks for that very nice introduction to our meeting today. We have a spectacular meeting for you today. The Kostas meeting is really reaching an apex here today with some wonderful speakers in the morning. And then in later morning, we'll have some poster sessions. We have lightning round presentations from our postdocs. We have afternoon panel discussion on the future of cardiovascular nanotherapeutics. And we have some more excellent talks in the afternoon. And then our feature, the Shark Tank, which was so popular last year, we'll have that in the afternoon as well. The Shark Tank, where 3 of our investigative teams present to a panel of judges their target product profile and their plans to develop a transformational product. Well, as I said, we have a wonderful program lined up. Our first speaker is a giant in nanomedicine, Peter Cullis, who is a professor of biochemistry and molecular biology at the University of British Columbia. And he is a scientific director and CEO of Nanomedicine's Innovation Network. As you know, there's thousands of lipids in biological membranes, and Professor Cullis was interested in understanding what the role of these lipids are in the stability of the lipid membrane. And he started focusing on that, and by making simple model membrane systems, he developed vesicles, particles that were able to carry small molecules. And he went on from there to generate new insights into cell membranes and the biophysics of lipid biochemistry, and discovered fusogenic lipids that could improve the cellular internalization and endosomal release of drugs from lipid carriers. He also developed ways to load drugs into lipid nanoparticles, driven by drug diffusion and pH gradient, and use that drug loading principle to get small molecules into the lipid nanoparticles. His work led to the first nanomedicine, which was Doxil. And since then, he's directly developed 3 drugs that have been approved by regulatory agencies in the US and Europe. And there's 7 more from his laboratory that are in clinical trials. And I think it's fair to say that his work has also led to the RNA vaccines that have saved countless millions of lives, and just a spectacular research effort by Peter Cullis. He's also a tremendous entrepreneur. He's co-founded 10 companies, among them Precision Nanosystems, and PNI supplies the machine that we use in the RNA Therapeutic Center and in the GMP facility to generate lipid nanoparticles that carry RNA. Professor Cullis has published over 300 papers. He holds over 60 patents, and He will be talking today about his work with lipids, lipid nanoparticles, and the COVID-19 vaccine. We have some prepared remarks for you. After his video talk, we're going to have him here in the studio for your questions. So, Professor Peter Cullis.
Hello, and thank you very much for the invitation to present at this Kostas conference. Just want to say a few words about what I'm going to talk about today. The title is Lipids, Lipid Nanoparticles, and COVID-19 mRNA Vaccines. Quite a broad spectrum of studies that I'll cover here. I do have a few conflicts of interest. Precision Nanosystems, I'm a co-founder. Acuitas Therapeutics, again a co-founder, and Nanovation Therapeutics, co-founder and chairman. Now, what I'm going to cover today is just to briefly cover a bit about lipids and lipid nanoparticles, and particularly formulation of nucleic acid polymers into lipid nanoparticles. A bit about the clinical validation of these systems, the initial clinical validation, which concerns a drug called Onpattro, which was approved in 2018, and then subsequently, very briefly, to talk about the COVID-19 vaccines, such as Comirnaty or BNT162b2, the Pfizer-BioNTech vaccine that resulted from these studies. Now, the efficient encapsulation, this is the step one in making a lipid nanoparticle. The efficient encapsulation of nucleic acid polymer in a lipid nanoparticle requires cationic or positively charged lipids. Fairly obviously, you've got a negatively charged polymer in the form of, say, RNA or DNA. And in order to get reasonable levels, reasonably efficient levels, then one has to complex that with the lipids of opposite charge, these cationic lipids, to form this hydrophobic complex, as you see here. Now, there's no cationic lipids in nature. They're very toxic molecules. They're only net neutral lipids or negatively charged lipids. So what we did, and this goes back to about 1995, 2000, is we employed what we term ionizable cationic lipids. And so these lipids have the properties of tertiary amine, as you can see here for this lipid called DODAP. And it has this property that at pH 7.4, it's neutral pH, it's relatively uncharged. However, if you go down in pH, below the pKa of the lipid, then it becomes protonated and positively charged. So it turns out that this molecule is much less toxic than permanently positively charged cationic lipids. So just to reiterate, we developed these ionizable lipids near neutral at physiological pH. And what we found, and that's what I'll indicate in a couple of slides from now, that we could load nucleic acid polymers at the low pH, at say pH 4, which is say the acidity of a lemon, so it's not that acid. And we found that the contents were retained at physiological pH values. Much less toxic, as already mentioned, and it had important ramifications in terms of intracellular delivery because these ionizable lipids appear to greatly facilitate the delivery inside the cell into the cytoplasm of the cell. So we devised a rapid mixing procedure to encapsulate these nucleic acid polymers in the lipid nanoparticles, basically dissolving them in water at pH 4, and then the lipids dissolving them in ethanol, organic solvent, but ethanol, one that's soluble, immiscible in an aqueous environment. And then very rapidly mixing these solutions. As you raise the polarity, one of the first things to fall out of the solution will be the positively charged lipid, the ionizable lipid that's associated with the nucleic acid polymer, obviously a pretty hydrophobic entity. If you do this rapidly enough, they'll get coated with other lipids that we incorporate, these structural lipids in this system. This is a PEG lipid, and you can generate what we term limit size systems where the PEG coating lipids on the PEG coating basically stay on the outside, and they have a hydrophobic interior, the limit size being the smallest size that's compatible with the various components, lipid components that one has present in the system. So you then dialyze away the ethanol, raise the pH to 7.4, and you can achieve effectively complete encapsulation with the sRNA being retained at pH 7.4. They're really a new class of lipid nanoparticles with a hydrophobic core, as opposed to an aqueous core. Lipid nanoparticles known as liposomes have an aqueous core. They're ideally suited to encapsulation of these negatively charged macromolecules. High encapsulation efficiencies. We can adjust the diameter in a very straightforward manner by varying the surface lipid to core lipid ratio over the range of, say, 20 to 100 nanometers. Relatively non-toxic. It's a scalable process and very reproducible. Okay, so this is how one gets to the lipid nanoparticle itself. How about the clinical validation that these systems are actually useful for some application? So what I'm going to talk a bit about now is the Onpattro story. This is a series of investigations that went on from about 2005 to 2012 to develop an sRNA-based lipid nanoparticle drug to treat a condition called hereditary amyloid transthyretin-induced amyloidosis. So the question that we faced back in about 2000, well, early 2000s, was can we develop a lipid nanoparticle delivery system that takes sRNA to the liver and enables gene silencing in hepatocytes? So this requires packaging, obviously, which we've done using the ionizable lipid. And then this has to be injected intravenously, get to the liver, extravasate from the vasculature into the cells in the liver, and actually be delivered then to the cytoplasm of the hepatocytes. So a pretty formidable challenge when you consider that nature does not want foreign genetic material to be to gain access to the cytoplasm, of cells. And so this is our objective, as I say, back in the early 2000s. There's many diseases that can potentially be treated by silencing or expressing or editing genes in the liver, as I'm sure everybody is well aware, everything from blood clotting disorders, metabolic disorders, liver cancer, hepatitis, etc. So we started with the question, can the very simple systems that we generated using the ionizable lipids, can they silence genes in hepatocytes? And so there's no targeting information on these systems. And so this is our the simplest system that could come up with that may have a chance. And so what we used was a in vivo assay that for the potency of LNP sRNA systems. This is a factor 7 mouse model. Factor 7 is made in the liver, and so if we silence it using an sRNA, we can then they can assay the blood for factor 7 subsequent to that, and see how well we're silencing the production of factor 7 in the liver. So we're injecting the LNP containing sRNA to silence factor 7 intravenously, and we're then assaying in the mouse model. We're then assaying for factor 7 in the blood some 24 hours later. What we found was that the potency of these lipid nanoparticle systems containing sRNA was very sensitive to the species of the cationic lipid that we employed. And so, particularly a remarkable dependence on the pKa of the ionizable lipid, the apparent pK, where the most potent lipids have a pKa of approximately 6.4. And you can see here how the potency varies as a function of the pKa. It's really quite remarkable. We can easily move from something that, and this is a log plot on the y-axis, something that's is over 100-fold less potent by having a pKa that's only about half a unit away. Here we're defining potency as 1 over the effective dose at which we would see 50% gene silencing of the factor 7. So we synthesized, screened well over 300 different ionizable cationic lipids in the course of this work. This was done with Alnylam Pharmaceuticals based in Boston, and then arrived at a lipid we call MC3 that was the most potent lipid when incorporated into the lipid nanoparticle system. And this just indicates the process that we went through over these 7 years of optimizing the system, starting off with a system that basically we could see gene silencing in the liver, but only at very high doses, 10 mg per kg, and moving down to systems where the potency was 5 micrograms per kilogram body weight. So here without increasing the toxicity of these systems, if anything, decreasing the toxicity. And so this is where we get our 1,000-fold therapeutic index. Now, the application, what Alnylam's clinicians decided was that a good demonstration of the potency of the system was a condition called hereditary amyloid transthyretin amyloidosis. And this is a rather nasty disease, a hereditary disease that arises from extracellular deposits of transthyretin. The mutations in the transthyretin gene can result in formation of these fibrils, which are deposited everywhere in the body, and but have particularly nasty ramifications in nervous tissue and in heart tissue, leading to neuropathy and heart failure. There's really no effective therapy, usually fatal within 5 years of diagnosis. You can see here that it's a wasting disease, particularly nasty for people that are diagnosed with this disorder, as they would be, say, in their 30s, as they would have seen a relative go through this process as a result, of course, of it being a hereditary condition. And so the precept here, the hypothesis, is really quite simple. To use a LNP system containing a transthyretin or siRNA to silence transthyretin, and thus reduce the levels of circulating TTR tetramers, reduce the deposition of the fibrils, and maybe even enhance clearance and rectify the disorder if you get very lucky. But in any way, you can see this as a potentially simple solution to what is obviously a devastating disorder. So the phase 1 study results that were undertaken in healthy volunteers indicated that for these individuals, there's effective gene silencing at dose levels of about 0.15 milligrams per kilogram, as can be seen here, with levels of gene silencing approaching 80 to 90%. Relatively quickly after IV administration and lasting for 2 or 3 weeks. So on the basis of this data for the clinical studies, a dose of 0.3 mg per kg every 3 weeks was chosen for subsequent trials. This very rapidly progressed to rapidly in the usual clinical context, to a phase 3 study. The drug was called patisiran at this stage, and became Onpattro when it was approved. The design here, there was 225 patients. They were divided into placebo group and a group getting the lipid nanoparticle formulation of the siRNA to silence transthyretin, administered at 0.3 mg per kg as already indicated, or they get sterile saline as the placebo. And we're monitoring here a change in the neural impairment score at 18 months. There's various secondary endpoints, weakness, quality of life, ability to walk, body mass index, etc. Now, the clinical trial results were announced in September of 2017. The primary endpoint was hit, and all secondary endpoints were also achieved. The p-values here, you can see that this is really quite remarkable, 10 to the minus 24. Really don't need to do the stats on this. The data indicates these individuals were getting better over the course of the treatment. In other words, reversing the course of a hereditary condition. these from the point of view of the neural impairment score, again, quality of life, muscle strength, ability to walk, nutritional status, etc., were all maintained stable or improved, and most were improved. And so therefore we've developed a drug here that's potentially a curative therapy for what is a previously fatal disease, and it certainly gives clinical validation to the lipid nanoparticle technology. So this was approved by the FDA in 2018 for treatment of this condition, the first FDA approval of an siRNA-based gene therapy drug. We would contend this is a pretty big deal, not only being able to halt the progression of a hereditary disease, but actually reversing accumulated damage. And this is something that is a general feature of gene therapies that they have can have quite remarkable power and targeting capability as compared to, say, small molecule drugs. So moving on now to the COVID-19 vaccines, I'll just talk very briefly about the Comirnaty or BNT162 story. This is the Pfizer-BioNTech vaccine. And first of all, we had to determine whether the LNP technology enabled mRNA gene therapies. And just to point out the differences in size here, siRNA versus, say, a 2 kilobase luciferase, which is a much larger molecule. Does the ethanol dilution and rapid mixing process allow efficient encapsulation? And are these lipids, such as MC3, effective for delivering mRNA to produce protein in hepatocytes? Now, the formulation process turned out to be equally applicable to mRNA as it was to siRNA, as is indicated here as cryo-TEM with a 100 nanometer bar. And the what's progressed since then is that one of the companies I co-founded, Kostas, has screened over 300 ionizable lipids using a luciferase model for gene expression. Gene expression, obviously, of the luciferase in the liver to gradually improve the characteristics of these systems. There are now some actually 30-fold, 40-fold improvement in gene expression in the liver compared to, say, MC3 containing systems to the newer systems that have been developed by Kostas. This has a great many applications. As an example here is expression of IgG following an IV administration of LNP systems containing mRNA coding for the IgG. And as you can see here, we're getting very high levels of plasma monoclonal antibodies following this IV administration. However, I'm not going to focus on that, just to say that there's a serendipitous event about 2014. We were approached by Drew Weissman of the University of Pennsylvania, who needed a delivery system to enable mRNA vaccines. And Drew had worked for many years with Catalin Carrico to explore the potential of mRNA therapeutics as vaccines. But they came to Kostas saying, we have a delivery problem. How do we get mRNA coding for viral proteins into muscle and immune cells in vivo? And of course, what Drew needed was a system that would gain access to muscle cells and antigen-presenting cells to stimulate robust MHC1 and MHC2 class immune responses, with the hope of getting a good protection against the particular, here you'd have mRNA coding for, say, a viral protein and get good protection against infection by that particular virus. So the first series of studies were done on Zika virus. And this consists of a lipid nanoparticle containing mRNA coding for the Zika virus pre-membrane and envelope glycoprotein, and was published in Nature in 2017. The results here were quite spectacular, indicating total protection against the Zika virus. Here, of course, we're injecting the LNP containing the mRNA coding for the membrane protein, in this case intradermally. and then challenging at 2 weeks or 20 weeks with the Zika virus itself. So this indicates the results. If you had luciferase as the mRNA or the mRNA coding for luciferase, of course, you get infection. Whereas for the system containing the mRNA coding for the Zika virus membrane protein, there's total protection both at 2 weeks and at 20 weeks. I just went the wrong way there. So in January of 2020, as a result of the studies with Drew Weissman, Kostas had partnered with BioNTech in Germany to develop a flu vaccine. But of course, when the pandemic hit, they immediately switched to see whether or not one could develop a potent vaccine against COVID-19. So all efforts switched in the January, February timeframe of last year. And here we have developing a system with mRNA coding for the SARS-CoV-2 spike glycoprotein. And as you're all aware, the phase 3 study, which was announced in November of 2020, indicated 95% effectiveness across all age and gender and race groups, reasonably well tolerated, and indicating here expecting to produce up to 50 million doses. I think now the dose, instead of being 1.3 billion, is now closer to 3 billion, and they're producing at the rate of 50 million a week. So this was approved by many regulatory agencies, as we're all well aware, and is now probably one of the most successful vaccines that the world has ever seen. Now, I just want to say a bit about the future, the extremely rapid and precise way in which these lipid nanoparticle gene therapies can be developed. Once you identify the protein to be silenced or expressed or edited, you can produce the siRNA, the mRNA, in a time on the order of a month or so. Encapsulation takes a day or so. So you have a new medicine that is available in time on the order of weeks. And so the possibility now exists for a great variety of vaccines, not just vaccines, but many other applications, rare disease, et cetera. There's going to be a few issues that are associated with these applications, but the future looks very bright. So this is the story that's gone on over the last 25 years, really starting off with formulating these nucleic acid polymers into these LNP systems, but recognizing we couldn't use the permanently positively charged lipids, the ionizable lipids that have really kind of revolutionized this approach, and having tremendous applications in a variety of diseases. These studies involve literally hundreds, probably thousands of people. I just want to mention Mick Hope and Tom Madden, who I've worked with for over 40 years, Steve Ansel, Ying Tam, Barb Ngui, Paul Lin, Sean Semple, more than 20 years, and many others that are indicated here, particularly the people at Alnylam, Mark Tracy, Akina Kink, Martin Mayer, and Nana Manoharan, as well as people from my group, and of course, Drew Weissman from the University of Pennsylvania. So with that, I'll close and take any questions. Thank you very much.
Peter, that was spectacular. Thank you so much for a wonderful, wonderful talk, and congratulations on a spectacular career. Thank you for joining us in the studio, in the DeBakey studio today, Peter, and what a wonderful career you've had.
Yeah, thanks very much. Can you hear me?
Yeah,
all
I can
right?
hear you. We can hear you fine.
Okay.
I got a few questions for you, Peter. I wonder if you could tell us a little bit about targeting lipid nanoparticles. You've been focusing on targeting the liver. What about how are we going to be able to target other organs for gene therapy, for RNA therapies, the heart, the brain? Do you have any thoughts about how the lipid nanoparticles could be modified so that you can target other tissues and organs?
Yeah, there's going to be this is going to be a bit of a stepwise approach. The first step, I mean, right now we're limited to the liver. Actually, we're taking advantage of a biological process here that they absorb ApoE and use that as a way to get into hepatocytes. They're just taken up the same way that chylomicron remnants are taken up. But in terms of getting to other tissues, what we need are transfection competent systems that circulate for much longer times, and so that's been a bit of a barrier. We're getting there. I think the first tissues you're going to see us hitting, other than the liver, are going to be bone marrow, then subsequently perhaps the lung. Getting to the brain is going to be difficult. Although there are ways of temporarily affecting the permeability barrier between the blood and the brain, these are all in their infancy, of course. Direct injection is another possibility. The interesting thing, once we achieve that, and I do think that over the next 10 years, whatever 5 to 10 years, we're going to see that happening, will then be to say, okay, well, what we really want is for these systems to be transfection competent to practice gene therapy in a particular area, so that we're not transfecting the whole of your body. Getting systems that's where you can turn off gene therapy in particular locations, perhaps using external radiation of some form. These are topics that a great many people in the field are really tackling at the moment. One of the issues that we do face, though, is that putting targeting information on the outside, such as a monoclonal antibody, has proven very difficult over the years, and so that I don't see that going away anytime soon.
That was great. That was a great answer. What about lipid modifications of the lipid membrane? Are there differences in cell preferences for lipid membranes, for the lipids in the lipid membrane?
Yes, I mean that's. But one of the things that we don't understand very well at the moment is, of course, when you put something in the blood, it changes its characteristics. And as I mentioned, there's the ApoE effect that causes uptake into hepatocytes. So all these nanosystems, when you put them into the circulation, they absorb proteins, serum proteins. And this will very directly affects their properties in terms of being able to be either circulate for a long time or be taken up by various tissues. And so I think that yes, altering the lipid composition, the sugar composition of the external surface of these nanoparticles is going to dramatically affect both their affinity for certain tissue types and their ability to transfect certain tissue types. So these are it's a bit of a gold rush, really. People are moving as fast as they can to investigate those sort of possibilities.
Peter, you mentioned the RNA vaccine, and clearly that's been a huge accomplishment for you and the people that have been working on RNA as drugs. There have been some, although the vaccinations are very safe and very effective, there have been some side effects reported, and there's been some question as to whether the lipid nanoparticles may be playing some role in the side effects that can occur, albeit rarely, with the COVID-19 vaccines. Do you want to comment a little bit about side effects in COVID-19 vaccines and generally lipid nanoparticles?
Yeah, in terms of an intramuscular injection, the side effects are pretty minimal. There does seem to be perhaps in terms of immune response that's a little too vigorous, shall we say, on injection. A slight increase over what might be seen from a vaccine that's based on a viral vector. But that some people have ascribed that to the PEG coating, to the polyethylene glycol coating that we have with these particles, which is a possibility. We designed that PEG coating so that it does come off, so from the exterior of these systems, so they can become transfection competent. But it is possible that that's playing a role in some of these rather rare, very rare, immune responses that can occur when you on intramuscular injection. I should mention that these systems, I mean, we're giving only, say, for the Pfizer-BioNTech vaccine, it's only 30 micrograms of the mRNA in the lipid nanoparticles. The actual dose is very small, particularly compared with, say, Onpattro, where the dose is well, I guess I should do the calculation, but maybe 1,000-fold higher, and that's repeated every 3 weeks, of course. And so in that cohort, these patients have done very well. There does not seem to be undue toxicity arising from these systems, and they have a very similar lipid composition, as I was pointing out.
That's really good news. We're having a little bit of a technical problem in getting questions from the audience. So I know you're going to be speaking to us a little bit later, Peter, when we talk about the future of cardiovascular nanotherapy. And at that time, maybe we can also take some questions. By that time, we'll have our technical issues figured out, and we can get some questions from the audience to you.
Okay, sure.
Any parting words?
Well, no, it's just a very exciting time in therapeutics. We're now getting to a point where the power of gene therapy is available to us, and so all of the molecular biology that has been, understanding that's been developed over, the last 50 years, we can now use much more, very directly, in terms of deciding new therapeutics. So that, the siRNA story for the Onpattro product for the trans therapy, there's a very simple, if you knock out that gene, if you don't have that pathological protein being expressed, then you have a dramatic effect on the disease. And similarly for the vaccines, of course, they're very straightforward ways of designing new medicines. So I think we're really into a very exciting time as far as therapeutics are concerned.
You just triggered another thought, and one of the questions I think is coming to the fore is, are there other ways to generate nanoparticles, perhaps endogenously, that could replace artificial LNPs? For example, exosomal approaches. Do you think there's any future there for exosomal delivery?
Oh, definitely. The problem, of course, with exosomes, as is with viruses in a way, viral vectors in a way, they're really quite complicated. I mean, in many ways, the lipid-based systems are mimicking biology, but we're using the simplest, the most basic parts of the lipid, the membrane. And so, we have bilayer. We use naturally occurring lipids to a large extent in these systems. But yes, we're going to learn a lot from the biology of these more complicated systems and how it is that perhaps they can get material more effectively to one area of the body or another. And maybe we can mimic that in a artificial lipid nanoparticle system, or maybe we find that for some applications, the more complex systems work better.
Mm-hmm. And would that mimicking include the use perhaps of membrane proteins or other recombinant proteins as ligands for a particular target tissue?
Yeah, I mean, it's tempting to do this. I mean, if one can get like low density lipoprotein, for example, it gets pretty much everywhere in the body. It'll be rather, it would be a great thing to be able to put that onto the surface of one of these lipid nanoparticles. It's just that reconstitution of some of these hydrophobic proteins is a bit of a trick, and I wouldn't say that we've mastered that yet. But yes, I mean, using biology very directly in that way is a really attractive way to go.
Well, thanks again, Peter, for a wonderful talk, and congratulations on a even more wonderful career. And we look forward to your comments about the future of cardiovascular nanotherapeutics later this afternoon. Thanks again.
Okay, thank you very much.
Well, with that, we'll move on to our next speaker. who's coming to us from Cedars-Sinai Medical Center. That's Professor Eduardo Marban, who's the Mark S. Siegel Family Foundation Distinguished Professor and Executive Director of the Smith Heart Institute at Cedars-Sinai Medical Center. And Dr. Marban is one of the leaders in cardiology. He's done pioneering work in two broad areas: biologically based therapies for cardiac regeneration and biological pacemakers. His lab was the first to generate a biological pacemaker. They recognized that TBX18 is a transcriptional factor that's a master regulator of pacemaker phenotype. So by overexpressing the TBX18 in cardiac myocytes, he converted them to pacemaker cells. And that work went from the basic lab into large animal models and is now in first-in-human clinical testing. The second broad category of the Marban laboratory is CDCs, cardiosphere-derived cells. So his laboratory discovered these cardiosphere-derived cells that are innate adult progenitor cells in the heart that secrete angiogenic and regenerative factors. And they took that finding from fundamental studies at the bench into large animal models, and then 5 clinical trials in patients with heart failure or Duchenne muscular dystrophy. His discoveries in gene and cell therapy have formed the foundation for multiple startup companies. Dr. Marban has had many awards and honors, such as the Basic Research Prize of the American Heart Association and the Distinguished Scientist Award of the American Heart Association. He was editor-in-chief of Circulation Research for many years. It's our primary cardiovascular research journal, and we're very pleased to have him here today. He'll be talking about mining active principles from extracellular vesicles.
Good morning. It's my pleasure today to talk about mining active principles from extracellular vesicles. The clinical target in this talk is an entity known as heart failure with preserved ejection fraction, or HFpEF. And it's a weakness of the heart muscle in the sense that patients get exercise intolerance and shortness of breath, as they do with regular heart failure. But when we measure how well the heart is beating using a conventional measure by echocardiography known as ejection fraction, it seems to be normal. It turns out that more than 50% of all the patients who are diagnosed with congestive heart failure have this entity called HFpEF. And its mortality is high. The patients are hospitalized frequently for acute decompensated heart failure. They have shortness of breath and exercise intolerance, as I've said. And unlike the more conventional form of heart failure with the dilated heart and a low ejection fraction, there are no effective treatments for this entity known as HFpEF. So it's been called the largest unmet need in cardiovascular medicine. Now, today's talk, if it were being presented in an ideal and fantastical way, would take us from the clinical features of HFpEF, as I've just review the effects of cells called CDCs that I'll tell you about, and then through predictable and iterative steps lead to the development of novel RNA drugs mined from extracellular vesicles. But in fact, the reality was a nonlinear and unanticipated drug discovery and development paradigm, which led to bio-inspired new chemical entities as the basis for next generation therapeutics, beginning with the paradigm of autologous cell therapy. And the cells that we wanted to use are ones we first described called cardiosphere-derived cells that are cardiac stromal cells with progenitor cell features. They have a distinctive antigenic profile with uniform expression of CD105 and lack of expression of hematogenous markers such as CD45. These work by secreting extracellular vesicles. These extracellular vesicles are angiogenic, antifibrotic, anti-inflammatory. They promote cardiomyogenesis and cardiomyocyte survival, and they're also immunomodulatory. And the diversity of these actions is rationalized, at least partially, by the diverse contents of extracellular vesicles, which I'll tell you more about in this talk. Now, CDCs themselves, the parent cells, have been used in several clinical trials of heart disease for various indications: ischemic cardiomyopathy, dilated cardiomyopathy. Duchenne muscular dystrophy is among the most successful clinical programs to date. It's gone to phase 2 and is now in the planning stages for a phase 3 trial for advanced Duchenne muscular dystrophy. And then 2 ongoing trials, one for pulmonary hypertension and another for HFpEF. So we studied the effects of CDCs in a model of HFpEF, and that finding has some mechanistic implications. In this publication, we implemented the dull salt-sensitive rat model of HFpEF. These rats, when they're fed low salt, have a normal phenotype. But when they're fed high salt, develop hypertension and other signs of heart failure preserved ejection fraction. And after already developing heart failure preserved ejection fraction objectively, we randomized the group of trials fed with the group of rats fed with high salt into a group with just vehicle phosphate buffered saline or phosphate buffered saline, including cardiosphere derived cells, CDCs, and then continued to an endpoint 4 weeks later. As one measure of diastolic dysfunction, or the failure of the heart to relax completely during beats, which is a cardinal feature of HFpEF, we measured this echocardiographic ratio called E to A. This goes down in HFpEF, and in the animals that were fed with placebo, it remained down. But in animals that got CDCs, it went back up to control levels and in fact approximated the levels in normal animals. Throughout the experiment, the ejection fraction remained normal, which is a hallmark of this HFpEF disease. Nevertheless, the animals that got only the placebo developed pulmonary congestion, as evident from this increase in lung weight over body weight, which was reversed by CDC therapy. And CDCs apparently had a survival benefit. The normal salt rats had no mortality. More than half of the animals died during the protocol if they did not receive CDCs, but those who received CDCs seemed to have a mortality benefit. Nevertheless, there was no effect on blood pressure or hypertrophy, but there were changes in inflammation measured either by serum inflammatory cytokines. For example, look at the changes in TNF-alpha, which went up and was partially restored by CDCs. And that pattern was true for all these other markers, as well as for histology for macrophages and leukocytes in heart tissue, which went up in placebo and back down in CDCs, and likewise CD45 for all white cells. Fibrosis was also attenuated by CDCs. Look at these picroceria stains, which representatively look like they're normalized by CDCs, and in the pooled data, this was confirmed. So mechanistically, one of the prevailing and sort of old-fashioned hypotheses for HFpEF is that it was simply a mechanical consequence of an increased afterload or blood pressure and intense hypertrophy. But we now know that these things were unchanged in the CDC-treated animals. There was no decrease in blood pressure, nor was there a change in the size, the thickness of the heart. But these changes in inflammation and fibrosis were reversed with CDCs, meaning that HFpEF and inflammation are likely primary, and this is consistent with a more modern hypothesis of HFpEF pathogenesis summarized schematically here. Well, what about exosomes? It turns out that these are extracellular vesicles that are products of a specific biosynthetic pathway involving ceramide. And if you use the ceramide inhibitor, GPW4869, exosome secretion is inhibited. These exosomes are released by all eukaryotic cells, and they have a modal diameter of about 100 nanometers. So why do we find exosomes to be of interest in the mechanisms of CDCs? Well, first of all, on the top panel here, you find that exosome production is necessary for the benefit of CDCs. And in the bottom panel, exosomes in the absence of CDCs completely can reproduce the benefits of CDCs. So let me walk you through this so you understand the evidence. This is a trajectory of left ventricular ejection fraction in mice that have undergone a myocardial infarction. A normal mouse has an ejection fraction up here of about 70 to 80%. So these animals, after a coronary artery is tied off 1 day later, have a very substantial decrease in ejection fraction. But if they're injected before this day with CDCs, they regain part of their ventricular function over time. However, if animals are injected with CDCs that had been pre-incubated in GW4869, the inhibitor of ceramide that I talked about that inhibits exosome biosynthesis, these cells were injected and were alive but had no benefit. Now, if you simply take cells out of the equation and just use conditioned media from which you isolate exosomes, either by spinning it down or by precipitation, in this case by precipitation, we found that the exosomes in the conditioned media, conditioned by CDCs, had just the same benefit, or the same kind of benefit, as if you had injected cells, but there's no cells injected here, just the exosomes from conditioned media. And this is very different from the trajectory shown in blue of just injecting vehicle, but not all exosomes are the same. So if you use conditioned media that's been exposed to fibroblasts, in this case human dermal fibroblasts, those exosomes, even though they're equivalent in number to the ones injected here, had no benefit at all on function over time. So given this kind of information, we wondered whether some of the effects of CDCs might be reproduced by exosomes, and we decided in this case to give them intravenously, because these tiny particles will circulate throughout the entire body and should, in principle, be effective if you get enough of them to the region of interest. So again, we used the dull salt sensitive rats, and after 7 weeks, the controls received low salt. The animals that were on high salt were randomized after having developed a HFpEF phenotype into just vehicle or into injections every week of CDC exosomes intravenously. And we can see that ejection fraction is unchanged in all of the groups, as it should be if the diagnosis is indeed HFpEF. EE prime, which is another indicator of diastolic dysfunction, which in this case goes up if the heart becomes affected by HFpEF, was normal in the control group, went up in the placebo group, and normalized somewhat in the animals that got treated with CDC exosomes. And likewise, lung weight over body weight went up in the placebo group, but was normalized in the CDC exosome group. So exosomes are the source of a Rosetta Stone inventory of non-coding RNAs, and we have not neglected the opportunity to mine those exosomes for defined factors. And much of the attention in the past has gone to microRNAs, such as microRNA 146A, microRNA 181B, both of which we've implicated in cardioprotection from myocardial infarction. MicroRNAs are attractive to use as candidate effectors in exosomes simply because we know how they work and they're good tools for dissecting their mechanisms of action. But they're actually not very plentiful in exosomal content. Other non-coding RNAs, such as fragments of the human yRNA4 gene, are actually much more plentiful. So we, in another set of studies, characterized the leading such fragment, which is called YF1, and showed that it could act on macrophages to also increase IL-10 and produce cardioprotection. And as I was saying, the percentage of microRNAs in the among the small RNA contents of CDC exosomes is only 7% of the pie, whereas these yRNAs that by and large have unknown function are at least 2.5 times more plentiful. And the single most plentiful individual species is this yRNA fragment called EVYF1, which we characterized in a myocardial infarction model, in an angiotensin II overload model, and more recently in a published paper on hypertrophic cardiomyopathy. So this entity called EVYF1 is a 56 nucleotide 5' fragment of the human yRNA4 gene, and on its own, when put into macrophages, we believe induces epigenetic modifications that lead to sustained changes in gene expression. This is our working hypothesis. And here shown is the global transcriptome data. And you can see that there's a qualitative difference between vehicle exposed macrophages and EVYF1 exposed macrophages. And among the prominent changes were increases in IRF7 and interleukin 10. And as I've already told you, we know that EVYF1 increases IL-10 protein levels in the secreted media. And also blocking IL-10 undoes the benefit in models of myocardial infarction. But we also have found that it works in hypertrophic cardiomyopathy mice. These are transgenic mice mimicking a human mutation in troponin I, and they spontaneously develop hypertrophic cardiomyopathy. At the time that they've already developed the hypertrophic cardiomyopathy, we either randomize them to injected vehicle intravenously or EVYF1 in a transfection reagent twice a week. Nothing much happened to body weight over time, but if you can look at these measures of ventricular thickness, which is increased in this model of hypertrophic cardiomyopathy, interventricular septal dimension and left posterior wall diameter or dimension, you can see that the vehicle treated animals got worse, while the YF1 animals got progressively better over 4 weeks, and likewise for posterior wall dimension. The animals looked healthier in terms of coat, color, and texture. They could run longer on a treadmill, and their heart rate to tibial length ratios were normalized. So EVYF1 we now understand is an anti-inflammatory, cardioprotective, and anti-hypertrophic molecule. It's mined directly from the exosomal contents and unmodified. It's a natural nucleic acid structure, 56 nucleotides unmodified. But could it be helpful as the bioinspiration or a template for better new chemical entities? And one of the motivations for creating new chemical entities is the fact that RNA drugs, such as these FDA-approved RNA drugs, are typically 18 to 30 nucleotides long. They're chemically modified for potency and stability, and they're uniformly given parenterally. And they tend to be in several families, either aptamers or antisense oligonucleotides, siRNA, and this is also an antisense oligonucleotide for Duchenne muscular dystrophy for exon skipping. We do not conceive of EVYF1 or any of its derivatives as working by any of these known mechanisms. So it would be a new member of a new drug class, but we wanted to mimic the success in getting to the clinic by perhaps truncating EVYF1 and chemically modifying it. So we started with YF1 as an inspiration, truncated it, and experimented with several point mutations and used locked nucleic acid chemical modification to create a new entity, TY4, which is a bio-inspired 24 nucleotide mutant locked nucleic acid modified small yRNA. And we tested this in another model of HFpEF. This is the two-head HFpEF mouse model, and also in a model of rat acute myocardial infarction. And although RNA drugs are said to be effective only parenterally, we tested both intravenous delivery and oral delivery, taking a page from our recent findings that exosomes can be absorbed orally, but that uptake of exosomes into the circulation can be enhanced by coating the exosomes with the milk protein casein. So here's evidence of intravenous efficacy in the HFpEF two-head mouse model. 5 weeks after initiating hypertension and obesity using a combination of oral LNAME and high-fat diet, we randomized the animals to receive either this new entity TY4 in a transfection reagent DARM effect every twice a week intravenously, or vehicle twice a week intravenously, and measured ejection fraction, which remained high and equal in all groups. However, the running distance went way down in the vehicle treated animals, but remained normal or was restored to normal. in the TY4 treated animals. Likewise, this indicator of diastolic dysfunction by echocardiography, E over E prime, went up in vehicle as it should in HFpEF, but was down again to normal levels in TY4. And likewise, this biomarker, circulating biomarker of heart failure, BNP by ELISA, went up in vehicle but back down in TY4. So 3 different indicators of efficacy. And in this myocardial infarction model, induced now in a rat by opening up the chest with a left-sided thoracotomy, tying off the left coronary artery for 45 minutes and then allowing reperfusion. And 20 minutes later, by either gavage orally or intravenously, we administered TY4 or vehicle. And then we followed the animals for 48 hours and quantified infarct size histologically. And we were quite gratified to find that the size of the infarct was about half in animals that received intravenous TY4, but even more surprised and gratified that it worked equally well when given orally. This is evident both in the pooled data as well as in the representative TTC stained images below. So next steps in developing this new chemical entity, bio-inspired from a mind factor within extracellular vesicles, will be to work further on the mechanism of action, including the identification of binding partners, and we're working on that right now, measurements of biodistribution, particularly after oral administration. We're going to optimize the dosing and the formulation, and then implement clinical trial grade but scalable manufacturing with which we will perform long-term tox studies, draft a clinical protocol, and proceed to regulatory filing so that we can do first-in-human studies. So just to summarize, this nonlinear unanticipated drug discovery and development paradigm started with wanting to use autologous cell therapy for the heart to try to heal it by actually transplanting cells in that would become new heart muscle. We recognized that there were benefits that were durable despite the fact that the cells didn't last for long. And this led to the allogeneic paradigm, the use of unrelated donor cells. When we explored the mediators, we identified exosomes to the mediators, which led to the concept of cell-free therapeutics. And there's already a filed regulatory filing to use CDC-derived exosomes as a therapeutic before FDA. We may do first clinical studies next year. Exosome mining to identify defined factors in the contents. It started in when we first gleaned that exosomes were important and started figuring out what was in them, and now continues. And this creation of new bio-inspired chemical entities is brand new and is hopefully going to lead to a next generation therapeutics. I'd like to acknowledge the folks who did the work that I've shown here today, and a special appreciation to my colleagues locally and elsewhere, as well as these funding agencies. And finally, this is where the work was done at Cedars-Sinai, and from which it's my pleasure to present today. Thank you very much.
Dr. Marban, what a spectacular talk. I really enjoyed that. Thanks for joining us in the DeBakey studio today at the Kostas meeting. Really a pleasure to have you here. Well, that's the first time I've heard a drug discovery program described as nonlinear, unanticipated drug discovery paradigm. But it's a great story. Certainly, I mean, there's been a thread throughout all of this. Your work is driven by the fact that you're an active cardiologist, you're taking care of patients, you're directing one of the best heart centers in the country, and that's always has been driving your work, it seems to me.
Well, John, I'm glad that the money I sent you by Zelle to make such nice comments actually was worth it. So thank you so much. All kidding aside, one of the great things about being a practicing physician, not necessarily just a cardiologist, but a practicing physician, is that you can rely on your own firsthand knowledge to try to decide what the great unanswered questions are. rather than have somebody else tell you about it. And even though I am, and that certainly has been a great benefit, but even though I am a cardiologist and very proud of that, I've also been forced by the fact that the processes being addressed here are so fundamental to go back to medical school and try to understand, try to remember, things like scleroderma and lupus and pulmonary fibrosis and all these other diseases that these next generation therapeutics might be bioactive against, because there's no reason to believe if you're targeting something as fundamental as inflammation and fibrosis or immunomodulation that it would necessarily be limited to the heart, as you well appreciate.
I've got a lot of questions for you, Eduardo. I do want to point out to everyone that if you go to the stage, which I'm on right now, there's a chat feature and there's a Q&A feature. I'm actually looking at the Q&A feature right now, so if you have any questions for Dr. Marban, this is your chance to talk to one of the leaders in cardiovascular medicine and biology. This yRNA that you found in the CDC exosomes, it increases IL-10, so it's anti-inflammatory, and it has anti-fibrotic effects. What is the mechanism by which yRNA is working here? Is it acting as a transcriptionally increasing the production of IL-10 somehow? How is it having this anti-inflammatory effect? What's its molecular mechanism?
Well, before we started exploring it, not much was known about yRNA in general, even though there are 4 human yRNA genes and something like 26 pseudogenes in the human genome. The only real confidence that we had about their biology is the fact that they served as the RNA molecules that bind to small ribonuclear proteins such as Rho and La. In fact, that's how ribonuclear proteins were first identified, is by probing the binding partners for Rho and La in serum from patients with lupus. But having said that, they were subsequently implicated in a very general sense in DNA stability. They had no known transcriptional regulatory role, but this 56mer, EBYF1, the index compound that we mined directly from the EVs, itself increases transcript levels of IL-10 and in the conditioned media increases IL-10 secreted protein. So the mechanism whereby that happens, we're sorting out now, but I can tell you, and we have some interesting candidate biomarkers or binding partners that give us some clues. Not ready yet for prime time, but I can tell you it's not acting by any of the classical mechanisms we associate with RNA drugs. It's not an antisense molecule. It's not an aptamer. It's not an siRNA, and it certainly doesn't seem to be anything like a mir or an integramer. So
that's exciting. I'm looking forward to hearing more about it. Exosomes also contain other cargo. Is it possible that in addition to this yRNA that there's some other cargo proteins or microRNA within the exosomes that are playing a role in beneficial effects that you observe with CDCs?
Oh, for sure, yeah. We, when you do first of all, there's the whole world of lipids and the whole world of proteins. And we've focused less on lipids and proteins, and not because they're unimportant, but because if you lightly disrupt the membrane enough to allow the contents of the exosomes to be exposed to ribonuclease, but not denature the proteins, you get rid of about 80% of the bioactivity. So we think the money is in the RNA, and these particular exosomes don't have that much by way of coding sequences. It's almost all non-coding RNA. If you just look at the small non-coding RNA family, up to 200 nucleotides, there's more than 10,000 species in this population of exosomes, and we maybe know what 400 of those do. So there's, to say that this is a treasure chest of potential therapeutic candidates is not is not an exaggeration.
Yeah, it sounds like a lot of room for discovery there. I have another question for you. In addition to having an anti-fibrotic effect, you said that there's some androgenic effects of the CDCs. And I was wondering if that might be some of the benefit that you're observing. Is the reduction in fibrosis due to some sort of transdifferentiation of fibroblast endothelial cells? Or, what is going on there? You're reducing fibrosis, you're gaining vasculature. Any thoughts about the processes involved in that benefit?
Well, we know when you do a single cell analysis, in particular transcriptomic analysis of the heart, for example, that many transcripts in many cell types are altered. It's not a magic bullet that only hits, let's say, endothelial cells, but we also know for sure that macrophages seem to play a central role in at least some of the diseases that I explored here, certainly in myocardial infarction and in HFpEF, even though the knowledge of the role of macrophages is still evolving. It's really kind of an interesting paradigm, because using transgenic mouse models, we can now deplete selectively different macrophage populations and then reintroduce them using adoptive transfer. And so we're trying to use those kinds of methods to test the specific hypotheses that maybe macrophages are mediating a lot of these as the initial effectors, because they're, as you know, they're very highly phagocytic cells. And we know for sure, in vitro or in vivo, if you take macrophages that have been exposed to any of these compounds, not only is their transcriptome very altered, but they're functionally altered as well.
Mm-hmm. Thanks. Eduardo, there's some questions popping up here on my screen, and one of the questions is regarding TY4. Do you think it would work if you gave it after tissue remodeling? I think this is referring to after heart failure has ensued. I think you answered that question, but any more comments on that? Does TY4 work after there's been fibrosis, cardiac fibrosis?
Yeah, we in the myocardial infarction model that I presented, it was given 20 minutes after reperfusion, which obviously is too early to reverse an established scar. So there, for sure, we're interfering or modulating the process of fibrotic formation. But in the HFpEF model, we start, at least in the HFpEF model that we've got the most data with right now, which is the two-head mouse model, we start 5 weeks after initiation of the L-NAME and high-fat diet, and by that time, they already have significant dysfunction and fibrosis. And so there, it's more likely a reversal rather than just a prevention.
Mm-hmm. Thank you. I know it wasn't the subject of your talk today, but I was wondering if you could just give us an update on your program with the biological pacemaker. What's the latest development there?
Well, we continue to focus on introducing a single transcription factor there that's an embryonic transcription factor that, at least in the developing heart, is important in the formation of the sinoatrial node and then disappears. If you introduce this factor into the adult heart in a focal way, TBX18, you can get that part of the heart to become basically a pacemaker. The cells not only function as pacemakers, but they look like pacemaker cells, as this is a kind of a reprogramming. That approach, we believe, is still very promising, and the only reason we haven't gotten into patients already is really pragmatic. We've had horrible luck with making clinical grade adenoviral vectors. And adenoviral vectors are actually helpful in this particular setting because we want strong focal expression that then goes away over time. AAV comes on slowly and then stays there, and naked DNA, we haven't gotten to express to the levels that we need to transduce a sufficient number of cells. So we've been working with an adenoviral core that has made clinical grade adenovirus for a number of clinical trials, and it's been years of troubleshooting. So if anybody out there has any great ideas for how we can do transient clinical grade vector for expressing a transcription factor, I'd love to hear from you.
Thanks, Eduardo. Well, thanks again for joining us, and a spectacular talk and a spectacular career. Keep up the great work. Looking forward to hearing more about your wonderful work. Thanks, Eduardo.
Thank you, John. Take care.
Bye now. All right. Well, continuing on this kind of idea about gene therapy-based nanomedicine, our next speaker, Dr. Bruna Corradetti, is going to be talking about RNA-based biomimetic strategies for angiogenesis and wound healing. And Dr. Corradetti is an assistant professor of nanomedicine here. She got her PhD in biomolecular science from Polytechnic University of Marche in Italy in 2012. And there she was working on stem cell-based therapeutic approaches for treatment of spontaneous tendon injuries. And then in 2013, she was appointed as assistant professor of medicine and genetics there. And fortunately, we were able to lure her here. And in 2016, in 2018, she became assistant research professor and also was appointed as associate professor at Swansea University in Wales. She serves as the secretary of the European Society for Translational Medicine. In 2017, she was recognized among the 100 Italian excellences. She's published more than 40 peer-reviewed papers and is the editor of a book entitled The Immune Response to Implanted Materials and Devices, published by Springer International in 2017. Dr. Bruna Corradetti.
Good morning, everybody. It is such a pleasure for me to be here and to be able to join the annual international meeting organized by the George and Angelina Kostas Research Center for Cardiovascular Nanomedicine. Before I go ahead, I would like to thank the Kostas family once again for the kind support they provided me with to develop my vision about the development of RNA-based biomimetic strategies for the treatment of chronic wounds. Also, I would like to thank the organizers for the invitation to share my work with you today. Based on the Center for Disease Control and Prevention, chronic diseases are conditions that last 1 year or more and require ongoing medication attention or limit activities of daily living or both. Chronic diseases are associated to cardiovascular diseases, cancer, and diabetes, and represent the leading cause of death and disability worldwide. They have a significant health and economic cost in the United States, with 90% of the nation's $3.5 trillion in annual healthcare expenditure. Chronic wounds are among chronic disorders, and in the United States alone, non-healing wounds affect an estimated 6.5 million people, and the economic burden for both patients and the medical system is becoming alarming. The great majority of chronic wounds are associated with disorders that are, for example, ulcerative wounds, diabetes mellitus, arterial or venous insufficiency, prolonged internal pressure, and nutritional deficiencies. The potential to regenerate decreases with aging-related chronic health conditions, which further complicate treatments and impair the quality of life. Chronic wounds are often the result of an impaired healing process. The healing process represents the physiological response to an insult or injury and is a well-documented series of cellular and molecular events that result in the deposition of a fibrous connective tissue at the site of injury. The main cause of a chronic wound is a resolved inflammation, which induces a non-migratory epidermis, impairs fibroblast function, and extracellular matrix deposition. It also induces a reduction in the bioavailability of growth factors and diminishes proliferative capacity of resident cells with an insufficient or impaired wound perfusion. The association between the inflammation and impairment of endothelial dysfunction is believed to further exacerbate as the result of the COVID-19 derived cytokine storm, with an impaired recruitment of endothelial progenitor cells to the injury site and an altered cutaneous healing. This scenario justifies the increased interest in finding more efficient therapies and management strategies to treat chronic wounds, or more likely, to prevent wounds becoming chronic. Therapeutic intervention is mediated by cell therapy through the use of bioactive cells like stem cells or immune cells, biomaterials able to mimic the extracellular matrix of tissues when fractions are lost. We have been successful in developing stem cell-based and tissue engineering strategies able to modulate the immune response and achieve functional tissue repair. In particular, biomimetic platforms are developed as biocompatible therapeutics that reproduce the molecular messages and cues from the native tissue microenvironment and modulate the immune response, resolve inflammation, and promote wound healing. In this graph, it showed that by employing biomimetic-based therapeutic intervention during the inflammatory phase, we have been able to tilt the scale from a pro to an anti-inflammatory environment, thereby favoring wound healing and expediting tissue repair, which is in this case indicated by dashed lines, while also avoiding the inconveniences of using synthetic materials. With the aim of developing biomimetic strategies, my research focuses on the understanding of the information exchanged between stem cells and immune cells during tissue repair. It is clear that stem cell communication with surrounding cells is crucial to create a pro-regenerative environment. In particular, this approach is at the basis of their demonstrated broad anti-inflammatory capacity, which is a property that accounts for the capability of these cells to modulate the behavior of immune cells that normally infiltrate the site of lesion or inflammation, switching, for example, the phenotype of pro-inflammatory macrophages into anti-inflammatory ones, or reducing the proliferation of lymphocytes. The therapeutic potential attributed to stem cells is believed to be mediated by the paracrine signals they release, which operate through a hit and run mode of action. All of this allows stem cells to exert their protective potential, their protective function against tissue damage and infection, and to support angiogenesis towards tissue repair. In particular, stem cells achieve these important tasks by releasing natural nanoscopic particles that contain crucial moieties able to modulate the phenotype of specific target cells. These extracellular vesicles that are called exosomes when in the range of 30 to 150 nanometers are of particular interest for the development of RNA-based therapeutics, as they are characterized by structural and cargo stability and also retain the inherent targeting specificity. In addition to this, they are not subjected to the effect of the environment, avoiding the sudden change in phenotype and function that we often observe when administering bioactive cells, like stem cells or immune cells. And for these reasons, exosomes are considered reconfigurable therapeutic systems. While exosomes offer several advantages over the use of their cellular counterparts, their translation into clinic is hampered by the low yield and difficult characterization. And with this in mind, I'm interested in decoding the information contained within the exosome that are released by stem cells to develop biomimetic approaches able to enhance their therapeutic potential while improving scalability and cost effectiveness. Supported by the European Commission, I'm leading a project to understand in the details how the environment affects stem cell capability to release extracellular vesicles and their properties. Stem cells were then exposed to different cocktails simulating bacterial infection and chronic inflammation. When inflamed macrophages were exposed to the exosomes isolated from the cells treated with each condition, we observed a marked decrease in the release of pro-inflammatory cytokines and a concomitant increase in the expression of anti-inflammatory ones, as the interleukin 13, which is displayed in this slide. These and other results that we obtained in the context of the project call for a rethinking in the stem cell-based protocols applied for the treatment of any specific condition. What we found is that the next-generation sequencing analysis performed on the exosomes isolated from stem cells exposed to different conditions reveal specific molecular signatures in terms of long non-coding RNAs and miRNAs, with differentially expressed RNAs identified as upregulated following exposure to inflammatory cues being involved in the angiogenesis and wound healing pathways. Furthermore, lipidomic analysis performed on the same samples revealed differences in the amount of specific lipid categories depending on the environment cells were exposed to. The pie charts reported here are a quick way to visualize those differences when considering the top 15 most represented lipid categories. Time-dependent changes were also noted over 48 hours treatment. And as an example, I report here the phosphatidylserine, which was found to be significantly overexpressed in the IT group, where the cells were exposed to the interferon gamma and TNF-alpha cues, and that group was responsible for the most marked macrophage polarization we observed. The creation of a dataset inclusive of all the master regulators utilized by stem cells during tissue repair has come to help during the development of the next project I'm going to highlight today, which is about the development of mRNA-enhanced grafts to be used in the management of chronic wounds. The overall goal of the project is to transdifferentiate fibroblasts to endothelial cells by means of mRNA-loaded collagen-based membranes, with the hypothesis that by integrating encapsulated mRNA encoding for angiogenic factors in the membranes, it would be possible to promote neovascularization at the site of an implant. This project has been conceived in collaboration with Dr. Cooke and his group, with a long-standing experience and great success in promoting transdifferentiation of fibroblasts to endothelial cells. In particular, Dr. Cooke's group has successfully transdifferentiated human primary dermal fibroblasts into inducible endothelial cells in vitro, using lipofectamine-based transfection methods and using a cocktail of mRNAs that encode for 4 endothelial lineage factors, ETV2, KLF4, GATA2, and FLIY1, suggesting this approach as an innovative way to enhance neovascularization and improve regeneration of chronic wounds. Following the treatment, a distinct population with a cell surface expression of endothelial-specific markers such as CD31 was identified by flow cytometry. After sorting these CD31 positive cells, cells were also analyzed to make sure that they could express endothelial-specific genes. Functionally transfected cells were then similar to mature endothelial cells, as they were able to form tubes of matrigel and also uptake acetylated LDL. On the other hand, the success of this project relies on our capability to bestow active signals on the surface of collagen-based biomaterials to efficiently modulate the immune reaction to the implant, reducing the pro-inflammatory environment and correctly triggering the healing cascade. In particular, we successfully recreated the extracellular matrix of bone and cartilage in terms of architecture and composition. The incorporation of loaded nanoparticles into the scaffold in a separate study increased the half-life of the payload, delayed the clearance by macrophages, and also allowed for the sustained temporal and spatial control over the release of the payload, inducing a macrophage differentiation. In this study, the mRNA encoding for specific factors have been provided by the RNA core at the Houston Methodist Research Institute. They have been encapsulated in lipid nanoparticles by optimized protocols and using nanoassembler as a microfluidic approach, which is available at the Department of Nanomedicine. Optimization of loading and release have been performed using the mRNA reporter, which encodes for the GFP. The characterization of the mRNA-based lipid nanoparticles produced was performed by DLS, showing the particles fall in the range of 50 to 90 nanometers with a polydispersity index assessed above 0.2 for the particles loaded with the GFP, but also for those loaded with the GFK cocktail. The RNA loading efficiency was found to be about 80%. When GFP-loaded nanoparticles were administered to human fibroblasts, we noticed a greater efficiency compared to the groups where cells were transfected using lipofectamine. The quantification performed by flow cytometric analysis confirmed a 40 to 50% increase in the percentage of cells expressing GFP at 8 hours from the treatment. Currently, we are working on the functionalization of collagen-based membranes with mRNA lipid nanoparticles. In collaboration with Dr. Taraballi, we screened the dataset obtained from the lipidomic analysis of the exosomes produced by therapeutic stem cells and identified phosphatidylserine as a potential player to be used in the formulation of lipid nanoparticles to increase their affinity for an annexin 5 functionalized collagen. In this slide, I show the data obtained from the successful quantification of the Fixie signal detected on the surface of the collagen membranes following the reaction using different concentration of annexin 5, both qualitatively and quantitatively. The formulation of phosphatidylserine functionalized lipid nanoparticles carrying mRNA allowed for the production of particles with a 60 to 80 nanometers in size and a preparation displaying PDI values below 0.2. The efficiency of RNA encapsulation for both mRNA encoding for GFP but also for the cocktail of angiogenic factors was assessed around 80%. The percentage of fibroblasts incorporating and expressing the mRNA reporter, the GFP, was found comparable to that observed following the treatment with lipofectamine and RNA, but significantly reduced compared to that observed for the standard formulation. Finally, when RNA was the RNA was recovered from the phosphatidylserine functionalized lipid nanoparticles and analyzed by a bioanalyzer, lanes corresponding to the base pairs of the 4 mRNAs loaded onto the particles have been identified, suggesting the RNA integrity was still maintained. While additional investigations are required to understand the transdifferentiation potential of the mRNA-enhanced collagen membranes, the data we obtained so far are encouraging, and we can conclude that biomimetic strategies provide an opportunity to achieve functional healing through the reactivation of the body's self-healing potential. They are extremely versatile as they can be tailored for the release of a wide spectrum of RNA. And most importantly, understanding the components of a natural exosome represents an interesting way to develop an effective RNA-based therapeutics. I would like to thank you for your attention, and once again, I would like to thank the Kostas family to support this research about the development of RNA-based grafts for the treatment and management of chronic wounds. My thanks also go to Dr. Cooke for his support and knowledge and constant advice during the development of the project, as well as Dr. Taraballi for her expertise, for sharing her expertise with us on the development of biomimetic materials. Finally, I would like to thank Ramiro Villarreal, who is a PhD student in my lab and who is the person who's been developing part of the experiments I presented here. Thank you.
Bruna, thanks for a wonderful talk. Congratulations. Very nice work.
Thank you, Dr. Cooke.
Wonderful.
I'm very happy to be connected with you, although it's just a virtual connection.
Where are you calling from?
I'm in Italy at the moment.
That's what I thought. Yeah, it's.
Yeah.
Thank you for joining us all the way from Italy. Well, we're looking
My
forward
pleasure.
to
I couldn't
getting you-
miss this.
We're looking forward to getting you back here in Houston. It's been a wonderful
Yeah.
collaboration. Ultimately, what we're trying to do, I guess, what you're trying to do here is to improve wound healing. And as you mentioned at the very beginning of the talk, it is a major problem. Wounds that are non-healing, such as diabetic foot ulcers. And one of the major problems is angiogenesis, the ability to generate a vasculature and get true tissue healing rather than scar. So
Absolutely, yeah.
You talked about how these 4 transcriptional factors, GATA2, ETV2, FLY1, KLF4, how those 4 factors, when overexpressed in fibroblasts, could change them into endothelial cells. And you're now putting those into lipid nanoparticles, and you're putting them into a collagen scaffold. And I guess the idea is that when you're trying to heal a wound, when you put that scaffold in the wound, you have cells migrating and fibroblasts, and they will come upon these lipid nanoparticles and get transformed into endothelial cells. Is that the idea?
Indeed. So we want to consider two parts. First thing is these nanoparticles have to be embedded in the collagen, but also directly available to the cells migrating towards the site of the injury or the site of the implant. What we really want to do is to, in a way, reactivate endogenous cells. Reactivate endothelial cells that are present at the site of injury, but also to induce the fibroblasts that migrate towards the implant and instruct them towards the differentiation into endothelial cells. For example, the reason why we also pick the phosphatidylserine among the lipids that we've been identified in the exosomes that we've been analyzing is also that phosphatidylserine present on the exosomes derived from stem cells is one of the ways through which endothelial cells can uptake, so to increase the uptake of this exosome by endothelial cells. So the approach is going towards 2 directions. The first direction is to modulate the phenotype of fibroblasts and transdifferentiate them into endothelial cells. But the technology that we are developing, I believe, has also another promise, which is the capability to instruct the endothelial cells and maybe face one of the challenges associated to the chronic wounds as the consequence of cardiovascular disorders, which is endotheliopathy, where endothelial cells lose their identity and their capability to function as such.
Wonderful. I saw, and maybe correct me if I'm wrong, but it seems that you're getting fairly high transfection efficiency within the collagen scaffold. You're getting, look like 80% of the fibroblasts, or did I misread that slide?
Yeah, well, so we still have to produce final results coming from the transdifferentiation from the collagen scaffolds, but the capability of these nanoparticles to being uptaken by the recipient cells and express the markers that the proteins associated to the coding mRNA that we are utilizing is very high. Yes.
Mm-hmm. That's spectacular.
And
so
it was interesting to see that those numbers are in some cases higher than the numbers that we obtain following the treatment with lipofectamine. So this part was interesting and deserves more investigation.
Yeah, very exciting. Well, I've got more questions for you, but I'm afraid that we're running a little bit behind, so we're going to have to say goodbye, Bruno. I do want to congratulate
Goodbye.
you on getting federal funding for this project. Congratulations.
Thank you. That was
Department of
such,
Defense,
yes,
right?
that was
Yeah.
such a good news.
Wonderful,
Thank
wonderful.
you.
Well, keep up the great work. All right.
Thank you.
Bruna. Our next speaker is Henry Pownell, who's a professor of bioenergetics here at Houston Methodist Hospital. He's a lipid chemist. His training was at Baylor College of Medicine and University of Houston, and he is going to be speaking to us today about the physical, chemical, and metabolic determinants of lipid nanoparticle stability. Henry Pownell.
I want to thank the organizers for this meeting and for the opportunity to present some of our studies. Today, I will speak on behalf of the lipids team about the physical, chemical, and metabolic determinants of lipid nanoparticle stability. In this connection, we have nothing to disclose. In 2020, a black swan event, the SARS-CoV-2 pandemic, stressed global public health and economies. One immediate and ongoing effect, as shown in the upper panel, was increased mortality. And there's also increased morbidity among survivors that made holding a job impossible. Both effects hurt the economy by taking people out of the workforce. As shown in the lower panel, the US gross domestic product fell by more than 3%. The lower GDP reduced tax revenues at all government levels, thereby producing additional precarity in education, healthcare, and national defense. Thus, preparing for future pandemics and better managing other disease is a global public health priority. In response to the pandemic, vaccines containing mRNA coding for a fragment of viral protein and a vehicle for its delivery, that is a lipid nanoparticle, were formulated. Post-injection, mRNA commandeers cells to synthesize and secrete a viral protein fragment, which elicits antibodies that neutralize the virus. The mRNA vaccines have some advantages. The mRNA only needs to access to the cytoplasm. It does not integrate into the genome, and its production is cell-free. The protein product has native properties. There are disadvantages. There is intrinsic immunogenicity. There is susceptibility to RNase degradation, and cellular uptake of mRNA, naked mRNA, is nil. To elude these disadvantages, vaccine mRNA has been incorporated into lipid nanoparticles. Many nanoparticles comprise a single bilayer sphere of phospholipids surrounding an aqueous core. Polar and ionic molecules are retained within the central aqueous compartment. Nonpolar molecules, such as rapamycin and doxorubicin, shown here, are sequestered within the hydrocarbon region of the bilayer. Amphiphilic molecules, including cholesterol and phospholipid with a pegylated head group, absorb to the surface with their hydrophobic groups penetrating into the acyl chain and their polar moieties facing the surrounding aqueous phase. Notably, addition of cholesterol to lipid formulation increases particle size. Here we show the formula of the nanoparticles used in the formulation of the Pfizer-BioNTech vaccine. As reported by Adriana Rodriguez of USA Today, the ingredients are pretty standard. The cationic lipid supports electrostatic binding to negatively charged therapeutics such as mRNA. The cholesterol and disteryl phosphatidylcholine, shown as DSPC here, provide bilayer stability, and pegylated lipids help the nanoparticles evade the immune system. Importantly, phospholipids are susceptible to phospholipases and other physiologic challenges shown here. Cholesterol and phospholipids spontaneously transfer among lipid surfaces on a time scale of minutes to hours. Phospholipid transfer is accelerated by lipid transfer proteins. Phospholipases degrade phospholipids to free fatty acids and lysolipids, which are hepatically extracted in a few minutes. Cholesterol and phospholipids are extracted one at a time by the HDL receptor, SRB1, and whole nanoparticles could be endocytosed by other lipoprotein receptors. The major determinant of the rate of phospholipid movement among the physiological surfaces is the hydrophobic effect. This force is described in simple detail by a book by Charles Stanford, who studied the properties of detergents such as fatty acid soaps. These in my cells were the simplest lipid nanoparticles. As shown here, phospholipids and free fatty acids are similar in having a head group and one or more acyl chains. Whereas phospholipids form bilayers, the same forces, the hydrophobic effect, drives fatty acids into my cells. Here we summarize some of Tanford's findings. My cells form at very high fatty acid concentrations. Each methylene unit adds 800 calories per mole to the free energy of my cell formation. Increasing the acyl chain length increases the binding of lipids to my cells and to phospholipid bilayers. Thus, we hypothesized that the rate of lipid desorption would decrease with increasing chain length. Stronger binding means a greater energy barrier to desorption. We measured the rate of transfer of a series of saturated fatty acids from lipid vesicles to serum albumin. According to our data, transfer of one physiologically abundant fatty acid with 16 carbon chain, that is palmitic acid, occurred with a half time of 3 milliseconds. In contrast, transfer of a 22 carbon acyl chain had a half time that was slower, 30 seconds. Thus, the addition of only 6 carbons to the acyl chain reduced the transfer rate by 4 orders of magnitude. Notably, the transfer time of a 26 carbon fatty acid that accumulates in inherited disease adrenoleukodystrophy is 3 hours. According to our rate data and transition state theory, the free energy of activation for transfer is 650 calories per methylene unit per mole. These data show the very profound effect that hydrophobicity, here in terms of acyl chain length, has on transfer rates. Moreover, the close correspondence between free energy of my cell formation and the free energy of activation for transfer to other surfaces suggests that the rate limiting step for transfer is lipid desorption from the nanoparticle surface. These observations provoke the hypothesis that phospholipids which have 2 acyl chains will transfer at a slower rate. Our data support this hypothesis and show that the rate of transfer of phosphatidylcholines decrease with increasing acyl chain length. Here, the addition of 4 methylene units increases the half time from 11 to 95 minutes. From the rate data on a series of phosphatidylcholines, we've formulated a master equation in which the transfer half time increases with the number of methylene units, N, but decreases with the number of double bonds, M. The middle panel shows the linear fit of the measured and calculated half times. Transfer rates for lipids containing 4 or more double bonds were abnormally slow. The longest transfer time was observed at 6 hours, shown by the arrow. As shown on the right, the effects of differences in polar head group were comparatively small, with the transfer rate of phosphatidylethanolamine being only 3 times slower than that of phosphatidylcholine. Here we summarize the in vitro rates for some physiologically relevant phosphatidylcholines. Data for 2 of these are highlighted in red font. One of these, C16 C18-1, which occurs in membranes and lipoproteins, has a transfer time of 147 minutes, a bit over 2 hours. The other contains a long chain omega-3 fatty acid, has a transfer half time of about 6 hours. These are the maximum half times that one might expect to observe in vivo. So what are the plasma lifetimes in vivo? To address this, we turn to our favorite natural nanoparticle, high-density lipoproteins, or HDL, which occurs as a nascent particle secreted by cells expressing the cholesterol transporter ABCA1, and an HDL reassembled particle that contains phospholipids and cholesterol and is made in vitro. Here we show the structure of the reassembled HDL comprising only phosphatidylcholine and the major HDL protein ApoA1. This structure is similar to that of nascent HDL produced by ABCA1 expressing cells. We then incubated ApoA1 labeled with I-125 with cells that were labeled with tritium-labeled cholesterol and C14-labeled choline, giving a triply labeled nascent HDL, which was isolated by size-elect exclusion chromatography. The HDL was injected into groups of mice, which were sacrificed at various times, after which their plasma was counted for radiolabeling. The results here show that both cholesterol and phospholipid are cleared from plasma within minutes. On the same time scale, most of the radiolabeled cholesterol and phospholipid appeared in the liver. In contrast, the clearance and hepatic extraction of ApoA1 occurs with a half time of about 8 hours. Thus, two lipid components of this nanoparticle are hepatically cleared within minutes after injection. Our proposed mechanism is as follows. After the transfer of HDL to plasma, both cholesterol and phospholipids are cleared by spontaneous hepatic extraction and via the HDL receptor SRB1. Concurrently, some phosphatidylcholine is hydrolyzed to lyso-PC and free fatty acid, which are transferred to albumin and then to the liver, where they are repackaged into lipoproteins. Here we show the plasma activities that can degrade phospholipids, whereas the reaction products, fatty acids and lyso-PC, transfer on the time scale of milliseconds, their plasma lifetimes are on the order of 5 minutes. These activities are endothelial lipase, less than cholesterol acyltransferase, phospholipase A, lipoprotein lipase, hepatic lipase, and the HDL receptor SCARB1. One way to extend the plasma lifetimes of the phospholipid component of the nanoparticle is to replace the phospholipids with their ether analogs, as shown here. We synthesize a series of ether and ester-based phospholipids containing tritium and C14 labels, respectively. These were incorporated into HDL and injected into rats. At various times, plasma was collected and counted, and the kinetics of clearance calculated from an exponential bit of data. The results showed two things. One, replacing ester with ether linkages doubles the plasma lifetime, as shown here by all 4 of the lipid analogs. Second, increasing the chain length of the diacetyl chain or dialkyl chain also increases the plasma lifetime, such that the half-time of the longest-lived ether analog is nearly 2 hours. Interestingly, there are naturally occurring phospholipids that could be expected to be highly stable. Here we show the structure of phospholipid from the microorganism belonging to the Archaea domain, which lives in harsh environments that include salt and thermal vents in the Pacific Ocean. These are built for survival because they are not oxidizable. They have a fluid-like structure because they have no double bonds. They contain at least 40 carbons, so they cannot transfer readily, and they have ether bonds to their glycerol backbone, so they are resistant to enzymatic hydrolysis. These studies and studies from other labs provoke new questions about the design of the vehicles for lipid nanoparticles. First, how do plasma activities interact with the lipid nanoparticle component? These are the lipases, lipid transfer proteins, lipoprotein receptors such as SCARB1, which extracts individual lipids from the nanoparticle. Next, can lipid nanoparticles' pharmacokinetics be optimized according to a better lipid structure, charge, stoichiometry, and size? Is there a rapid, inexpensive, cell-based pre-screen for vehicle efficacy? What are the structures of mRNA lipid nanoparticle vaccines according to cryo-electron microscopy? Can chemical modifications, for example, acylation of APOs in reassembled lipid nanoparticles, be optimized for stability? How are pegylated lipids catabolized, and what are the rates? Does the HDL receptor extract hydrophobic drugs from the lipid nanoparticles? With that, we thank everyone for their interest and refer you to our recent review that summarizes some of our findings and concepts. Thank you.
Thank you, Henry. Thank you, Henry, for that wonderful talk. I'm hearing a little bit of background here.
Tell him to turn his audio down.
Okay. Henry, thanks for joining us today, and thank you for that thoughtful talk on lipoproteins and their composition. I was wondering, actually, and someone else here in the chat is wondering if there might be better transfection efficiency with endogenous lipoproteins versus lipid nanoparticles. Could endogenous lipoprotein particles be used for mRNA transfection, for example?
Yes, if they would have to have some hydrophobic characteristics. Most of these depend on the water solubility of the mRNA, and according to most of the diagrams I've seen, they're encapsulated. So I don't think it would be a good strategy just to use lipoproteins, because you just will get low binding unless you incorporated a cationic lipid into the lipoprotein to enhance the binding to the negatively charged mRNA. I think the lipoprotein approach is more amenable to delivering hydrophobic drugs or amphiphilic drugs. And it's very interesting to hear Dr. Kostas talk about ApoE for targeting. And I think there are strategies to use fragments of ApoE that have been described with non-transferable acyl groups on them that extend the plasma lifetime. And I wondered what actually the lifetime of the nanoparticles was in plasma, and whether or not there was some sweet spot for the lifetime of a nanoparticle.
Right. And I guess do we have any idea what the lifetime of lipid nanoparticles are when they're injected into muscle, such as a vaccine?
Really good question, and I've wondered about that myself. And the chemistry of determining that lifetime, and then what I spoke of was really plasma chemistry, but the muscle chemistry is going to be very different. I don't know what kind of enzymes or transfer proteins are exposed to the nanoparticle, and trying to determine the stability of the nanoparticle with an intramuscular injection be difficult to do. But we have to start with the fact that they work.
Mm-hmm. Do you, based on your understanding of lipid kinetics, the work that you've shown us, what kinds of developments do you see in improving lipid nanoparticles for systemic infusion? What can be done to enhance the longevity, for example, of these LNPs circulating in the blood?
Well, we would really like to make some of the lipid nanoparticles just slightly altering the formulation that's been used by Pfizer BioNTech. So the disteryl phosphatidylcholine is made up with a longer chain fatty acid, and the amount of cholesterol is modified, or maybe even another sterol would work better. I think the cholesterol is there to stabilize the nanoparticle at the beginning, but according to plasma chemistry, the cholesterol is gone in minutes in plasma. May be different in muscle. That's an interesting thing to do. But the other thing, maybe we don't want the nanoparticle to be too stable. So there may be some optimal lifetime for a nanoparticle for it to have its maximum therapeutic efficacy.
Thanks, Henry. Well, we're going to have to move on to the next talk, but thank you so much for giving us that lesson in lipid biochemistry and lipid nanoparticles.
Thank you so much. Enjoyed it.
We're going to move on now to our next speaker and the last speaker in this session, which is Dr. Joan Nichols. She is a professor in the Department of Surgery and tissue engineer. We were fortunate enough to recruit her from the Galveston National Laboratory at the University of Texas, where she was professor of internal medicine and microbiology and immunology. And we're so happy to have her here to continue her work on lung regeneration. She is an expert in human immune response, particularly in the lung. She's done work in stem cell regeneration of the lung. She's generated these stem cell organoids that are living models of human tissue. And she also has done some very interesting work at recellularizing acellular whole lung to produce lung tissue as an approach to bioengineering human lungs. So today we're going to hear about her work developing models to support vascular tissue engineering. Dr. Joan Nichols.
Good morning. I'm Joan Nichols. I'm going to talk to you today about developing models to support vascular tissue engineering. Basically, my group has been designing methods to support tissue engineering for a long period of time. What I'm showing you is a publication in 2016 where we bioengineered our first whole lung. Prior to this, we've been just doing small portions of the lung. But what you can see on the left-hand side is an acellular scaffold, and as on the right side, you're seeing in this image what that same scaffold looks like after the process of recellularization, where specific cell populations are added back to specific regions of the scaffolding material. Although this process worked fairly well for us, we had some issues in that we were really good at developing the microvasculature, which is one thing that we've been working on a lot for a number of years, and modeling these responses. And modeling means evaluating small pieces of material, small tissues for how an individual factor or cell type influences what's going on. So in this image, what you're seeing from microvasculature from that same lung, capillaries are well developed. There are no red blood cells in it because we don't use red blood cells to transport oxygen through our tissues. We have different mechanisms. There are type 1 alveolar epithelial cells that are present. There are type 2 alveolar epithelial cells present, as well as pulmonary interstitial macrophages, which are critical to, in our case, development of some of the tissues, specifically the microvasculature. What we found for these lungs, though, is that we weren't very good at the process of developing the larger blood vessels. We knew there were issues with pulmonary artery and vein, in that modeling these, in working with just these components, we found that we couldn't get a good patent vasculature, and they were very leaky. But even in this case, you can see there are regions that you have big dark holes, and these are where the larger blood vessels would exist within this developing tissue. And this is a nice 3D CT that's demonstrating just the vascular component alone. So moving on from this, and despite some of the issues we had with it, we moved on to a pilot project where we produced lungs to be transplanted into a large animal, or pig model in this case. And so the lung you're seeing in front of you is actually on day 30 of culture. It's been fully recellularized, and it's a single lung. We're doing single lung transplants for this for the pilot study. In this case, because we knew that we had issues with specifically pulmonary artery and vein being well developed enough to support the pressures that are needed to support circulation through the lung itself, our goal was to just reconnect the airway, allow the tissues to survive, and the lung is the only place that this can happen, and to evaluate formation of systemic circulation alone. Just redevelopment of systemic circulation alone and methods to support this process. So just the airway was reconnected in these studies and not pulmonary artery and vein. So in this pilot study, on day 1, a pneumonectomy was performed, and then the left lung in this case was removed as a cell source from the pig following the pneumonectomy. The right lung was left in place, and the animal can survive on that right lung alone. Cells were isolated from different regions of the lung, tracheal, bronchial, lung vascular populations, and then the process of recellularization of an unrelated pig scaffold was done. And this is an unrelated acellular pig scaffold that we had prepared ahead of time to receive the cell populations and to support their development. These lungs are grown for 30 days in culture. At the time of surgery, the tracheal anastomosis was done, but again, no pulmonary artery or pulmonary vein. And pigs were survived at different time points to allow us to assess a number of things, including reestablishment of the systemic circulation off of the aorta. So this was a major component for us to consider. We also evaluated things like transcriptome of the engineered lung. We did basic histology. We looked at microbiome reestablishment, and somebody using this as a platform to manage what happens in terms of transplant microbiome reestablishment that may be problematic. And we evaluated basic lung function, but with only systemic circulation, you're not going to get oxygen. You're not going to get oxygenation of the tissues from this because it's just the systemic circulation that supports the lung itself. And our focus, again, was reestablishment of that bronchial circulation or the systemic circulation in this pilot. So what I'm showing you here is a lung at 2 months post-transplant. The gross image and the image on the other side showing you how well the lung is expanded in the animal. A lot of imaging was done during the study following the transplant of these lungs into the pigs to allow us to watch for that reestablishment of the systemic circulation and to just evaluate how well the animal was tolerating these lungs, how well the vascular system that we had produced was aligning and reestablishing the circulation throughout the lung itself, and how well it was aerating and functioning as a basic process in terms of work or breathing and that type of thing. So when you look at what you see in the gross image here, native lung is the larger lung. Remember, our lungs are always smaller because we're not perfect in our isolation process to let us collect all of the cells from a single lung when we're getting ready to do the process of recellularization, nor is recellularization a perfect process. And we lose cells in that process as well. But what you can see looking as you move down the trachea and into the branch, follow the trachea down and look until you move into the actual engineered lung itself, you can see a number of large vessels that have redeveloped to support this lung systemically. And what I can't show you here, because we don't have a perfect image to do that, is the redevelopment of the systemic circulation off the aorta. So basically, this pilot study provided us with a great deal of information. We know at each stage of development how much the lung continued to form and develop following transplantation. We could watch the process of maturation of that vascular component, the larger blood vessels in the tissue. We could look at development of the systemic, the blood vessels and the vascular portion that would support this lung in terms of systemic circulation. And of course, we could establish what's going on in terms of development of microcirculation, which again, this is what we had developed first and the process that works the best. So when I talk about modeling, I need to tell you that everything we know about engineering a whole organ, we determine by modeling responses. Using small pieces of scaffold like is showing here. This is the hematoxylin eosin stained section showing you there are no cells. There's no residual nucleic acids left in the scaffolding material. It's a clean slate to function as a skeleton to support tissue development. And these scaffolds can be modified using things like keratinocyte growth factor, supernatants from cell populations like mesenchymal stem cells or macrophage supernatants. Or you can even co-culture cell types in here, whatever cell type you would need to evaluate in terms of how well cell types support development of a particular tissue, in this case, distal lung. This information was published in 2018, by the way, because this is the modeling that led to being able to develop the large lungs for transplantation. So what do I mean by modeling? Well, you take that scaffold and modify it with whatever you need to assess, and then you look for tissue development, and all of this is actually done in vitro. None of this is in vivo. So you're looking at what reconstitution or recellularization of those tissues looks like overall. And what you can see is that there are type 2 cells present. And these are often referred to as corner cells because they lie in the corners of the alveoli, which they're doing here. And type 1 cells are present, and those are the cells that would actually be involved in supporting gas exchange. So in terms of what we needed to do as a next step to really focus on vascular development, we had to go back to the acellular lung scaffolds. And this is a acellular pig double lung set. And you can see the trachea is noted here. The pulmonary artery and vein are on the other side. You're not seeing them at this moment. But what we did is we would fine dissect these large lung scaffolds to allow us to isolate the pulmonary artery, the pulmonary vein, as well as small blood vessels that are within the scaffold. Small to us, but large in terms of development of the overall vasculature of the lung. We're showing you an image here at the bottom on the left, or those are some of the small blood vessels that would be used as a tool to model vascular development for those vessels. But you can also use pulmonary artery or vein or pieces of those pulmonary artery or vein. Now, the important part about working with the vasculature in these acellular scaffolds is that the blood vessel vasculature of an acellular scaffold, the blood vessels that are in here may retain some of the structures like tunica media and tunica intima shown in this upper image. These are just the scaffold that's left behind. There are no cells here. There's no basement membrane. These are very delicate structures if you look at the image below, where you can see how fine and filamentous it is, which means that some of the materials that we've been working with, like modified hydrogels, would not allow us to either deliver cells properly to this population or deliver factors to this population because they're not retained within the scaffolding material. And that's why I'm doing a lot of modeling to assess delivery systems and methodologies to just support vascular development is critical to our project overall. So we developed these small bioreactor systems, and I'm showing you one here. That's a single reactor chamber. They're very small. And they actually support the culture of these squares of acellular scaffold that I showed you. And this image is showing you a number of pieces of acellular scaffold cut from a pulmonary artery or vein. I believe these are from pulmonary artery because they're relatively thick at this stage. This is just the scaffolding alone. This allows us to assess things like cell attachment after treatment of these scaffolds, some delivery system assessment. And obviously, we also have to do three-dimensional structures. So the yellow portion you see in the middle here of the chamber, with a small modification of this chamber, we can run whole vessels in this, or in a larger chamber, actually culture pulmonary artery or vein intact. That allows us to actually look at what happens with development of the larger vessels three-dimensionally as well over time. This is all in vitro culture again, to give us a methodology to assess and determine, well, what do we need to have present? What factors, what cells to support better vascular development and development of good patent vessels. So we assessed a number of delivery systems. And initially, because the hydrogels didn't work very well for us, and you can see from this map here, that's comparing VEGF-loaded hydrogels to particles that are also loaded with VEGF. What you can see in this case in the hydrogel, which is in the triangle, is it very quickly releases its load and then drops down within a few hours of not being able to support much in terms of cell attachment or cell function. In this assessments, we compared the hydrogel to use of the silicon wafer microparticles developed by Dr. Liu here in the Research Institute. And what you can see here is that the pore sizes in this case can be modified to allow for small pore or large pore sizes. And we assessed delivery of VEGF in this system to determine which would be better, small pore size, large pore size, or a mixture. In these studies, what we found is both releasing the VEGF into the system or supporting attachment of cells and viability and proliferative ability of these cells. Basically, a combination of 30 and 60 nanometer pore size particles supported the best process for getting cells to, well, attach, grow together, develop a patent system, and remain viable and proliferative through that process. So again, this is work that I'm presenting you here with the particle evaluation that was just published recently. So in terms of modeling, what am I really getting at for these vascular models? Well, we can evaluate dispersion of a delivery device, in this case, the silicon wafer microparticles, as they're being deposited within the vascular acellular scaffold. We can evaluate on the small pieces that I showed you, pulmonary artery and vein, how well this particular delivery system and what factors, what delivery system and what factor in this case, we were using vascular endothelial growth factor delivery to assess cell attachment in cells that are labeled with a marker that allows us to track them within, without having to go back and stain them later. So these are a vascular endothelial cell population that obviously the hydrogel with VEGF wasn't enough to support much cell attachment, just a little tiny bit, but using 30 or 60 nanometer sized pore sized particles really provided what we needed, and a combination of these two together supported over initial release of factors early to allow for attachment and later release of this factor that supported good tissue development. And that allows us to also do this same process and evaluate both microparticle dispersal in a 3D blood vessel, as well as, in this case, again, the vascular cells are labeled in green, attachment and development of tissue by the attached vascular cells, focusing just on development of vasculature alone. So what do I mean about evaluation of what's happening? Well, using these models that we're working with, you can look at, well, basic cell attachment. These are endothelial cells attaching to the tunica intima region of the blood vessel. We can watch as those cells grow together and proliferate over time, which with the proper growth factors present isn't an extensive period of time while they're growing together. We can even look at, beyond that, how well these cells actually move together to produce a patent vessel, and how well those tight junctions develop in these same tissues to allow us to produce a nice patent vessel over time, provided the right cell types and factors to support this process. And this works not just in the flat pieces of tissue, but three-dimensionally as well to support initial cell attachment and then in vitro development. And this blood vessel was developed in vitro. In vitro development of vessels to the point where we're developing very good smooth muscle layers in these vessels and extremely well-developed endothelial components in these blood vessels. And you notice there's nothing else developed around this vessel, it's just the vessel alone, in this case, that we were evaluating development of. So we can control this process a lot better with focus just on the vascular system alone. So what does that look like for development of a whole vessel? Again, and not just in cross-section. In this case, the lung-derived vascular cells were labeled with carboxyfluorescein succinyldiester, which is a fluorescent dye that allows you to see where the endothelial cells are actually coming together to form this individual vessel. And then this was also stained for VEGF using an antibody against VEGF with a red fluorochrome. And you can see actually the release of VEGF within this component that's developing this vessel that lets you see where the degrading microparticle is as it's continuing to release this vessel over 15 to 20 days of bioreactor culture. So now coming back to the whole lungs, we've now developed methods bringing this all together that will let us look in whole scaffolds and evaluate how well blood vessels are growing. And this is highlighting a region of vascular component that we're looking at. We can remove sections of this to assess how well an individual delivery system has delivered to that site. And that's looking at some of the microparticles that were tagged with a fluorescent dye. We can look at the particles within capillary beds. In this case, we're looking at the delivery system in a small capillary, and you can see the size and shape of these individual silicone wafers within the capillary bed to allow us to reach some of the smallest regions. And I believe this is why we're getting such good microvascular production, including ways to, when we're working with the whole lung scaffolds, to tag these particles while we actually put the particles, pumping them through the pulmonary artery, and evaluate what happens over 30 minutes of dispersal, 120 minutes of dispersal, to actually monitor how well we're filling the vascular tree of that acellular scaffold with the particles that we need to support growth factor release over time. We can also deliver cell populations this way. And actually that's part of what the process is in terms of modeling in the small tissues and bringing it back to the whole lung structure. So I need to mention too about other nanofluidic delivery systems. We are really fortunate that we've had a long-term association with Dr. Grattoni, who's also at the Research Institute. And the work that he's doing to develop nanofluidic delivery systems, and I'm showing you one here, this small 7.2 millimeter size bullet-shaped delivery system, for us, we think is the exact thing that we're looking at to be able to enhance pulmonary artery and vein development. This can be placed in larger vessels or in regions that we would need to have delivery of a controlled product. This is actually repurposing these delivery systems that were made for drug delivery for use for them growth factor delivery in our systems. And I think this is really going to make a big difference in how we manage, and that's one of the things in terms of managing factor delivery in a temporal-spatial manner that says both the time you release and where you release is really critical. And these can also be placed in the pulmonary artery and vein and then removed from them before we move to transplantation. Or if necessary and they need to be allowed to be present in some tissues, they've already been evaluated for that process. And so that makes it really good for us, but just repurposing these delivery systems. The same as what was done for the microparticle, the silicon wafer particles, because they were actually produced for chemotherapy drug delivery. And we just repurposed them for our growth factor delivery. One of the most intriguing things is the possibility of using a nanochannel membrane structure, also developed by Dr. Grattoni, that allows you to put a drug in place and then control release of that drug by using an app on your phone to actually control the time you release a therapeutic. And in our case, we're looking at this for late stage delivery in pulmonary artery and pulmonary vein while they're in culture. while they're in in vitro culture to allow us to control where we're releasing and when we're releasing a specific factor that needs to be present for maturation only. And again, these can be removed following before we actually move to the step where we're transplanting these lungs. So having these abilities to manage, to evaluate delivery systems first before we move to doing it in a whole lung, and then slowly moving this process along is really a big, it's a major step for us, and it's what's going to allow us to move on to our phase 2 studies. So our phase 2 studies are actually going to be where we connect both the airway and the pulmonary circulation. In this case, our goal is to establish oxygenation of blood. And we'll do this by doing the same process, remove the left lung, isolate cells from it, grow along on an unrelated pig scaffold, an acellular scaffold, bring that back and do a full transplantation. And then once we're ready to assess the ability of that bioengineered lung to oxygenate, possibly after being in place for a month or 2 months, we will actually clamp off the native lung so that we're, in terms of the vascular system, so we're only getting blood flow through for the engineered lung, and allow us to assess what's going on in terms of the ability of that engineered lung to oxygenate and provide the function that the lung is there for, basically. So I really need to stop at this point and mention the people that do this work with me. There's an amazing group of people that I work with, and I'm very fortunate. Jean Niles, who's my lab manager, Lasenia Argueta, who did a lot of the modeling for the vascular component, Maria Grimaldo, who's learning that process now and will be a major influence. I did not mention Dr. Arcaria. Dr. Arcaria is at Baylor University, and he will be instrumental in our step 2 process because he's actually going to produce biomaterials that are specifically developed for the pulmonary artery and pulmonary vein to provide support and slowly degrade over time. I also want to mention the people that were involved in the phase 1 study, the clinical team that actually did the transplant. Saverio LaFrancesca was the surgeon, and the team that supported all of that work, including Jason Sakamoto, who was at Methodist when I was working with him on the phase 1 study, but has now gone on to consulting work, as well as Luba Frank, who has done all of the pressure evaluation studies and all of the imaging in the animals that we worked with. And at this point, I'll take questions. Thank you.
We're having some technical problems there.
There we are.
Wonderful. Sorry about those technical problems. Wonderful talk. We have time for just 2 brief questions. The first one is from Dr. Krishna Murthy. Dr. Kumar Krishnamurthy his question is regarding the mechanical properties of the scaffold, the decellularized scaffold that you're using. Can you comment on the compliance of the decellularized scaffold and how that affects the lung function?
So the decellularized scaffold itself gets stiffer. That process of removing the cells makes it much more stiff. And so when you're doing basic work of breathing studies, you can see that there's a big change here. There's a big difference. In terms of looking at what happens with, if you recellularize, you return to relatively normal work of breathing levels. And this information, a lot of it has been presented in some past publications in terms of production of human lung for evaluation as well as pig lung.
I guess the idea would be to regenerate a lung using someone's autologous cells, correct? That would be the idea, using a decellularized scaffold from a donor and then recellularizing that with autologous cells?
Possibly it's down the line someday. That's a general view of what we'd like to do. But you're never going to get enough cells to be able to do this. There's also the issue of what cell types you isolating from someone's own lung. So eventually we'd like to use this process to where you bank cells from tissues that are not, or donor lungs that are not suitable for transplantation, but bank those cell populations, have them pre-HLA evaluated for tissue type, and be able to, when you have a donor that, a lung that comes through, you store those cells, and then you use them when you have someone who needs a lung to produce them. And that's another option for making best use of donor lungs that are not suitable, as well as cadaveric tissues.
Mm-hmm. So if you're using the decellularized lung from donors and applying these cells that you've banked, what is the effect of the diseased scaffold on the formation of a lung? What effect will that have on the lung
You've
function?
asked the million-dollar question. So you're not going to use necessarily a diseased scaffold to produce these tissues. You're actually going to use from, again, lungs that are not suitable for transplantation, that have had some minor issue in terms of the process of procuring them or management of them. Those will be the lungs that will be used to produce the scaffolding material. Unless you have scaffolding from somebody that is appropriate, and we can show, we do a lot of assessment with MRI and CT to look at each of the scaffolds we use, the whole lung scaffolds, as well as what the lungs look like from what an individual would have pre-transplant, to get an idea of what the best process moving forward should be. And basically, the best process is probably going to be using scaffolds from unrelated donors that are clean and well prepared so that they don't drive any immune response. Clean meaning removing all of the cells and nucleic acids that would have been in that scaffold, and then working with cell populations from a matched donor. So we're still going to be looking at HLA matches, but it's possible in the future we may be able to use techniques that would allow us to, well, alter the genetic profile of those cells that we're using and use them instead of gene therapy in an individual, use it in the tissues and cells themselves before we build the actual lung to be used. Mm-hmm. And if that process isn't reasonable, you understand that we're developing lots of low-hanging fruit in terms of processes that give us information that would allow us to perhaps even heal tissues within you without having to go through the process.
Very exciting. Thank you, Joan. And I'm so glad you're here with us at Houston Methodist now, and I look forward to working with you. Well, we're going to move on.
Thank you very much.
I wish we had time for more questions, but we have to move on now to our lightning rounds. We chose 10 of the poster abstracts for presentation as a lightning round. Each of the postdocs gets 2 minutes to talk about their work and get you excited about going to their poster session. So that's going to follow. There's 10 of talks, so that'll be 20 minutes. Grab your lunch immediately after that, and then please rejoin us for the poster session, which will go till 1:45. And then at 1:45, of course, we'll start the afternoon session with our panel on the future of cardiovascular nanotherapeutics. Okay, now for the Blitz round.
Hello, everybody. 2 years ago, this meeting, I presented for you my preliminary data, suggesting that telomerase mRNA could rejuvenate the cells. The goal of my study is to develop a therapy for children who are suffering from progeria syndrome. It is a premature aging syndrome, and these children actually dying because of the heart attack and a stroke at teenage years. Today, I have finished part of my study that shows the proof of principle. We treated their vasculature with telomerase mRNA, and we observed that telomerase could recover the proliferation of the cells, the growth of the cells. Telomerase reduces inflammation and damage in their cells. And telomerase were able actually to restore both function and morphology of their vasculature. And more importantly, when we use virus to deliver telomerase in the mice model of the progeria, telomerase therapy caused the extension of the life of the progeria mice. To apply this promising therapeutic approach in human and actually move on to the clinical trial, now we are working to optimize the lipid nanoparticle delivery of telomerase mRNA. And that therapy could be beneficial for any age-related vascular disease. Thank you very much.
Hi, everybody. I'm Stefano Serpelloni, and I'm a PhD student in Dr. Taraballi's lab. And I'm working in collaboration with Dr. Raimi on the development of a system to prevent the occurrence of vascular graft infection. Vascular grafts are used for dialysis access creation and arterial venous reconstruction. Vascular graft infection is one of the major complications related to this application. And morbidity and mortality as high as 75% of the cases, and limb loss up to 70% of the cases. The current strategies rely on the systemic administration of antibiotics and the surgical removal of the vascular graft when infected. And for what it concerns the surgical option, the problem is that it's a really challenging operation, because when we replace it with an extra anatomical bypass or autologous vein, the prolonged time of operation and instability of the patient is one of the main concerns. So in this framework, our idea is to develop a system exploiting the nanotechnology in order to encapsulate vancomycin and chitosan, exploiting the electrospinning technique. Exploiting this technique, we are able to obtain the local and sustained release of antibiotics up to 10 days. And spurred by these exciting results, we are also evaluating the possibility to encapsulate also gentamicin in order to increase the effectiveness of our system. And with this in mind, we aim to create a new paradigm of drug-eluting vascular graft, exploiting the electrospinning technique and nanotechnology.
Telomerase is known to extend the life of cells. However, we found a new role. Telomerase could prevent DNA damage. People receiving chemotherapy or radiation, they often have an accumulation of DNA damage in their cells. With this, it comes inflammation, and it just works as a cascade of negative effects, because this inflammation can also activate cells in a way that they start losing their cell identity. For example, vascular cells, they can become stiff, they stop working properly, and they can become fibrotic tissue. However, we found that if we treat cells with telomerase after being exposed to a chemotherapeutic agent, it could prevent DNA damage, reduce inflammation, and properly preserve cell morphology and function. There are two very important things that we need to address right now. The first one is to understand how telomerase works, so we can prevent DNA damage. This way, we can reduce or stop any DNA damage accumulation in the cells. This is extremely helpful for people with cancer, because understanding how telomerase works, we can reduce the side effects of the therapy, and we can increase the efficiency of this therapy. Not only for them, but also for people that are suffering from diseases associated with aging. The second one is that developing of a reliable, efficient, and safe telomerase therapy using lipid nanoparticles, so we can target cells or organs being affected. Please join me on the poster session, where I will be sharing more of these fascinating findings. Thank you.
Hi, my name is Amberly Royal. I'm part of the Filgera lab here at the Houston Methodist Research Institute, and today I will be showing my team's findings over the photothermal properties and radio opacity of near-infrared sensitive gold nanoshells. Gold silica nanoshells, as represented here in schematic A, can be fabricated to exhibit strong resonance in the near-infrared region of the electromagnetic spectrum. This gives them the ability to conduct optical heating when excited with near-infrared light. Since they also have a metal shell, they also possess high X-ray attenuation and density, which gives them the possibility to serve as a possible CT contrast agent. Throughout this experiment, we attempt to optimize the parameters necessary for the nanoshells to serve as a thermoplasmonic modality and a CT contrast agent. We started by confirming their residence at the 808 nanometers via UV vis, as seen here in figure B. In schematic C here, I have our experimental setup, which was used to determine the relationship between gold concentration and power output and the time necessary to reach 50 degrees Celsius, which is a clinically relevant temperature where tissue destruction occurs. Throughout the 1,000-second period of illumination, we took temperature readings via a FLIR camera Here in figure A, I have saline, which was illuminated 5.5 watts at the 1,000-second time point. And in figure B, I have gold nanoshells, which were concentrated to 1 meg per mil at the same conditions. There was a 27 degrees Celsius difference between these two, further confirming the thermoplasmonic abilities of the nanoshells. And figure C here, I have our time to 50 graph. It was observed that concentrations of nanoshells 1 meg per mil or higher, regardless of power output used, 4 watts, 5 watts, or 5.5 watts, they were able to reach 50 degrees Celsius in 3 minutes or less. We then moved to investigate the radiopacity properties via Siemens and Dion's high resolution micro CT. We started by concentrating the nanoshells from 0.5 megs per mil to 5 megs per mil, as seen here in figure D. It was observed that the X-ray attenuation rate was dose dependent, as seen here in figure E. It is also seen that nanoshells concentrated 1 meg per mil or higher were able to exhibit a distinct signal from the tissue background, further confirming their ability to serve as a CT contrast agent. Thank you for your time.
Hi, I'd like to share with you some of the work that we do at Queens College in New York, which is part of CUNY. The title of our talk, or actually the poster, is Towards Non-Toxic Nanomedicine, Fabrication of Sol-Gel Hybrid Nanoparticles. The emerging field of nanotechnology has the potential to transform biomedical research and medicine, both in terms of medical diagnostic and in terms of medical treatments. But one of the main challenges in achieving these goals is that many of the nanoparticles that are being considered or developed have a major challenge, and that is that they're toxic in biological systems. So we aim to overcome this challenge by fabrication of sol-gel-based non-toxic hybrid nanoparticles. The sol-gel method is a synthetic method that allows for the formation of porous solid matrices using starting materials such as tetramethyl orthosilicate. One can achieve a matrix that contains a silica-oxygen networks, which is chemically and mechanically stable, optically transparent, and most importantly, non-toxic. And such matrices have been used to also immobilize different analytes and be used for a variety of applications such as sensors or biosensors. So here we present some of our work on different fabrication methods that allow the formation of silica-based hybrid nanoparticles, which we term nanogels, that have improved size, solubility, and stability, and are well suited for encapsulation of biomolecules for applications such as drug delivery or to serve as nanoprobes.
Recently, research from our laboratory summarized on my poster revealed that the major components of high-density lipoproteins are cleared from plasma at different rates. Cholesterol is hepatically extracted by spontaneous transfer through water and by the HDL receptor. This occurs on a time scale of minutes, which is similar to the rate of in vitro spontaneous transfer from the HDL to other lipid surfaces, including LDL, the major cholesterol carrier in human plasma. In vitro, transfer of phosphatidylcholine from HDL to LDL occur on timescale that is a function of acid chain length. The rate of HDL to LDL transfer of the major plasma component of the correct mRNA vaccines occur with a half-time of 6 hours. However, in vivo, the same phospholipid is cleared from plasma within minutes because it is hydrolyzed to free fatty acids under lysolization by multiple plasma enzymes. Lastly, the major HDL proteins, ApoA1, is cleared by a hepatic receptor with a half-time greater than 6 hours. This data suggests that the performances of lipid nanoparticles could be improved by modifying the structures of the key components, especially the phospholipids. This change could include using phospholipid with long acid chains and with a structure that are hydrolysis resistant. The latter could include those contain ether in place of ester bonds or branched fatty acid chains.
Good afternoon, everyone. Today I'm going to talk about RNA-based therapy for familial hypercholesterolemia. Familial hypercholesterolemia is the most prevalent genetic disease worldwide. It is characterized by elevated low-density lipoprotein cholesterol in serum and increased risk of cardiovascular disease. It is often caused by a mutation in LDL receptor. The LDL receptor is responsible for hepatic uptake of LDL cholesterol and its clearance from the circulation. The current available treatments is not overall optimal to treat FS patients. So we developed a novel mRNA-based therapy to overcome the limitations of currently available treatment. To do so, we design, manufacture, and characterize LDLR mRNA constructs, and then we test its in vitro expression in Hb2 cell lines. Then after that, we test its functional properties by performing LDL uptake assay and found that LDLR mRNA increases LDL. Then to do animal tests, we synthesize two different lipid nanoparticle formulations. One is cationic and another one is ionic. And then we encapsulate GFP mRNA in lipid nanoparticle formulation and run real-time analysis of GFP expression in oncocyte, and then found that YAMC3 LNP are better for our study. The overall goal of the study is that we want to bring this new therapy to the clinic. Thank you.
Hello, everyone. My name is Rhonda Holgate, and I work in the Cooke's lab in the Center for Cardiovascular Regeneration. My work is on using mRNA therapy to accelerate skin repair and rejuvenation using a lipid nanoparticle delivery system. Burn and chronic non-healing wounds are a substantial economic challenge and public health burden. So far, the standard of care for these wounds are skin grafts. However, skin grafting comes with a set of challenges. Usually, a split thickness skin is removed from a donor site, which is of a similar size to the wounded site. Therefore, a large wound requires a large surface area donor site skin. This leads to donor site morbidity and pain, longer hospital stays, multiple surgical procedures, and usually an unsightly appearance from the mesh used in the grafting procedure. Therefore, in collaboration with Avita Medical, we're working on a novel approach to eliminate the need for extensive skin grafting and promote efficient wound healing using the patient's own skin cells. Delivery of telomerase RNA to skin cells and then using these cells to promote wound healing is an approach that shows great promise. So far, our data shows efficient uptake of our LNP formulation with excellent RNA expression. We're at the stage where our experimental model is established, and we're working on determination and measurement of enhanced healing using molecular biology techniques that assess skin quality. The success of this therapy will indeed transform the way we approach wound healing and skin rejuvenation. Thank you.
Hello, my name is David Chang. This is my poster presentation for the telomerase nanotherapeutics for enhanced skin wound healing and regeneration. This is an industry-sponsored project from Avita Medical. And the main purpose of this project is to demonstrate that the telomerase RNA therapy would be applicable for skin wound healing as well as skin rejuvenation. And the idea is simple. We are packaging the telomerase third mRNA in lipid nanoparticle. This will be delivered to primary skin cell that Avita Medical has developed in helping wounded patients. And this type of skin cell is known as RES, regenerative epidermal suspension. Basically, a healthy part of the skin from the patient will be isolated and they will be suspended in single cells. And this single cell will then be reapplied into the larger area of the wounded area. And our lab, Dr. Cooke's lab, has developed this lipid nanoparticle encapsulated mRNA that can be added to the rest and then reapplied back to the patient to have an enhanced healing capability and possibly skin rejuvenation. There are 3 goals that need to be accomplished within a 18-month period. We need to demonstrate that the cellular delivery of TER works. So that means that our lipid nanoparticle can be applied to the primary skin cells. Second, we'll have to demonstrate that the skin cells that are transfected with the lipid nanoparticle will have an enhanced activity in the wounded area. And third, we want to demonstrate intradermal delivery of TER to the human skin. And this will be applied to humanized skin model.
I'm Nada from Dr. Kissa's lab. Today, I'm presenting our project entitled A Circular RNA Vaccine for COVID-19. As you know, COVID-19 caused 4.8 million deaths around the world within 2 years. Fortunately, mRNA platform using spike antigens was a quick solution to develop and mass-produce vaccines. However, it still requires a 2-dose regimen and may present a waning of antibodies level after some time. Our project aims to produce an RNA vaccine that is efficacious as judged by neutralizing antibody production with 1 single dose. To achieve this, we use circular RNA, which is more stable than linear mRNA. We first prepare a plasmid with SARS-CoV-2 spike, which will serve as a template for the in vitro transcription. And then after that, we circularize and purify our RNA. After several quality control steps, one of which is shown here by the agarose gel, we check the transcript level using qPCR and saw that the circRNA levels shown in blue remained more stable compared to linear RNA in the cells. Our circRNA is also translated into spike protein for at least 72 hours, which is considerably longer than when we tested with linear mRNA. Overall, our RNA vaccine is stable and produces a high level of spike antigen. Our next goal with our collaborations is to optimize and deliver the candidate vaccine and test in vivo using a challenge test. And our goal is to apply this technology to develop further vaccines for other diseases. We thank everyone for supporting this project at Houston Methodist.
Welcome back to the Kostas Symposium. We're starting our panel now on the future of cardiovascular nanotherapeutics, and I'm joined in the studio by Dr. Francesca Terabali, who's assistant professor in orthopedic medicine, and Biana Godin, who's assistant professor in nanomedicine. And by video, I'm joined by Bruna Corradetti, who you heard earlier today, Roman Sukoverson, who's the scientific director of the RNA Corps, Dr. Henry Powell, who you heard also earlier today, and professor of bioenergetics, and Dr. Peter Cullis, who is joining us from University of British Columbia. Welcome, guys.
Glad to be here.
So today we're going to be talking about the future of cardiovascular nanotherapeutics. And I'll kick off the session. We're each going to talk for about 2 to 5 minutes, and then we'll have a panel discussion. And if there's any questions from the audience, we'll take those as well. For my part, I would say that the future of cardiovascular nanotherapeutics is limitless. And because I'm Irish, I'll say more than just that. I do think that, in part, the future is limitless of cardiovascular nanotherapeutics because of the happy intersection of RNA and nanomedicine, which has already resulted in saving countless lives. I mean, it's really incredible to see how rapidly this field has taken off. I think, as we all know, there's a potential here to replace the recombinant protein industry, but even go well beyond that because RNA is so flexible and so rapidly made. It has superior safety with respect to DNA drugs or other forms of gene therapy. RNA molecules can be personalized. We can target undruggable targets, such as enzyme deficiencies, and the therapeutic development is rapid, as we've all seen with Moderna and BioNTech's vaccines. So here at Houston Methodist, we are also developing our own roadmap to RNA therapy, and we're helping small companies and academic groups develop their approaches for RNA, taking our RNA ideas all the way to the clinic. That's our intent. And we're able to do that because we have a lot of great people here and facilities that really facilitate the generation of RNA ideas, RNA innovation, RNA synthesis, its purification, its validation. We have the GMP facilities to make it clinical grade. We have wonderful GLP animal facilities to do the animal testing. We have regulatory assistance. We have our nanomedicine folks, our nanomedicine team, and we really have all of the elements that you need to take RNA all the way to the clinic. We have a first-in-man clinical trials unit, and we have a big hospital system for doing such clinical trials. And here's some of my favorite people on this slide. Our nanomedicine faculty that help us in making lipid nanoparticles. And pictured is the instrument from PNI, which Peter Cullis had a lot to do with in generating this instrument that helps us to rapidly make lipid nanoparticles and do it clinical grade. So we have your machine, Peter. And then we also have a nice interaction with VGXI, a partnership. This is a local Houston company that was making DNA drugs. They saw what we were doing with RNA, and they decided that they wanted to partner up with us. And they have gone from a 150,000-foot facility in the Woodlands. They just broke ground last year on a facility in Decent Park, 44 acres. They're planning on making a 1 million square foot facility for nucleic acid drugs. And then, of course, we're doing RNA innovation here as well. We're growing that group. We're recruiting RNA faculty. This is Dan Kiss, who's made a circular RNA vaccine. And this is going to increase the applicability of RNA for other purposes. Because it's longer lived, you need less RNA to do what you need to do. In terms of our own cardiovascular RNA portfolio at Houston Methodist, Roman Sukoverson has a lot to do with this, and his team, starting up in the left upper hand corner there, cellular longevity. The telomerase technology is something we're very excited about. We demonstrably improve human cells, extending their lifespan and improving their function. And then we have a couple of RNA drugs that we're developing for high levels of cholesterol, familial hypercholesterolemia, where the LDL receptor is deficient or absent. PCSK9 monoclonal antibody can be encoded into RNA as well to lower cholesterol. You heard today from Eduardo Marban about biological pacemakers, and he uses TBX18, the transcriptional factor that is a master regulator for pacemaker function in the heart. And so we're working with him on that. Across the street at Baylor, we're working with our colleagues there on a vaccine for Chagas disease. It's a trypanosome that is a major cause of heart failure in South America. Bad news is that with global warming, that parasite's moving north. Then also Keith Euchre has a vaccine for heart failure, and we're making his HSP60 RNA vaccine. So a number of exciting new developments in RNA drugs for cardiovascular. So we don't inhabit the same ecological niche as the big three RNA companies. We're in a different ecological niche. We want to help small companies, academic groups with great ideas about RNA get all the way to the clinic. So I think the future is bright. I think there'll be a lot of small companies that are developing new drugs. There'll be a lot of diversity, a lot of new drugs, and we hope to help in that process. So now what I'd like to do is turn the floor over to Francesca Tarabali for her remarks.
Thank you, Dr. Cooke. Hello, everybody. Thank you for the invitation here. I didn't prepare any slides for this part because I will present very short after this panel my research. But what I wanted instead to touch base with you is the opportunity that we have right now in this precise moment after this, what I call it nano revolution. You will see in my presentation, I call it nano revolution this year. It's true that we passed through a pandemic, but at least we can, if the life give you the lemon, right? We make lemonade. So right now is the moment to start to develop in a pipeline of nanotherapeutics, especially if you see the list of clinical trials with nanotherapeutics is increasing, and this is the moment for all of us to start to come out with a new approach, but more importantly with translational approach. For my experience, I was a basic researcher, and then I end up at Methodist, in which actually they teach me how to be translational. And what is important, I believe, of our group of the Center of Nanotherapeutics, as well as the Nanomedicine department, as well as my department, is that we know the basic science, and now we have the opportunity to bring all our knowledge in the clinic. And I believe that this is the moment. I believe that now we have to focus our efforts. It's true to come with new solutions, but at the same time have all our knowledge and move it as fast as possible in the clinical scenario. Because it seems easy. Right? It seems easy that we develop a nanoparticles, in my case, or a nanostructure object, and that is pretty nice, and I get a good publication out of it. But then it cannot reach the patient, which is my ultimate goal. That is the ultimate goal of all the people over here. So we have the honor to be present in the science, in this field of science, in this moment, and I think that we have to exploit it and push as much as possible all our technology towards the clinical side, pushing as much as possible the translation, so the scale up in our case, and thank God that we have Dr. Callis developed his machine that can be easily scale up the process of nanoparticle encapsulation and nanoparticle formulation as well. But also the regulatory part has to help us to shorten as much as possible and give us the regulation to put on the market these new nanotherapeutics. For what pertains to cardiovascular research, if you check in the clinical trial, there's not too many right now, but I think that Of course, with the boost that we have with the vaccine this year, and with the boost that we will have shortly after with all the other nanotherapeutics, we can exploit this kind of medicine also for cardiovascular application. For sure, we need indication, and this is, I think, the good job the Methodist does, the conversation between the clinician and the scientist, and the clinician that comes to us as a scientist and say, listen, I need this one, because in the clinics we don't have anything. Most of the time we don't have this exchange of perspective with the clinical practice, but with this new revolution, I believe that is the time right now to join and let's say shorten this gap between the clinical part and the translational scientists, and move as fast as possible any solution to the in the clinical scenario. That's it. This is what I want to communicate. Since I believe that I'm a translational scientist, I love to be in GMP, and this is, I think, is important to push right now.
I love your passion. Dr. Taravalli. Let's hear from Dr. Bruna Cordetti.
Yeah, so I would like to join what Francesca just mentioned about the fact that this is the moment, this is the era where we really need to take advantage of the environment we are experiencing and put together the knowledge that we share from different perspectives, of course, and different backgrounds. I also would like to share just one slide that summarizes my point of view on what could be the future of nanomedicine for cardiovascular, for the treatment of cardiovascular diseases. Because we've been talking today, and probably other presentation will come to discuss about the role of exosomes in the development of RNA therapeutics and lipid nanoparticles as delivery system for therapeutic agents with a specific focus on the RNA. So what I really believe is that we've been also discussing about the possibility to target specific cell type and the possibility of exploring the content of these exosomes, not only in terms of RNA, this is my personal point of view, but also in terms of lipids that they contain, because it is clear and every day more clear that the lipid composition of these particles also play a role. Now, what would the approach be? Would be to restore what the body already knows, the healing capability of the body, right? So we don't have to include anything that the body doesn't know exactly, that doesn't serve to the body to activate its own machinery. And so looking into the natural molecular and chemical systems that are exploited by the body to induce regeneration, to induce repair, to provide protection to a tissue. That's a way to go. So we've been discussing today about the possibility to target specific cells, also to about the possibility to take advantage of other specific cells for the production of these therapeutics, knowing that there are also limitations in the production of these exosomes. And so the reason why I really want to push this idea is because for the development of RNA therapeutics with a specific effect for a specific application, we can use them as an example. And the case I propose for the phosphatidylserine is just one of the several usage that we can do by taking inspiration from them. And in that case, merging what we know from the nature with the cutting edge technologies that we have available at the Houston Methodist, but also in the institutions we collaborate with, it would be really possible to develop scalable approaches, scalable therapeutics with a standardized approach, but also make sure that we can actually produce the amount of therapeutics that we really need, with the possibility to tailor them for any specific application. Yeah, with this, this is my point of view.
Thank you, Bruna. That was wonderful. And now I'd like to turn to my comrade in arms, Dr. Roman Sikoverson, who's the scientific director of the RNA Corps. And Roman and I talk every day about RNA and RNA drugs and what we can do. Roman, tell us what you think the future is.
Sure, thanks, Dr. Cooke. Just give me a second and I'll share my screen. I do have a couple of slides. Dr. Cooke just provided a nice overview of our center. I don't need to do that, but you know, as a person with a special interest in RNA therapeutics, when I think about the future of cardiovascular nanotherapeutics, I look at that through the prism of the RNA, of course. We know it's a biological software. Yes, COVID proved you know, that RNA can be used successfully in the clinic, but of course, the technology is so powerful that it can be applicable far beyond the vaccine and infectious disease. And so I agree totally with Dr. Cooke. Indeed, my point on the future is reflected in this paper that we published this year. And the title of the paper, The Limitless Future of RNA Therapeutics. And I think we just can easily adjust the title, The Limitless Future of Cardiovascular RNA Nanotherapeutics. And doing some search in the literature, I found, to my surprise, that there is a broad range of cardiovascular applications where RNA therapeutics are being developed right now. So it covers the whole spectrum of CV diseases, from lowering the LDL cholesterol, and that's what we're working on, for instance, and you know, all the way to treating arrhythmias and all other things. So it's quite exciting time to see how RNA therapeutics emerge for the cardiovascular diseases. And of course, they can't do that without the nanotechnology. So this paper that I'm showing here, this, I believe the first publication in 1989, where RNA was transfected or delivered into the cell using the lipid nanoparticle. And so a little bit over 20 years, we have this publication that recently emerged that reports the interim results of the first clinical trial that uses RNA with nanoparticles to treat the disease that's very relevant to the cardiovascular field. So the amyloidosis, we know, actually kills the people because the amyloid gets deposited into the heart. And so the interim results are very promising. So we do see the great progress, and I believe it's just the beginning. And to summarize my vision, I think that RNA and lipid nanoparticles are the yin and yang of the transformative medicine.
Thank you, Roman. I love that
So
yin and
sharing
yang.
here.
And I'm feeling it up here, the yin and yang of RNA and LNPs. Diana. Dr. Godin is assistant professor of nanomedicine and will now give us her views of the future of nanotherapeutics.
Sure. How do I switch to my presentation? Yeah, so I have a few slides too. So when we're talking about generally about therapeutics and medicine, we always, as Francesca mentioned, everybody probably mentioned their presentation, the opportunities of nanomedicine are definitely directly correlated with the challenges that we encounter in the medical field, right? So we're talking about, in terms of therapeutics and diagnostics, is early detection. It can be point of care diagnostics, targeted and safe therapeutics, of course, and any other fields. But what I want to focus today is actually prevention, because we didn't talk about prevention, how actually nanotherapeutics can be used for prevention. You know, like I believe in 1500, the Erasmus said that prevention is actually much better than the cure. And he didn't say it about the medicine or nanomedicine, but I mean, it's definitely true for medicine as well. So if we're talking about historically, the first FDA-approved nanomedicine, and Dr. Koulis, of course, participated and somehow in the development, and I know very well Dr. Barinholtz that also, I mean, that developed this. the most important effect of this first FDA approved nanomedicine was actually prevention of cardiotoxicity of this drug. Toxic for the heart, and it was the limiting toxicity in terms of the therapeutic index. And so when this drug was approved and clinically used, then there is the study that actually summarized a few clinical trials about 20 years later, and one of the effects that they all found is that it was a remarkable reduction in the cardiotoxicity. So encapsulation in liposomes prevented cardiotoxicity of the medication, a very useful medication, but it was again, the cardiac toxicities were the limiting toxicities of it. So nowadays, I mean, I think we've mentioned almost in every presentation today, and obviously from the development, as Dr. Koulis was mentioning, and translation and mRNA that Dr. Cooke was mentioning. So what do we have? The most commonly used nanomedicine today is COVID vaccine, right? Now, we did not talk about this aspect. Actually, COVID, there is a very specific bidirectional interconnection between cardiovascular diseases and COVID. So it's not only that cardiovascular diseases, the presence of cardiovascular diseases predisposes to having more complicated disease when the person is infected, but it's also the other way around. So actually having COVID, it actually increases significantly risks of cardiovascular diseases afterwards. And if one of the papers on the American Heart Association website that I found is actually mentioning that the growing number of studies suggest that many COVID-19 survivors experience some type of heart damage even if they did not have underlying heart disease before they got sick, and even if they were not sick enough to be hospitalized. So, and obviously there are several possible mechanisms that are listed in the literature, but of course there is a direct damage, which is connected to the fact that COVID is entering the cells using the receptor that is widely expressed in the cardiovascular system, SE2 receptor, as well as, of course, a cytokine storm that is the very prominent inflammatory condition that can damage vasculature and cardiac tissue. So, as I mentioned, that we're currently using a preventative measure from the future cardiovascular diseases. I used 3 times already. So, and this is the COVID vaccines. And the other aspect that I want to talk about, the prevention, is actually that is not currently in the clinic, and I believe that this is one of the future directions we can take. And this is prevention of cardiovascular diseases and complications during the pregnancy. When the person is pregnant, when a woman is pregnant, there are 2 bodies develop in one, right? So, and we can prevent fetal cardiovascular diseases as well as maternal cardiovascular diseases using nanomedicines. And I will, in a minute and a half that I have left, I'll try to kind of pinch into that. So, for example, the tocolysis is a very important way of prevention of the preterm labor. And preterm labor currently happens in about 10% of the successful pregnancies. It is a huge financial burden. It brings billions, actually tens of billions. I think I have the number, 65K per birth, and then $30 billion annually, only in American, in the US. So not only that, I mean, obviously it's a financial burden because it causes a lot of, it can cause a lot of problems in the growing fetus, because the fetus is not well developed when the baby is born. So indomethacin is one of the medications that is used for tocolysis to prevent the preterm labor. The problem is that it was found that indomethacin is causing a stricture of ductus arteriosus, and this is a stricture of the blood vessel in the heart of the fetus. And when it happens, it needs to act in the maternal vasculature. But it actually, when the drug is given, it crosses the placenta freely because it's a very small molecule. So the placenta that is connecting between mother and the fetus can be viewed as a mesh. If we encapsulate the small molecule in a larger entity, it actually does not cross across the placenta, and it retains in the maternal circulation. By doing that, we actually could decrease significantly the levels of indomethacin in the fetal circulation. And so, I mean, I believe that this can be a future for tocolytic therapies that prevent fetal effects, cardiovascular effects. Talking about maternal CVDs, there are a lot of conditions that aggravate during pregnancy. So there is preeclampsia, there is impairment in kidney function, there should be a control on blood coagulation, and there is a lot of problems with high cholesterol and triglyceride levels during pregnancies, because this is a very big burden on the maternal circulation. So there are several drugs that can be used, but they are not used because they actually cause fetal abnormalities because of the same reason, right? So they cross to the fetus. If we can make sure that the drug stays specifically in the maternal circulation without crossing to the fetus, using the particles, using the larger entities that can encapsulate the small molecules, it will definitely prevent and allow for the use of the CVD drugs in pregnancy, and this way it will prevent the future complications. So I hope I could just deliver my point, and of course, I mean, there are so many aspects we can discuss about nanomedicine, as I described in my first slide. There are very definitely, not only for mRNA, but for nanomedicine, there are endless possibilities. It's not only particles. It's devices, it's different surfaces, it's a lot of things that we can actually use. But in terms of my perspective, I do believe that we need to try to prevent the conditions rather than cure them. So this is my point of view. Thank you.
Nice. Nanomedicine is a preventive.
Preventive,
Very nice.
yeah.
Now let's turn to Professor Kostas for his view of the future.
Okay, I better share my screen here. Where are we? Yeah, I suddenly realized this morning that there was this 5-minute. Let's see if I can get the full screen presentation. So I just put a couple of slides together. This is by no means complete. I should say that I really enjoyed the sessions today. This was certainly and also very impressed by the scale of the operation that you've got going on down there, Jonathan. It really is quite something. I'll have to pay a visit in person one of these days.
We're looking forward to it.
Just to say, there's a few things that are quite obvious now that we're really good at doing already. So I just wanted to emphasize that first, and that is that I think that the liver produces 60 or 70% of the proteins in the body, at least in the circulation, and so it's really built to make stuff. And we can make hepatocytes make whatever we want. So not only that, we can edit certain genes that are present there. So this is technology that's available today, and I think the opportunities here are really incredible. We're already seeing some examples of that in terms of gene editing, which I think. John, somebody mentioned already, and this is just knocking out PCSK9 to get, this has been published by Intellia, no, this was by Verve, a vaccine against atherosclerosis, if you want to think of it that way. Quite a remarkable opportunity there. Using the liver as a bioreactor, this is just pointing out that it can be done, but we can get supramolecular levels or supraphysiological levels of say, erythropoietin in the blood. And we can produce whatever monoclonal antibodies that we might want, or whatever other protein that we might want. So this is the opportunity. Obviously, I'm not an expert in the cardiovascular aspect of this, but has already been indicated by a number of people. There are quite a repertoire of appropriate proteins that we could be expressing, and all of those are fair game at the moment. The systems are available. The therapeutic index, at least for most of them, is achievable. In other words, you don't have to produce too much protein. So now we're talking about what are the most appropriate proteins to express. The other part of this, and this is really what Brenda has been telling us about, is to incorporate these systems into polymer scaffolds and use them as for tissue engineering or gene delivery or wound healing, as was indicated. I mean, all of these, again, this is in slightly earlier days, but it's a very possible thing to do. And so for a center such as yours, really, the sky is the limit in terms of the potential opportunities. So I'll stop there, but we really are in one of those times where the ability to translate is much more apparent than perhaps it usually is.
Thank you, Peter, for that, and I hope the future also holds some collaborations between us, because that would be lovely. All right, well, I just want everyone to know that I am looking at the Q&A panel on the stage right now and the hop-in application. So if you have questions for anyone on the panel, feel free to go ahead and ask that. I'll be monitoring it as we go forward. Well, we have another 20 minutes or so. Let's talk a little bit more about your preventive ideas, using nanomedicine as a preventive. Tell us a little bit more about that.
So again, I mean, during pregnancy, I think it's pretty apparent how you can prevent those, because pregnancy definitely aggravates even. Preeclampsia can cause long-term cardiovascular damage.
Is there something about that placental maternal vasculature that's different from other vasculatures that adds maybe a greater barrier to lipid nanoparticles? Because obviously the lipid nanoparticles are going to have to go throughout the body and are going to have to be coursing through the microvasculature everywhere else. Why would they not be able to course through the placental circulation?
The placental barrier is not only built from the endothelial cells. There are also trophoblasts, and there are other cells that are building this barrier. However, I mean, it's built in the way that the nutrients should pass. I mean, this is the main idea of pregnancy, of course, right? Because there is a growing fetus that should be fed. So the nutrients are usually small molecules, and the drugs are usually small molecules. So the drugs that are used in the medicine and cardiovascular field, I'm not talking about the future mRNA therapeutics, but the current therapeutics are usually up to 1K kilodalton weight, usually somewhere between 300 to 700 Dalton, and they are also amphiphilic. So these molecules, they tend to pass through many membranes in the body. This is normal. So the idea in using the nanomedicine for this purpose is actually to increase the hydrodynamic diameter, right? Not of the molecule, but of what is surrounding the molecule, so that the aggregate itself will not cross this barrier, they will not cross the placenta. It's actually the opposite of what usually we're trying to achieve, right? We're trying to achieve the target, right? to reach the target. Here we are trying actually to prevent it to go to a fetal circulation. So it will not make harm.
Yeah. Speaking about targets, I mean, in your case, you're trying to avoid delivery through the placenta.
Correct.
There are other difficult barriers for lipid nanoparticles, and one of them, of course, is the brain-blood barrier.
Correct.
And just parenthetically, I'm going to be talking about a lot of organs other than the heart, because the vasculature goes everywhere.
Oh yeah.
And I'm a vascular biologist, so I think cardiovascular is also important for the brain. Obviously, the vascular dementia may be the most common cause of dementia.
Right.
Let's talk about that for a minute, about barriers to lipid nanoparticles. Any thoughts about that, Peter? About we mentioned that briefly before about the blood-brain barrier and delivery to the brain. Any new thoughts about how we can use nanomedicine for that purpose? I think you're muted, Peter.
I've been following a little bit the focused ultrasound stories, and I suspect that might be an interesting approach if you can temporarily affect the permeability barrier in a certain region of the brain to allow escape from the circulation of, say, nanomedicines of one form or another. I think that has potential. I mean, there's the other route, of course, is to go in intrathecally or intraventricularly, which is obviously more less preferred. But I do have the sense that we're going to be able to get there, one way or another, particularly for some pretty serious disorders that can affect the brain. It's going to be a bit brutal, first of all, I suspect, the direct injection approach. But I'm hopeful that something like the focused ultrasound can give us the temporary access that's necessary.
Mm-hmm. And in that case, I guess the focused ultrasound is to increase the permeability of the blood-brain barrier. Yeah. But then there's other approaches where the focused ultrasound is used to actually burst the small bubbles. Now, I guess those are not nanoparticles, right, specifically, but those...
No, they're more on the micron size. And that's high intensity, which evidently heats up your skull and is most uncomfortable for parts of the brain. I don't know.
But it seems to work in the heart, however. I think there's some data to suggest in large animal models that approach can potentially be used for preferential delivery to the heart. But again, it's not nanoparticles, it's microparticles that you can burst with the high frequency ultrasound.
Right. But also, I think in terms of diseases, they frequently impair blood-brain barrier. So, I mean, at least in animal models, and of course, I mean, we don't know exactly what happens in the clinic, right? But for example, brain tumors, they frequently have higher permeability in terms of, they even call it differently. They call it brain tumor barrier and not blood-brain barrier because the permeability changes. So, I mean, I guess there is some potential, and there are some studies that actually show increased permeability. And as Dr. Koulis mentioned, I mean, there are also other routes that can be explored. For example, nasal route was shown in the literature, at least, to be more efficient than intravenous, just because, I mean, it's through olfactory route that you can get there. So, I mean, we'll get there. As completely agree with you. We'll get there. I don't know how at this point, but
well, also the delivery intrathecally, right?
Yes.
And then you have to understand what's going on after you inject directly in the brain, but at the same time you have an access to understand what's going on and then progress from there eventually.
Right. But you're right to mention the nasal delivery because that's been used for decades. I mean, I remember in medical school, we were, for patients with SIADH, the syndrome of inappropriate ADH, we were administering something intranasally. Was it DDAVP? I can't remember now, but
there are a few.
But there are some indications for intranasal delivery to the.
Yeah, I mean the circulation in the head is a bit different. So olfactory route can actually get to the brain, to certain regions in the brain, and actually sublingual It can also reach the brain in some cases. So it depends on the molecule. It depends on what we're delivering, right?
Mm-hmm.
I mean, in the case of nanoparticles, it would be much more problematic. But the small molecules can also be.
Bruno was talking about another way to deliver RNA using scaffolds, and maybe you can elaborate a little bit on how that could be applied for wounds and such.
Yeah, so, we always look into the composition of the extracellular matrix that we want to recreate through approaches of tissue engineering. And so in the case of wound healing, Francesca and I actually have been developing several approaches for modulating the phenotype of endogenous cells at the site of wound. Mainly focusing on the inflammatory stage of the wound, which is where the intervention is applied. And in case of using these approaches of collagen or polymeric scaffold, as Dr. Kallis was mentioning, by functionalizing them with a lipid nanoparticle, we need to consider the fact that we need these particles to be accessible to the cells directly. Instead, in the past, we were embedding the particles within the fibers of the collagen as a way to avoid clearance from macrophages, for example, and being able to modulate endogenous cells. In this case, we want to make sure that cells at the site of implant are able to access these particles and uptake them. So that's the reason why we decided to functionalize the collagen scaffolds with particles just on the surface, and as a way to induce the transdifferentiation of fibroblasts into endothelial cells. As I also mentioned during the presentation, this approach has a wide range of opportunities because the number and the type of RNA being utilized for this type of functionalization is incredible and is enormous. And also the target cells represent, there are several targets for the use of these kind of materials. Also, what we need to consider in this case, for example, in terms of the target cell that we want to modulate, that part would depend on the stage of the wound, the process that we are considering for the therapeutic intervention. So in the case of the collagen scaffolds that I was proposing, the idea would be to apply them so that the endogenous fibroblasts are recruited and modulated and transdifferentiated towards the endothelial cells as a way to support vascularization of the wound and healing.
That's fascinating, and you triggered a thought because we cardiologists use scaffolds all the time. We call them stents, and we make wounds all the time too in the vessel wall when we do angioplasty and stenting. And of course that can cause abnormal healing, which we call restenosis, myointimal hyperplasia. And that event has been largely dealt with by long-acting drugs that are coated onto the stent. And I began to think as you were talking about how that kind of principle of long-acting release of drugs over long periods of time could be useful for RNA. And I just wondered if any of you have any thoughts about how lipid nanoparticles or nanoparticles in general could be used for to preserve the RNA and release it slowly over time. Because of course RNA, the problem with RNA is it gets degraded very rapidly.
Well, what I think that for a slow release, like stage release, that we say more than 24 hours, I believe we need something, and now probably Peter Kallis is gonna kill me, something more stable than LNP particles, or something like, I don't know if you remember, or I don't know if in the US there was a machine that delivered this at the time when there was the marble, right? When you were a kid, and you put the coin and the marble come 1 to 1. So either a device that pull out the particles at the right time, or change the particle with a more stable polymer, or a more stable construct that allow the stability of RNA, as always, but the stage release, so a dynamic of release more controlled. LNPs, I don't see them that you have an injection right away, as we do right now, the COVID, and then you can release over the time. It might be the release criteria, if you inject locally, is definitely for diffusion and everything slow down, but you need to come out, I believe, this is my personal opinion, with a device that control the release eventually, either mRNA or of the nanoparticle as well.
I think it should be a hybrid system. I mean, from my perspective, because you definitely need the nanoparticles in order to survive in the circulation after it's released. Okay, you need something, because RNA, if it is just released to the circulation, it's just not there in a few seconds, right? So I mean, the RNases are so prominent, but I agree with you that I mean, it should be some sort of polymeric, probably, scaffold that will degrade.
Will degrade on the time and release.
And release the particles.
To the nanochannel device that Alessandro Grattoni makes that will slowly release the lipid nanoparticles over time.
This is one of the this is not a degradation, but there are also polymers that are degradation based, so that will degrade slowly, and this will be the time limiting step, right? So whenever it's for the release.
That would solve a big problem for RNA therapeutics, because it would prolong the effect of the RNA molecule. I mean, we're innovating. Dan Kissa is, as you know, is generating circular RNAs that last longer. Still, though, I mean, if you want RNA that's going to last for weeks to months, I think you need something like you were talking about, some sort of a hybrid machine that releases lipid nanoparticles over time. Peter, any thoughts about that? About how to modify the nanoparticle approach to prolong RNA effect? I think you're muted again, Peter.
I think I agree with the conversation. I think you're going to see, I mean, right now, the LNPs that are used for vaccines, and to some extent also the ones that are used for drugs like Onpattro, are relatively unstable. In the sense they've been designed that way so that they will transfect the first cells they see, which, say in the case of Onpattro or hepatocytes. There certainly is a lot of scope for getting them to be much more stable, and so there's quite a bit of work going on in that direction. But to say we're going to get to say past a week or so is probably optimistic. And so if you want to get to systems where you're looking at things over weeks and months, you have to combine that, as has been suggested, with some sort of device. We are going to see various evolutions of the LNPs themselves, which are considerably more stable than the ones you see now.
Thanks for that. We've got some questions from the audience, and I should just touch on a few of these. So here's a question. We know that different mRNAs can have very different size ranges, which is true. We work all the way from 200 base pairs up to 10 kb. How does that affect the stability of the LNPs? And if it has an adverse effect, how would you address that effect? So
I think this is the question.
Yeah.
We have the star from
the star here.
Okay, I'll take first go. But there really isn't any intrinsic problem in encapsulating 10 kb versus 1 kb. So the only issue is the number of copies of the mRNA you get per nanoparticle. And so if you're up to 10 kb and 80 or 70 nanometer particle, you probably only get one copy. Now the issues there come that it's like mRNA as opposed to siRNA. If there's one little part of that mRNA that's exposed and gets clipped for some reason, then of course the whole molecule is degraded. And so the stability problems get larger as you get to these larger mRNAs. But the ability to encapsulate, there's really no problem at all.
Mm-hmm. Thanks for that, Peter. And this one is for you, Roman. What do you think is going to be the next step for addressing the long-term storage and stability of RNA delivery systems? So I think there's two questions there, storage and stability of RNA, and then the RNA delivery system that incorporates it.
Sure, that's a great question. Well, I think that it's been partially addressed already for the RNA at least. So we came up with the lyophilization protocols that allow to lyophilize the RNA and reduce the temperature storage requirements for this. We actually done some stability studies that prove that the RNA can be stored even at 4-degree long-term, months. And also, I believe there is a publication, at least one, that describes the lyophilization of the RNA encapsulated into lipid nanoparticles. Here in the center, together with Dr. Godin, we are planning to run, and I think we already done some preliminary experiments to lyophilize the RNA encapsulated into lipid nanoparticles. So I guess to answer that question, a short answer would be by lyophilizing the RNA that's already encapsulated within the particle, we could increase the stability, both the RNA and the system for the delivery.
Thanks, Roman. We have a few more minutes, and I'm wondering if you could give us maybe an update on the work that you're doing with bone, the fracture putty, or the other work that you're doing with nanoparticles for bone healing.
Yes, actually it's the right time because today one big review went out on the application in orthopedics of nanoparticles because it's not very well explored as a realm in the application. Of course, there are studies like more nanomedicine based, meaning with bone cancer. So this is what we do mainly. We try to target primary cancer in the bone or metastasis to the bone as well. But also what we want to try to do is to try to tackle all those inflammatory based conditions of the musculoskeletal realm and mainly osteoarthritis. So right now we want to what my research is going towards that direction. And speaking about biological barrier is another problem because cartilage doesn't have vasculature. So how can I arrive in a place in which there are no vasculature? Vasculature is our highway to bring therapeutics where we want. So we are trying to strategize biomimetic application in order to target this particular tissue. Exploiting inflammation, so targeting the inflammation in order to target the real problem of the cartilage itself. This is where we are going right now in the orthopedics. For sure, there are some product already. They are nano-based. They are liposomal formulation for let's say, augment the lubrication and they reduce the friction in case of pain at the joint. And this is already market available in Europe. Of course, the Europe realm in comparison to the American is different. This is another problem that we face in the technology development, that we are seeing two completely different markets, especially for orthopedics. I still don't know why we have a complete difference between Europe and American market and development, and it's not due to the regulation. It's not the difference in the regulation. It's the difference in the approach of the clinical practice. And so we want to, but this is also something that we can get inspired from, so understand what's going on in Europe and mirror what is happening over there here, and just using the same kind of approach. So for example, a nano formulation for orthopedics it can be possible is that bone itself is very vascularized, so we can eventually think about deliver some therapeutic or RNA-based therapeutic for osteoporosis, for instance, because we can reach those parts. For cartilage, instead, is a little bit more challenging and needs a little bit more invention, because we need to create the avenue to arrive over there, as well as we were discussing before with the brain problem. But we like the challenges.
Thank you, Francesca. All right, well, Peter Cullis, we're going to let you have the final remarks here on the future of nanomedicine in the cardiovascular arena. And we can't hear you, Peter.
The phrase of the year, right? You're on mute. Yeah. Anyway, all I can say is that the path you're on at Houston Methodist is it's certainly going to be very productive. I wish I could get as big an effort going here as you've got going there. It's a very impressive infrastructure you've assembled. The prospect for the mRNA therapeutics is, as you said, it's limitless. We're moving into a third generation of pharmaceuticals. First generation small molecule drugs, second generation biologics, and now we're moving into an era where we can use the cell machinery to make a protein, silence a protein, edit a protein in a way that that nature does it. So it's intrinsically a less toxic and much more effective and targeted procedure. But the other side of this is that it can be highly personalized. And can be very rapid. I mean, once you know the gene that you want to silence or edit or express, the medicine, making the medicine is a time on the order of a couple of maybe it's a month or two, and you can get this highly targeted entity that may be suited to a person that has a particular form of cancer or is suffering from some rare disease. It's going to upend the whole of the regulatory process, aside from anything else. That where we're the normal clinical trial process, which now takes 15 years and $1 billion or more dollars, is no longer viable. With these neuromedicines, so it's a fascinating time and very, very disruptive. I think we're all going to have a heck of a lot of fun with this. The prospects are really very exciting. So I'll just leave it at that.
Thank you, Peter, for those fine remarks, and thank you for all the panelists on a really wonderful discussion about the future of cardiovascular nanotherapeutics. Thank you so much. We'll now turn to our next session on engineered micro and nanostructured materials for cardiovascular disease. And joining me in the studio is a professor and chair of the nanomedicine department, Dr. Alessandro Grattoni, who will introduce our first speaker.
Yes, thank you for the kind introduction, Dr. Cooke. So I'm pleased to introduce to you Dr. Tejo Desai. Dr. Desai is the Ernest Preen Endowed Chair and Deborah Cohen Endowed Professor of the Department of Bioengineering and Therapeutic Sciences in the School of Pharmacy and Medicine at University of California, San Francisco. She's also professor in residence and Department of Bioengineering at UC Berkeley, and the inaugural director of UCSF Engineering and Applied Science Initiative, known as HIVE, which is a very similar concept to what we have designed here with Houston Methodist and Texas A&M, but on the West Coast. Desai research spans multiple disciplines, including biomaterials, engineering, cell biologies, and tissue engineering, of course. She seeks to design new platforms that are enabled for the advancement in micro and nanotechnology to overcome existing challenges in therapeutic delivery. She has been recognized nationally, internationally. She is a Popular Science Brilliant 10 and the Dawson Biotechnology Award. She is the president of the American Institute of Medical and Biological Engineering as a fellow of IMB, CRS, and other organizations. So without further ado, let me introduce to you Tejal Desai with their presentation on engineered micro and nanostructure material for cardiovascular disease therapy.
Hi, I'm Tejal Desai, and I'm at the University of California, San Francisco. Thank you so much for the opportunity to speak here today. Today, I want to talk to you about some of our work in engineering micro and nanostructured materials for cardiovascular disease therapy. To get started, I thought I would tell you a little bit about my lab, which is interested in developing advanced biomaterials for a range of therapeutic applications. We combine material architecture with chemistry, morphology, and manipulating the shape and size in order to address a range of biological conditions, everything from penetrating tissue barriers to modulating the immune system, and how to get drugs or therapies into the right tissues or cells at the right time. But today I thought I would focus on some of our work in using micro and nanoscale materials to modulate the cardiovascular microenvironment. So for a number of years, we've been interested in the area of heart failure, where myocardial infarction results in heart failure in over 20% of patients. So what is going on? Well, after an MI, you typically get necrosis and inflammation within the heart. That results in cardiomyocyte death and infiltration of leukocytes. Subsequently, there is fibrosis, where fibroblasts that are resident within the tissue actually transition to myofibroblasts and become activated, thus resulting in this extracellular matrix deposition. And finally, all of that results in this reduced vascularization to the site of injury, resulting in endothelial cell death and this lessening of vessel formation. And this is a cycle which repeats over and over again, leading to eventual heart failure. So we've been interested in this area of fibrosis, how we can intervene during this transition where all of this excess matrix becomes deposited. And so a number of years ago, we showed that if you introduced structural cues, these are discrete biomaterials that have a certain size and stiffness, that you could actually modulate the phenotype of fibroblasts within the cardiac microenvironment. We showed that these materials, termed microrods, actually lessen the expression of collagen 1 and alpha spin mesylactin, key hallmarks in the fibroblast to myofibroblast transition, but also have a interesting effect on both TGF-beta1 production as well as receptor expression. And we showed that in inflammatory environments, this actual behavior was actually markedly increased. And so we did a range of different studies looking at what are the differentially expressed genes with primary cardiac fibroblasts that were exposed or interacting with microrods and without. And lo and behold, we showed that a whole range of different genes were differentially regulated. Again, most relating to this myofibroblast activation state, where we saw the collagens as well as some of the MMPs being downregulated, as well as cyclin D1, a marker of proliferation, and TGF-beta. And in fact, this behavior was dose dependent. The more microrods or microstructures we put in contact with the cells, the more effect we had specifically on things like TGF-1 beta. We went on to show that this actually can be used therapeutically by injecting these microrods into an infarct model in the rodent. And what we showed is that over 42 days, we could actually see this resulting improvement in the change in injection fraction, where soluble microrods or microrods that were injected versus soluble materials of the same composition, in this case hyaluronic acid, actually had this increased beneficial effect, where we can not only maintain ejection fraction, but actually improve it. And at the local level, we saw that cells interacting with these microrods actually had less collagen deposition around their architecture. So we became really interested in this area of cardiac fibrosis, and specifically TGF-beta. One of the things we realized is that if we could locally inhibit the production or sequestrate TGF-beta, that may be a promising strategy to dampen the fibrotic response. And in fact, if we look at the extracellular matrix, there's a whole host of different players, right? There are multiple cell types, there are collagens, elastin, and one of the things we became interested in were these slurps. These are small leucine-rich proteoglycans. And one slurp in particular, called decarin, has been shown to have some interesting properties, particularly with respect to wound healing and in cardiovascular environments. So decarin actually has a core protein that specifically binds to TGF-beta. And so when that TGF-beta is in the environment, it can specifically sort of soak up that excess TGF-beta and modulate its presentation. Decarin can also serve as a initiator of vasculogenesis. And also interesting to us is that decarin can actually modulate fibrinogenesis, that is the production of collagen fibers and aggregation of that matrix. So we actually created a new formulation of microrods where we take hyaluronic microrods and we load them specifically with this decarin. And what we see is that these decarin loaded rods can actually have a potent effect on the formation of fibrils, whereas we see this dose-dependent response on the size and the formation of collagen fibrils in solution. And we also see this dampening effect, again, with respect to collagen expression and alpha smooth muscle actin. But what was quite interesting to us, and what we've recently shown, is that decarin microrods actually have even a more improved performance compared to hyaluronic microrods alone, or free solutions of decarin or hyaluronic acid. And in fact, the decarin microrods both result in this vast improvement in ejection fraction, but also we see a more maintenance to normal levels in terms of end systolic as well as end diastolic volume. And so we're beginning to really sort of look at the mechanisms for this. But one of the things that's very promising is that when we look at the tissue itself, we see that those tissue samples that were loaded or injected with our DECMERs, our decarin microrods, actually have both a more robust wall thickness as well as a visually recognizable diminishment of this collagen or fibrosis. So that's one aspect of sort of modulating the cardiovascular microenvironment, where we can use biomaterials to both play a role structurally as well as from a biochemical perspective. But we've also been interested in sort of the wider array of cardiovascular intervention. In particular, how can we mitigate inflammation after the insertion of different cardiovascular devices? Things like balloon angioplasty or bypass grafts, recognizing that there is a major unmet scientific need in vascular medicine and surgery. And so for this, we've been working with Michael Conti, who's Chief of Vascular Surgery at UCSF, on developing materials to present a specific class of therapeutics called specialized pro-resolving therapeutics. So what we know is that traditional anti-inflammatories often lower the magnitude of inflammatory signaling. But what we're really interested is not just lowering the magnitude, but rather reducing or lessening the duration of inflammation. And that's where pro-resolving molecules come into play. They actually drive the immune system towards a resolution of inflammation, such that you go into the repair state at a much more rapid timescale. We know that delivery of these molecules have had beneficial effects in a range of other tissues. And we were really interested in whether we could see this as a therapeutic strategy for vascular interventions. And so what we showed, and originally the Conti group demonstrated, is that one particular resolving therapeutic called Resolvin D1, if you could apply it to a gel, in a gel formulation on the vessel, you can actually attenuate inflammation in a traumatized vasculature. And so we thought, okay, how about trying to think about whether we can use this for, again, possible applications in grafts or other ischemic injuries. So the in vivo effects of these SPMs have been documented quite a bit. And we actually reviewed the literature looking at all of the different types of resolvins and what they had been shown. But one of the things we really wanted to look at is how could we continually apply this to this vascular setting in order to change the microenvironment of the vessel. And so we developed two different formulations. One was a peravascular wrap, in which we really applied towards vascular anastomosis, trying to incorporate a synthetic RVD1 that we created. And the second was a targeted nanoparticle formulation, where you could imagine using either in a vein graft harvest solution or directly into arterial injection. And so our biodegradable wrap was essentially a multi-layered thin film device that we really designed to have this asymmetric delivery of resolvin, such that the resolvin would only go into the artery and not into the peripheral tissue. It was flexible, as you can see, quite thin. And showing you on the right, this is the release kinetics of the RVD1, only towards the inside as opposed to the outside. And what we showed in a series of different studies is that we could use this in peravascular delivery approaches within acute arterial injury models. This is one example in a rodent model where we showed that using these wraps loaded with RVD1, we could reduce the neo-intimal area and have more patent vascular interfaces. We also showed this in a rabbit model where we were able to have local RVD1 treatment showing reduction in vein graft hyperplasia at 28 days. And what we noticed with the wraps is that we could, similarly to the gels, be able to reduce the thickness and maintain that over time. And the benefit of using a wrap is that we can apply it once, whereas a gel washes off quite rapidly. And so you have to keep applying it. And interestingly, we saw that the peravascular delivery of RVD1 actually inhibits this early inflammatory response, similar to what we hypothesized, and limits the recruitment of these cells, these immune cells, to the vein graft. And so here we see both leukocyte infiltration as well as macrophage infiltration is significantly reduced with the wrap being able to deliver this RVD1. And so what we have worked on recently is scaling this up, making this wrap something that can be used in large animal models as well as preclinical studies. In sort of learning how to translate what is seemingly a fairly simple device, but a multi-layered film that actually has a potent biologic. And you can see us here putting this wrap onto a vein and actually putting it around this and being able to basically tuck it into place, and it just self-sticks into that area. And finally, as I mentioned, we're also interested in using this approach in a nanoparticle formulation, where we can deliver SPMs, but do this in a targeted manner. And so what we've developed is a PLGA particle system, but the system presents a targeting ligand that is targeted towards tissue factor. This is a peptide found on binding sites that are often presented during injury during the coagulation cascade. And so when you have no injury, you really have little of these nanoparticles binding to the area. However, under an angioplasty where you've had injury on the vessel wall, you actually see that the nanoparticles target to that wall of interest. And in this capacity, we can both deliver the RVD1 in significant levels, and we can have good coverage over the vessel injury site. And what we've shown is that with this targeted approach, we can actually get very good reduction in leukocytes within that area. And similarly, sort of this mitigation of the acute inflammatory response. So I'm going to have to end here because we're out of time, but I just want to end by saying that by harnessing micro and nanoscale cues and combining them with both biological and pharmacologic agents, we have an opportunity to intervene in a number of these different disease areas, and particularly have some exciting potential applications in the cardiovascular space. And with that, I want to thank my lab, the folks that contributed to this work, as well as our funding sources, and happy to answer questions either now or at a later time.
Thank you, Dr. Desai. We do have time for some questions now. If you want to stay with us, we have about 10 minutes for questions. I'm sitting next to the stage here on my computer, and if you have any questions, just send it to the Q&A at the stage section of your hop-in application. That was great. Thank you so much. Myointimal hyperplasia, restenosis, is been largely resolved in the coronary circulation with drug-eluting stents for the most part. It's still a problem in the periphery, and it's a major problem with aortovenous grafts for hemodialysis. So is that where you're headed with your current therapeutic approach?
Absolutely. So we became very interested in exactly what you said, which is the peripheral applications. And as we were looking at this, really looking at what are other places where you might have that, restenotic or integral hyperplasia. And so certainly in hemodialysis, in other anastomosis, even in vascular grafts where there's a mismatch and oftentimes in the mechanical interface. And so really thinking about, perhaps designing materials that have this sort of multifunctionality. The nice thing about resolvins is that you're really, resolving inflammation as opposed to putting in what many of our drug-eluting stents do, which are these neoplastic or sort of anti-proliferative agents.
Mm-hmm. Correct, that's true, yeah. Just parenthetically, when you get to the point where you need to do your GLP studies, we've got great vascular surgeons here, and we actually have a great GLP unit, and we would love to work with you, advance this toward the clinic. So don't forget us.
And we will definitely take you up on that. Absolutely.
Francesca,
No, I have a curiosity actually. What you show about your device that you are doing the scaling up right now. So can you tell me how long did it take to you to arrive at that point from the day that you probably sent your invention disclosure just to have a partner to understand if you share the same struggle?
Yeah, it's a great question. We probably, I would say from when we started, I would, it's probably 3 to 4 years.
Okay.
It was actually accelerated because we were part of this NIH Vita grant program, which is accelerating therapeutics to the clinic, which really helped and sort of incentivized us to get there faster rather than later.
Sure.
But there is definitely, so there's the science, which you want to do, but then a lot of this is not exciting, right? It's thinking about how do you sterilize, how do you package when the surgeon opens it up, how do they know which side is up, which side is down? So there were a lot of little things that we hadn't even thought of as we were doing this.
Yeah, and this is what I was mentioning also before, that we have to form this new category of scientists that find exciting also this part that you were saying, the packaging and the sterilization, because those are data that don't go in paper. Most of the time
you don't publish it.
Yeah, you cannot publish, but it takes a lot of efforts and a lot of time. So I'm a nerd enough that I'm curious every time of the packaging was my limited step every time, because yes, they say, no, you have to put the direction. And I say, what? Yes, the direction. So you had the same problem, I see.
But yeah, and even, it seems like, oh, it's just, a material we've used these polymers for decades or almost half a century. But, just even scaling up, we needed something that was thin enough to be able to conform, and many machining and sort of manufacturing companies weren't able to get that resolution that we needed in terms for the film. So just finding that was a challenge initially.
Go ahead.
Oh, please.
Alessandro.
How long would resolving wrap last once it's implanted, once it's inserted? And are there concerns of potential migration or within the body? And what is the way that you are addressing those?
Yeah, resolvin is, for good or for bad, very unstable. So in fact, once it is released, it is a very short half-life. It is often taken up quite rapidly by the tissues given its lipid composition. And so, which is the reason we wanted to formulate it in something where the polymer actually stabilizes it to some extent and allows for that slow release. So you do not see it sort of dispersed elsewhere in the tissue. It almost instantaneously gets taken up at that tissue site.
You mentioned that docahexanoic acid is the precursor for resolvin. Of course, docahexanoic acid is fish oil, and it has many actually beneficial effects on the vasculature itself. It has antiplatelet action, anti-inflammatory actions, maybe mediated by resolvin. I don't know, but why not just use docahexanoic acid?
In fact, we actually did some early studies looking at that, and there is some evidence. It is just, it probably has some of the same features, but it's not as potent. And so I think to the extent that, this is the precursor that we can actually, and it's taken up much more readily by immune cells, which we're really targeting, we found it to be much more potent within these types of applications. But from I would say the nice thing about fish oil, it is, over the counter. You could you could dose yourself, but it's certainly something that does have some beneficial effects.
Yeah, and giving it locally as you are is a much better approach. I would think probably has more potent effects than giving it systemically.
So and you could take that, fish oil itself, if you look at the constituents of what they put into that formulation, actually has some of the same elements. So yeah, I think you could use that, but this is a much more sort of readily metabolized precursor.
Yeah. Well, thank you, Dr. Desai, for that wonderful talk. I'm afraid we're out of time, and we'll have to move on to the next speaker. But thanks for joining us today at the Kostas Cardiovascular Nanomedicine meeting.
Absolutely. Thank you so much for having me.
Bye now. Dr. Grattoni?
Okay, so I'm pleased to introduce to you the next speaker. You have already heard from her, Dr. Francesca Taraballi. She is currently an assistant professor in the Center of Musculoskeletal Regeneration at Houston Methodist Research Institute and in the Department of Orthopedic and Sport Medicine at Houston Methodist Hospital. Dr. Taraballi research is focused on the development of biomaterial, both injectable and implantable, that can target the immune system toward tissue restoration. Her main research area of interest is to develop new therapies and devices for articular cartilage repair. So she will present today about the biomimetic nanomaterials for cardiovascular application. Francesca.
Thank you. Thank you, everybody. Thank you, Dr. Grattoni and Dr. Cooke, for this invitation. And I will talk this time of some part of my research that instead is dedicated to cardiovascular application. I will start to point out this guy, which is called Thomas Kuhn. I don't know if any of you know it, but he's a philosopher that at the beginning of the century spoke about the structure of scientific revolution. And basically, he showed up that the scientific revolution normally passed through a moment of crisis, and when this, and exactly in a moment of crisis is when the normal paradigm got overcome. And with this in mind, I believe that follow the thought of Thomas Kuhn, we are in the nano revolution, what I was mentioned before during the panel. Finally, with the COVID vaccine, also my mom discovered what I'm doing, because I have a PhD in nanomedicine and she never understood pretty much what I was doing. But finally, we are in a moment in which when you speak about nano and nanotherapeutics, the people start to understand the need and the big variety of things that can come out from this. And what I work in is to develop technologies that are inspired by nature. And why by nature? Because basically mimic the function, the structure, and the composition. We can achieve solution that nature already found, and we can mimic what are the nature solution in order to find a big solution to big challenges that that unfortunately we face every day in the medical field. We are able in my lab to go directly from nano to micro and to control, let's say, the shape and the composition of different objects that we can use to develop new device of new therapeutic solution for cardiovascular application. This is basically a chart that envision all the kind of technology that I have. I will talk today a little bit more implantable scaffold, a little bit of colloidal solution, and also nanoparticles. I will start with this study about congenital heart defects. Congenital heart defects are more often present than what we thought. Basically, is the 1% of the live birth in US have a cardiac defect, a congenital cardiac defect. And what we have right now in the clinic is basically this kind of patch that are able to rebuild or seal this defect in the heart. What's the big problem of this this device, this membrane? That basically they don't grow with the baby, and so the baby needs to go over and over under surgery in order to continue to repair, because unfortunately these are plastics. Plastics is very good material, don't get me wrong, but is not very compatible with the human. It's inert, so it doesn't create too much a problem, but it doesn't grow. So what it was, again, our idea in collaboration with Rice University and Professor Jacob, that now is UC Boulder instead, is to develop a method to develop this kind of patch that is completely biomimetic, and it will be cellularized with cells from that are isolated from the amnios of the a kid that will born with the defect, in order that will be completely compatible with it. And we will have these cells harvest directly from the amnios, and they're reprogramming in pleurestem cell. And once we have the iPSCs, we can, with the material that it control the release of particular molecule and support the growth of the cells that we are implanting, to create this patch that then we are able to implant once the baby will be born. This is, of course, is an overarching idea that, by the way, was pretty much like from NSF. So we decide to develop this dual release of nanoparticles. In this case, are silica nanoparticles developed for sol gel. We have a method to delay the release of silica nanoparticle, embedding them in an outer shell of PLGA, And with these little tricks, we are able to achieve completely different kinetic of release. So in this case, what we want to develop is a system that delivers two factors that induce iPSCs in cardiomyocyte and as much as possible. Then eventually we can discuss about this. But that we want to apply a method dealt with the nanoparticles to deliver this kind of factor at the right time. And this is what we did. These are the example of the nanoparticles. The silicon nanoparticles are really around 260 nanometers, and you can see here these are the nanoparticles. They are very porous, so we can load them with whatever we want, and then depending on the shell that we put outside, or the polymer of choice, or the thickness, on the thickness of choice, and so on and so forth, we can regulate the release of these particles. As you can see here, we have an immediate release for the first factor that needs to be released at the beginning, and more stage release of the second factor, the green line. And so as a proof of concept, we check with the right control, meaning just administer these small molecules directly in the plate. We were assured that we achieve also with the nanoparticles the same grade of differentiation of iPSCs towards cardiomyocyte, and we did it. And we published this a couple of years ago, and unfortunately I don't know how to click with that image, but it's always the fancy thing in which you can see the cardiomyocyte beating. Just imagine that that is a cardiomyocyte beating. With the same concept, so with the same kind of nanoparticles embedding in an extra shell, so we can deliver a different kind of molecules at different times. We also, as a proof of concept, use this electrospan patch, so basically is an implant in this case, and also in this case we use two different molecules to release to enhance vascularization, in this case PDGF and VEGF beta. Is important because these two factors, in order to vascularize the area, needs to be released in a different manner, and if they are not released in a different manner, you cannot achieve a vascularization as well as the stabilization of the vasculature. So this was our aim, and this is a gross image of what had happened. You can see that when we deliver with the same system only PDGF and VEGF in the time, we do achieve some vascularization, but it's not the same when we administer both of the growth factor. And this is particularly important because growth factor used in clinics has a lot of degradation problem, stability problem, and instead with these nanostructure particles, you can see that we were able up to 21 days the release of the two growth factor. They are basically big proteins. Now I want to talk a little bit about biomimetic nanotechnologies still for cardiac approach. This is actually was a project that was successfully sponsored by the Kostas Foundation, and it was in collaboration with Dr. Valderrabano. Basically, he wants, as I said before, I'm often repeat myself because I mentioned everything in the panel before, but I love the fact that in Methodist we can talk with the doctors, the clinicians. They can tell us the needs that otherwise we will not envision anymore. So he came to me, he came to me and he said, well, Francesca, we need something to seal this part of the heart because sometimes there is a problem in this articular part of the heart called LAA, and I want to seal it as much as possible in the way that then necroticize and reduce the problem of atrial fibrillation. And he showed with some device that are already available in clinics, like the Lariat, which is as well as the Watchman, to close this part of the art, but unfortunately both of these devices has limitation in the closure. So sometimes they can arrive over there, but since this particular part of the art is different and there is a lot of morbidity patient by patient, we have to create something that we can inject and it can stay over there, in which the flow of the blood is very turbulent and it can seal. So I say, okay, let's see what is stay in a place where it goes. And actually we have mussels, are animals that stays pretty much anchored to a rock and, and exploit a lot of turbulent flow. And so we develop a material, nano-based material, nano-injectable thermosensible gel that is able to be injected at the cold temperature and turn in a stiff gel, and is also adhesive because we introduce these molecules in the structure chemically, of course. And as you can see here, we try first to see if we can inject and block a vein on an artery. We try both in a rabbit model, and then we went to a large animal model as well, and we approach this kind of procedure with the lariat. So basically we approach from the external part, we put this laser, and with the catheter we enter inside and we put the gel. You can follow the gel because also the doctor told me, Franci, I have to be able to see the gel, so I have to put inside the structure of the polymer something that is radio opaque so they are able to see it. So you can see this black part is when the gel is injected in the LLA. Here you can see the LLA is over there, still over there. When this is when it's completely sealed and the catheter is being retracted, and this is actually after 5 weeks, you can see that there is a complete seal of that part. So the LLA is completely necrotic, and so we solve, let's say, the problem. And this particular device now is in further optimization for the scale up. Finally, I want to speak about nanoparticles and biomimetic nanoparticles for this problem. We mentioned before, actually, the people that spoke about this before on me are way more experienced than me, but I will try to do my part and speak about the biological barrier. We have a lot of problem. We barely mentioned during the panel the problem of biological barrier, because we think that when we have nanoparticles, these nanoparticles can travel through the blood, arrive at the target site, and deliver the payload. Unfortunately, we wish that it's gonna be like this, but we have a lot of problems which are called biological barrier. That is like the MMPs, so our immune system most of the time cap these particles. The endothelium need to be overcome if we want to target the stroma of a tissue or a tumor or whatever, the brain, as we talked before. The extracellular barrier sometimes is pretty dense, especially in solid. Let's think about solid tumor, but let's think about fibrosis, how the tissue is very dense and compact, so these particles need to enter over there. And of course, at the end, we have the cellular endosomal compartment, which can be another moment of failure of our nanoparticles. So we got inspiration from the nature, and we say, as we say before with the muscles, also this time, what is nature does already this thing. So avoid the MPS, extravasate, and target, and leak out from the endothelia. Actually are a leukocyte. Our leukocyte, you can see over here, they arrive at the site where they're supposed to arrive, and they extravasate, and of course they are not kept by other leukocytes. So this is where it comes from, our idea to develop lipid nanoparticles that include the leucosome membrane protein in order to overcome all those biological barriers that I just mentioned, but at the same time in the development we understood that these particles are particularly good, as well as the source cells, to target inflamed endothelial, because as leukocytes target inflamed endothelial as well as the particles that come from them. So this is what open a huge amount of disease that can be targeted with this system. Of course, one of the first is the cardiovascular disease, because we saw and we heard about how much inflammation is involved in cardiovascular diseases, but as well as cancer, of course, and the musculoskeletal disease. That's why I'm part of the orthopedic department, and so on and so forth, because this tool is particularly good to target inflammation. So we let's say, play with it a lot, and we try to understand how we can modify in order to tune these particles, how solid is this particle, how much I can scale up these particles as well. This is a very good work that is being published at the beginning of the year by Dr. Asaf Singer and Dr. Manuel Sushinita. And you can see that we can include inside the particles different amount of protein, and with different amount of protein we can achieve different kind of functionality of the particle itself. And we want to understand, of course, I have to tell you that as Italian I put a Super Mario here, that is my personification, in order to travel around the vasculature network. This is what my particles has to see, has to escape the immune system, as we mentioned before, has to arrive at the target tissue, it has to extravasate inside the endothelium, and of course has to deliver then the payload instead and eventually save the princess, but I didn't have space in the slide. So, and this is what we study right now. We want to be sure that our particles is efficient in every of these steps as much as possible. And here you can see this is a live image in intravital microscope, so we are able, and we have here a Methodist, the intravital microscope core, in order that we can able to see. This is the green part is the vasculature. In this case we have a cancer vasculature, and you can see the target of this leucosome at the side of this vasculature throughout the time. So they can really target and here you can see in the time is basically 15 minutes. You can see how rich there is at the side of the vasculature ready to extravasate. As well as we are studying what's going on in the endothelial cells. This is a study that we did in endothelial cells because we want to understand, first of all, if it works as we say that it works, and I think that is important for the clinical translation, as well as is if also this product that we put in the body can be recycled by the cells or can be used in a different way, or as Dr. Corradetti was saying before, what is also the part of the shell, so the lipids. So we study, in this case we load the leucosome with sRNA, and we study the intracellular trafficking, how it's called. So you can see that these are the way in which the particles can enter in the cell, and we can target basically this kind of process. You can see here there are some particles at the external part of the cells. You can see some particles that are inside the cell. If you follow the green, if you follow instead the red, the empty red is actually our sRNA. So you can see that is outside the endosomal compartment, so we overcome also the last barrier. So we are ready for the princess right now. This is very important. I want to mention as the last piece of this presentation, the fact that we want to use this nanotechnology, but this is a real message that I really care. I want to let you understand that every time that we release a nano object in the body, we have to be sure 100% that this nano object doesn't affect too much the regular type of the cells. This is a study that we did with different kinds. These are not liposomal or LNPs, these are silica nanoparticles, but you can see how the cells, these are endothelial cells, recycle these particles, and how they have a transient moment of autophagy activation and intramembrane biomarkers, let's say, tuning. And we can also take an effort for this, because of course autophagy in a lot of disease is altered or is an important part of some disease. So we can use nanoparticles eventually, just bare nanoparticles, to elicit some type of molecular pathway inside the cell cells in order to achieve the outcome that we want. But it's very important that we take for the translation of this nanomedicine in account the system of biocompatibility and what are the biocompatibility standards. Having said that, I really want that we will see in this list, which is basically the ongoing clinical trial with nanoparticles. We want to see more clinical trial nanoparticle in cardiovascular application, because I do believe there is a huge margin. There are only few in this list that unfortunately, maybe for you is not so small, but I'm pretty sure there are a couple of LDL for arteriosclerosis. And having said that, these are all messages that I really care to send. The fact that there is a lot of challenges for this nanoparticle and nano object in the regulatory terms, that I hope that with this new nano revolution we will be able to overcome. And I'm very, very glad that I'm part of it. Like to come back to my mom and say, you see, that that PhD was the right choice, even if you didn't understand what it was. I want to acknowledge, of course, all the people that made this happen. They are the people of my lab. You see two colors. These are my actual people present in the lab. Of course, we have a lot of turnaround. These are the people that been part of the data that I present, but now are in a better place for sure. And the agency that sponsor my research, NIH, CPRIT, DOD, and of course the Kostas family, because they will be very important for what I did in the cardiovascular, as well as what I will do later on. And of course, I want to thank you for your attention, and I'm happy to get any questions.
Thank you, Dr. Tarbali, for that wonderful talk on biomimicry of nanoparticles. We only have time for 2 quick questions because we're running a little bit behind. Dr. Grattoni, do you want to start?
Yes, Francesca, what specific membrane protein will you incorporate in the leucosome to target inflamed vasculature?
This is a big question. So I tell you what we are studying, because right now we approach, let's say as an engineer, so we say, okay, this is pure, we put it over there and we inject. We did proteomic analysis, so we have an indication of which protein we are able to embed. And what is most of the time for exploit the tropism, we always work with the CD47, CD45, CD11B. Those are the markers that we check. But I'm pretty sure that we don't see right now, we don't have a clarity of the spectra of how this particle works. I believe there are extra iteration more than just LF1. ICAM1 directly on the receptor that we introduce. And for sure, I anticipate a question. This can be a short limitation of the translation of these particles, because we cannot predict perfectly that those are the membrane protein that we are able to introduce in our formulation. However, we have a lot of repetition that assure to you that at least when we have those, we have the effect.
Mm-hmm. You know, my question actually relates to something that you were alluding to just a minute ago, and that is that in your original leucosome, you were taking membrane proteins from leukocytes and incorporating that into the lipid nanoparticle, and then you were going to use that to deliver things to the vasculature that was inflamed, and it worked. But your control experiment, where you just used the leucosome itself, also had anti-inflammatory effects. So the leucosome itself
and actually
seems to be anti-inflammatory.
I have to tell you that we never really claimed that part, just because we are afraid to claim it, because we don't know yet how to explain it. Mm-hmm. That the particle itself, just the fact that they have this membrane, is able to reduce the inflammation just per se without any anti-inflammatory cargo. However, we might claim in some of the paper, for sure not in the first Nature material paper, because we were like, oh my god, no, if they ask something else, we don't know how to explain. At least we are technologists, we are not like deep in the molecular part, so we know that it's reliable, that the yield is good, that it's working exactly as it's supposed to. but we don't know yet. Yes, I can tell you we saw the effect, anti-inflammatory effect of the particle per se, but I cannot speculate how it is. We are working on it to understand if it's mediated by an extra interaction from the particles with the immune cells, and then the immune cell, so basically the nanoparticles tune the immune cells that tune the environment, or is a direct interaction between the nanoparticles and the endothelium, and so to tune the environment. It can be most likely both. So the interaction that these particles has with the immune microenvironment that includes also, of course, also endothelial cells, because also endothelial cells can activate and react.
It's very promising, and you know, this being able to target the activated endothelium will really treat a lot of inflammatory diseases.
Yes.
Atherosclerosis included.
Exactly. In fact, one of our project is exactly on arteriosclerosis. I know that you published something, a big paper already on that kind of application, and especially for that application, we need to be sure 100% that we are ready to scale up this platform, and we have rigorous tests and QC tests that tell us yes, this, even if you did like, I don't know, 20 ml of these particles, these are good enough to be injected in a human and be a real treatment. So far we are not ready yet. We are in the, let's say, medium-sized animal model. So until a rabbit, it works.
Well, it's very promising, and best of luck in getting there. I think we're going to have to move on to the next speaker that Dr. Grattoni will introduce. Good job.
Well, I'm pleased to introduce Dr. Omid Vaizeh. Dr. Vaizeh is an assistant professor of bioengineering at Rice University. He's a CIPRI scholar in cancer research. He holds a dual PhD in material science and engineering and nanotechnology from the University of Washington. The Vaizeh laboratory utilizes advanced nano, micro, and microfabrication technique in combination with molecular engineering and cellular and molecular biology to develop platform of implantable devices tailored for in vivo chemical sensing and delivery of therapeutics. Dr. Vaizeh laboratory is developing technologies for the improved management of cancer, type 1 diabetes, as well as rheumatoid arthritis and other autoimmune diseases. Dr. Vaizeh is also a co-founder of a biotechnology company, Sigilon Therapeutics, in Cambridge, Massachusetts, that is committed to commercializing and translating these technologies to the clinics. It will present today about immunomodulatory biomaterials for cell-based therapeutics.
Good afternoon, everyone, and thank you to the organizers for this opportunity to present some of our research efforts in developing immune-modulating biomaterials to enable cell-based therapeutics. Real quick, I just wanted to go over some financial disclosures, and any real or perceived conflicts of interest are managed by our Dean's office here at Rice University. My lab is interested in developing cell-based therapeutics for a wide range of applications. And when we think about the pharma industry today, much of the drug pipeline is actually in the form of biologics. And biologics need to be injected. And for many chronic illnesses, this is actually quite a bit of a challenge, because sometimes it's difficult to get the dosing right. And the chronic nature of the disease requires many, many injections, because much of the biologics have limited half-lives. And of course, emerging in the clinic is gene therapy, and that will be successful for some indications, but it has some challenges. In particular, re-administration is a problem. Ability to treat pediatric population is a problem, and adjusting dosing remains a big challenge. So we're excited about leveraging this concept of cell-based therapeutics, whereby we engineer cells to produce biologics of interest. Many of the biologics that are on the market today are actually produced by cells in bioreactors and then packaged up in vials and actually administered to patients. So our concept is, can we just bring that manufacturing to the patient? And we think the way to do that is to leverage advanced biomaterials formulations. And we're excited to advance on this idea of developing what we call living drugs. These are therapies that are cell-based, and these cells are developed to secrete different proteins of interest based on how they're engineered. And also, these cells can have sense and respond features whereby they can produce the drug on an as-needed basis to match the physiological needs for the patients. So this is what the promise is. And one area that we've focused a lot of our efforts in over the years has been to advance this for the treatment of type 1 diabetes. Type 1 diabetes is an autoimmune disorder whereby, for reasons we don't yet quite understand, the body's own immune system starts attacking the insulin-producing cells of the pancreas. And as these cells are killed off by the patient's own immune system, the patient now has to self-monitor blood glucose levels and administer insulin multiple times throughout the day. For many patients, this could mean 5 to 7 injections per day. And this represents a big burden. And no patient seems to get it quite right because dosing is tricky. And you can imagine for pediatrics or other populations, this becomes even more challenging. So what has existed for a while now is a great proof of concept that you can actually cure type 1 diabetes by leveraging cells. In fact, today, there are what's allowed, and there's good proof of concept for, is to take the insulin-producing cells from a cadaveric pancreas that's donated and actually isolate those insulin-producing cells, re-administer them to a diabetic recipient, and along with immunosuppression, this concept actually works. Many patients are now over decades insulin independent because the cells can engraft and actually facilitate the therapeutic efficacy that's needed. They sense blood glucose and secrete insulin. But the challenge is, because of the immunosuppression, not many patients can actually qualify for this. And on average, only 20 to 100 patients per year undergo this procedure. We are excited about developing biomaterials that can protect these islets and other engineered cells to avoid the need to do immunosuppression. And if we can do this right, the concept is to engineer biomaterials that are porous, that allow nutrients and oxygen and the therapeutics to diffuse in and out. But by tuning the porosity of these hydrogels, you can actually exclude immune cells. And we know that in order for those immune cells to be killed off, there has to be contact mediation. So by preventing access of the immune system to these encapsulated cells, we can actually address this challenge of allogeneic rejection of the transplanted cells. The material that has been widely used for this application, one of the materials is alginate. And alginate in particular has been good for this application because it naturally produces, when you form hydrogels of it, it naturally produces a porosity, which is perfectly suitable for this application. Nutrients can diffuse in and out, but immune cells can't actually diffuse in. The other nice feature of alginate is that the cross-linking reaction that's involved is actually a divalent ionic cross-linking scheme, whereby divalent cations such as calcium or barium are used to facilitate this hydrogel formation reaction. And it's a very biocompatible reaction whereby 98% of the cells remain viable. Now, this concept actually of using alginate for this purpose has been around for several decades. The first was this paper in Science in 1980, where they demonstrated that, alginate does enable protection of these transplanted cells. In this study, they took pancreas islets from a donor, which was immune mismatch, to a diabetic recipient. And they demonstrated without immunoprotection that T cells can find these transplanted cells and kill them off quite rapidly within a couple of days. With encapsulation, the cells persist much longer. few weeks. But what ultimately happens is that a few weeks in, the material is recognized as foreign by the immune system. And as such, scar tissue forms around the material. And this serves as a barrier to diffusion of oxygen and nutrients. And so the encapsulated cells don't live very long. This has also been observed in human clinical trials with a number of alginate formulations, as well as in non-human primates. And so in this much more robust models of both human patients as well as non-human primates, where there's a very robust recognition of foreign biomaterials and scar tissue formation, the observations has been that, it works for a few weeks, but for this to be practical for patients, we need solutions that can enable viability and survival of these transplanted cells for many, many months, if not years, before it can be adopted in the clinic. And when it comes to the biomaterial choices, we know that any material that's implanted into the body provokes an immune response. And what happens is macrophages will recognize the material as foreign. And as they recognize it as foreign, they secrete cytokines, which recruit myofibroblasts and other immune cells, and combined, they build a matrix around the implant to wall it off. And this matrix is dense in collagen, and this serves as a diffusion barrier for the oxygen and nutrients. So the encapsulated cells die off shortly after the matrix forms. So we were excited to tackle this problem and think about ways where we can improve the biocompatibility and actively include immunosuppressive chemical modifications onto alginate to address this fibrosis challenge. And we leveraged libraries of biomaterials, and we kind of borrowed from the pharma industry approach that they take is they do these phenotypic screens whereby they know they have a desired particular outcome. And in our case, it's a material that doesn't get fibrosed, but it's hard to know exactly what the solution is going to be. So these pipelines that allow for parallel testing of hundreds and thousands of materials are quite an innovative way to actually arrive at the solution of finding materials that don't provoke an immune response. The challenge is always coming up with ways whereby you can actually evaluate the fibrotic response in an in vivo setting, because the in vitro assays are not very predictive, since fibrosis involves multiple types of immune cells, and it's hard to recapitulate that in vitro. In vivo, our first strategy was to implant little bits of the material under mouse skin and look for signs of inflammation using this ProSense imaging assay. And where you see a lot of signal, that means it's a material that's likely going to be fibrotic and very inflammatory. So our goal was to find these low inflammatory materials. And then the leads were tested in other implant sites as well as with cells for ability to resist foreign body response of fibrosis. So we can do these screens, and we've done this now with different subsets of the libraries. And what we've shown here is a plot of the first 774 formulations that we tested. So the baseline alginate formulation set at 1, some of the modifications actually make the material more inflammatory, but we're particularly interested by these formulations down here, which were ultra low inflammatory and much less fibrotic. And these lead materials actually work to actively suppress the immune response. And so here is an intravital imaging assay we performed just to illustrate how they work. Without the modification, alginate spheres attract a lot of macrophages, which are in green, and the spheres are in magenta. You see that these macrophages have adhered, they're secreting cytokines and recruiting additional macrophages, and combined, they work to build that scar tissue. This is 1 week in, and already you can see quite a lot of macrophages there. And we contrast that to a formulation, same time 1 week in, whereby they have this small molecule that prevents adhesion of these macrophages. And by preventing that key event, we notice that these immune modulatory coatings actually facilitate inhibition of macrophage activation, and there's healthy number of macrophages there, but since they're not adhering, they're not recruiting more cells, and so these results suggest that the fibrosis is kind of squashed. And this is a different way to look at it in a static condition. 2 weeks in, here we've actually identified the macrophages in green and the myofibroblasts in red, and the material is in white. And we've shown that in situ, actually, when we do intervital imaging, you see that without the coating, in this case is PDMS, there's a very strong immune response, as well as myofibroblasts that are associated with the coating. There's fewer immune cells, and actually even adjacent to the implants, there's less macrophages, suggesting there's less inflammation. And this translates to other materials beyond alginate. We've done some trials with polystyrene here, whereby these coatings can really prevent immune response and scar tissue formation on a range of biomaterials. And these modifications translated to non-human primates, whereby these alginate spheres that have these coatings were retrievable and mostly fibrosis-free after 6 months implantation. Now, coming back to the diabetes problem, as I mentioned, the preferred cell type for this application is the stem cell-derived islets. And now there's many formulations which have advanced to the clinic, which suggests that stem cell-derived islets have matured as a technology to be practical in the clinical setting. And we wanted to see how well our encapsulation solutions synergize with those. We did this study where we published in Nature Medicine that our lead formulation can actually protect these cells and allow for viability and survival for up to 6 months. And when we explanted those capsules, we see that they're fibrotic free, and they do have beta cells, pancreatic beta cells inside them that are viable, and they express the right markers, suggesting that these cells are still active and functional. And as I mentioned, many formulations over the years have been tried, many different iterations of alginates with different modifications. And these have gone all the way to clinical testing. We did a side-by-side comparison where we tested these formulations, contacted the investigators of these various trials, and actually did a comparison in non-human primates and showed that, they saw fibrosis in humans. Actually, in non-human primates, similarly, they're fibrosed. And we contrast that to our lead modifications of these hydrogels. And we noticed that in non-human primates, they're actually quite clean in terms of no fibrosis, no scar tissue formation. And we can implant these into different sites of the body. We've identified this lesser sac, the bursa ommentalis, which is already nicely vascularized, as a particular site where these capsules could be implanted and have the right access to nutrients and oxygens they need. And when we load them, in this case with NHP islets, which went to NHP recipients, again, immune mismatch, we were able to demonstrate that we're finally, for the first time, able to maintain viability of these allogeneic cells without any immunosuppression. And over a 4-month period, we maintained over 80% viability in these trials. And the cells that were implanted are glucose responsive, insulin secreting, which suggests that they remain functional. And we have a company that we've started based on this technology, and we're excited to report that actually this particular formulation is now advanced to human clinical testing in the context of a hemophilia trial, where the engineered cells secrete factor VIII. And so Sigilon Therapeutics is pushing this forward, both for diabetes as well as the hemophilia example. But we feel that the formulations that we've developed, while they look very promising, we believe that there's perhaps other formulations out there could perhaps work better or facilitate protection of cells at higher densities, which is a challenge that remains, because as you have more cells within the matrix, they become stressed and they secrete more and more inflammatory cytokines. And so you need the biomaterial to be that much better at suppressing the inflammation. So we wanted to expand this screening approach and come up with innovative ways whereby we could actually multiplex and test many more formulations simultaneously. We came up with a barcoding strategy whereby we leverage genetically barcoded HUVECs from different donors as a way to barcode the material formulations. We could simultaneously implant these into mice, extract them at 4 weeks, and identify the ones which have viable cells, and then come back and de-identify which formulations they come from. And in mice, we've been able to demonstrate the ability to do this with up to 20 materials simultaneously, because you're at this point volume limited. And in larger animals with non-human primates, we've been able to expand and actually test 100 materials simultaneously by using this barcoding strategy. And so this additional expansion of screening capabilities have actually allowed us to screen a lot of different analogs of our lead material and identify ones that are best suited for encapsulating and protecting higher densities of cells. We apply that selection pressure during the screen. And so the outcome is a material that not only prevents fibrosis as an empty capsule, but works that well such that when it's loaded with lots and lots of cells, it performs that similarly. And we've done tests now where we've actually loaded these with higher densities of pancreatic beta cells, going up to 16,000 IQs per mil of these spheres, and demonstrated that we get robust viability and survival with our formulation. And this is an 80-day study we've done, and we have ongoing studies with even higher densities, which look promising in the ability of these new form materials in protecting the pancreatic islet cells from attack. So with that, I wanted to just thank my team here at Rice University, our funding agencies, JDRF, as well as NIH, DARPA, and I'm happy to take your questions. Thank you so much.
Thank you, Omid, for the excellent presentation. We have time for just a couple of questions. And so in what way this immunomodulatory alginate capsule could be applied in the setting of cardiovascular diseases?
Oh, great. Yeah, thank you. You know, the beauty of this encapsulation technology is that we can implant a diversity of cell types. In fact, I didn't highlight it here, which I should have, but we had a publication recently where we encapsulated mesenchymal stem cells within these same alginates and implanted them in the cardiac space with the goal of modulating the inflammatory responses that occur post myocardial infarction. And we showed in that study, which is published, that these same materials can help protect and encapsulated therapeutic cells in that context as well. So I know when it comes to cardiac therapies, there's quite a lot of emerging cell trials, not just MSCs, but other types of cells as well. And so that's where we feel like this tool could be best leveraged towards cell delivery for a number of different applications, including cardiac, of course.
Thank you for that. And so beside macrophages deposition, have you looked at other immune cells, such as neutrophil, as an example, and their adhesion onto the alginate materials?
So we have looked at a diversity of immune cell types. We actually did a study where we applied a systems biology approach to elucidating the foreign body response. We published this in Nature Materials in 2018, I believe, or actually it was 2019. And the concept there was to evaluate which of the key immune players plays a bigger effect when it comes to the foreign body response. I mean, in that study, we actually depleted neutrophils, and we noticed that the fibrosis response was not significantly impacted. But when we depleted macrophages, they seem to be one of the key players orchestrating the key fibrotic response and the outcomes when it comes to cell failures. So that's why we primarily focus on macrophages, but we have looked at other cell types as well. And in our experience, they sort of correlate with each other, where as you get more macrophages, you also get more neutrophils and fibroblasts and some BNT cells as well. But the macrophages seem to be the most critical in that when you deplete them, it actually makes the fibrosis go away. So that's why we primarily focus a lot of our evaluations on macrophages.
Very interesting. Well, thank you very much, Dr. Vese, for your presentation. And now we are going to pass to the Shark Tank. John.
Thanks, Alessandro. Now for something different. The Shark Tank was very popular last year, and we're going to do Shark Tank Cardiovascular Nanomedicine again this year. Joining me are the Shark Tank judges, Professor Alessandro Grattoni, who is the Frank J. and Jean Raymond Centennial Professor and Chair of Nanomedicine, and Dr. Chris Lincoln, who's the Director of Office of Technology Transfer. I'm sorry, I gave you another job. Who's the Office of Translational Production and Quality, and has had a lot of experience in GMP manufacturing processes. And replacing me on this chair in just a moment will be John Schultz, who's the Director of our Office of Technology Transfer. And these gentlemen have a lot of experience in the regulatory roadmap, how to take things from basic idea all the way to the clinic, in fact into space even, with Dr. Grattoni, who's done experiments on the SpaceX Dragon. So with no further ado, what we're going to do is invite the other judge in, John Schultz, and he'll replace me in this chair, and then we're going to have the contestants come in that will be introduced by Dr. Grattoni.
Well, I'm pleased to present the first contestant of the day for our Shark Tank. Release of antibiotics. Okay, here we are with Shark Tank, and I'm very pleased to introduce to you the first contestant of the day, Dr. Taraballi. You have heard a lot from her earlier, and she is partnering with Dr. Rahimi, who is an assistant professor of cardiovascular surgery here at Houston Methodist. They will present a project titled Nano Against Infection, Nanostructure Coatings of Vascular Graft for Temporospatial Release of Antibiotics. Dr. Taraballi.
Thank you. And thank you everybody for giving me this opportunity. I will talk a lot about these tubes today, which is actually a vascular graft. What is the vascular graft or vascular bypass? Is a surgical procedure that aim to reestablish the blood flow in a part of the body. And unfortunately, and this can be, both limb, like leg or arm, as well as, and it can be venous or arteries. And what is the big problem in vascular graft? Actually, are bacteria infection. Graft infection is the major cause of complication of vascular surgery, and the morbidity and mortality is high of the 75%, and sometimes in the best case scenario you can lose the limb, but the percentage to this happen is 70%. What we can do to prevent vascular graft infection? Normally has been administered systemically antibiotics, of course, and as Dr. Rahimi introduced to me, they do a paste in the OR and they put on top of this in a completely off-label manner. In this way, we cannot control the release of the antibiotic. However, material sciences can help. And so what we can do, we can use the electrospinning technique to spin on top of this graft nanofibers. They are made by polymer and loaded by antibiotics. As you can see, nanofibers can be spun on anything and can be very sticky onto it. And as you can see here in this image, they can be loaded for pretty much whatever we want if we deal very well with the chemistry. And so this is our prototype in which we spun in a circular collection a coating, a nanostructure coating on top of this graft that is made by nanofiber, as you can see here in the magnification, and they are loaded with antibiotics. Which antibiotics we can cover both gram-positive and gram-negative, but the most important part for infection is the timing. So we need to achieve the delivery at the site and with the right timing, meaning we cannot be too early and we cannot be too late, as well as when we catch a train, but we have to deliver exactly at the same time what we want to deliver, in our case antibiotics. So what's the market though? Is this actually is a profitable place? Is a market that is growing? It is actually. With the aging of the population, we envision to arrive to a market of 3.3 billion by 2027 of vascular graft itself, and as well as if we speak about the technology itself, the nanofiber technology market seems to increase as well in the next 4 years. And most of the application of the nanofibers, of course, this technique of fabrication can be applied to any kind of things, in our case for medical device, and we can see that also this kind of technology will be very well used. What's the team? The team is this one. I'm working closely with my cardiovascular surgeon, Dr. Rahimi, and then you can see Stefano Serpelloni, that is our Spider-Man, so is the person that actually is a bioengineer that do the nanofibers, as I showed you before. And then let's see what I want to do with this $10,000. First of all, I will be very happy. Second of all, this is our project. Of course, it's in the matter of 1 year, so we want to really, we already have several preliminary data that I didn't present yet, because also we have an invention disclosure pending, but we achieved the first part of the loading of vancomycin and gentamicin. So the first month is just to close that part of optimization of the device, and then we're gonna speak about efficacy in bacteria in vitro and cytotoxicity, as well as the animal model by the end of the year. Of course, $10,000, for as much as happy I will be, will not be enough to cover everything. We have other funding, and we also are will use this money to apply for other money, specifically R21 and AHA model. With this, I think I close my minutes. I hope that I convince you all. Thank you in a word on nanofibers.
Grazie Francesca, thank you to Francesca. I switched to Italian because there's too many Italians even in your team for a minute. I'm sorry. So I have a few questions for you, and then I will ask the other sharks obviously to participate. So all of these graft, the vascular graft, will have different size, and depending on the vessel they need to be treated. So because of your coating will be release the drug depending on the surface area of the graft, How will you be able to modulate release based on different size of graft for different vessels?
So this is a very good question. What we want to do is, at the beginning we were thinking to do a sort of ribbon. So basically, as you said, the release is based on the area of the coating and the thickness. So a lot of more than the area come from the thickness, how thick is the wall that we create with this coating. But at the same time, what we were envisioned to do, instead to have a device, have another device that we can basically attach as a ribbon, and measuring the length, we can measure the quantity of the drug release. But at the same time, as long as it is the vascular graft, as much more probability to do infection, as much probability to have an infection at the site. So most of the time, the surgery in the OR, what they create this space of antibiotic and they cover all the graft. So what we want to try to do is have a way more regulated device that allow us to release as much as we want. And again, we envision different size, but especially different thickness to control the amount of drug released.
Perfect. Other sharks, do you want to ask some questions?
I've got a quick question. Dr. Taravalli, I was happy to hear that you'd be happy with the $10,000, but I was also very happy to hear that you've got a pending invention disclosure with our office as well, because that was really going to be my first question. But really, as a follow-on, I'm wondering, you envision this being done at a manufacturing facility, correct? At a factory that makes the grafts, correct? Mm-hmm. Okay. Who are the market leaders in this area that supply these grafts?
Well, no, this you catch me not super prepared, because I normally let you go towards that thing, but most of the time, all the, for example, Medtronic, I believe, has some kind of similar eluting device, so a device can release the drug at the site of implant. And when we actually talk with the surgeon, he told me that he has some contact with like companies that normally provide this kind of device to him, in order to have at least the first people to talk with, if they will be interested in something like that.
Yeah, I'm sure the market's growing so fast, they're very competitive, so opportunity to differentiate themselves with your technology would be quite interesting.
And also another thing that I want to add, since it's a coating, is in the coating of the vascular graft, but the invention disclosure part is on the method of fabrication and deposition. I cannot enter more in the details, but in this way we can actually coat any kind of medical device. So the escalation of this kind of technology can be also extended to other kind of device that has the same problem of infection. I'm thinking about the orthopedic implants that has a big burden of infection-related device.
Thank you.
Yeah, so I think this is an idea I definitely can get behind. It's a really great idea, but I need a few things that need to be clarified. So when you're doing the spinning, are the drugs included with the fibers as you're spinning them, or are they loaded afterwards? And I mean, I know you probably can't tell me a lot, but give me high level what you're talking about.
No, the drugs are loaded and spin together with the polymer. So you basically do a solution of X amount of drug A, drug B, and you use different kind of polymers at a certain concentration, and you spin together.
So how do you ensure a burst release, which is what you're calling for, and then a extended release over, however long it's going to be, 7 days, 10 days.
Yes, actually we want to achieve even extended. I didn't have time to in the presentation to say, but it's very important for infection, the timing. So what we envision is like that depending on the polymer that we use, the hydrolyzation speed of that polymer, we can tune the release kinetics. So we're gonna have a burst release in the first week, in the first 24 hours after, towards the first 7 days, and then we're gonna have at the same time a stage release up to 30 days. How we achieve this? With layer by layer deposition. So as much as is exposed to the blood, so basically the polymers start to degrade and start to release the drug embedded in it.
My last question, are you concerned about uniformity of coating? And is that going to have an effect?
Yes, it has an effect. Is a limitation of the technique. We have it under control, and what we actually do to monitor, and probably in the poster that the student presented, we show what we do. We basically get the whole graft, and to understand if it's delivered in the same way, we chop it, and we understand what is the loading efficiency in the different parts, what is the thickness and what is the release, in order to have homogeneity of the delivery and the loading, as well as it's not just the delivery but also the loading throughout all the length.
Thank you, Dr. Taraballi. This has been a very exciting technology and presentation. We need to move forward with the second contestant of the day. And therefore, I will be happy to introduce Dr. Carly Filgueira, who is an assistant professor in the Department of Nanomedicine and also in the Department of Cardiovascular Surgery at Houston Methodist. Dr. Filgueira would also is teaming up with Dr. Maturia, which is a clinician from the Department of the Division of Cardiac Electrophysiology from the Houston Methodist. Dr. Filgueira will present today about her project on nanoparticle-guided treatment for cardiac arrhythmias. Welcome, Dr. Filgueira. Welcome to the Shark Tank.
Hello Sharks, my name is Nilesh Mathuria, Associate Professor of Clinical Medicine at Weill Cornell and a practicing cardiac electrophysiologist here at Houston Methodist.
And my name is Dr. Carly Filgueira, an Assistant Professor of Nanomedicine at Houston Methodist Research Institute, and we're here to seek $10,000 for our proposed company, Nanoablation, which will disrupt the cardiac ablation market with a revolutionary way to ablate abnormal cardiac tissue that causes cardiac arrhythmias.
Carly, tell me more. How does this work?
Our technology is based on small nanoparticles that act as nanoantennae and are able to generate sufficient heat to cause tissue death. This tissue necrosis would be controlled to only occur where the particles are localized, so that healthy tissue remains viable, but abnormal tissue is destroyed. Our nanoantennae are able to specifically target areas of the heart with abnormal conduction by placing site-specific ligands that bind the nanoantennae to targets overexpressed when damage occurs to the heart. Some known targets published in the literature include angiotensin 1, myosin, and phosphatidylserine. This technology would be used in patients with recurrent arrhythmias, a challenging patient population with a high morbidity and mortality rate despite our current clinical standard of care.
As a clinician, cardiac ablation is our current practice for medically refractory cardiac arrhythmias. Specifically, radiofrequency ablation is the most common modality used today in practice. Typically, this involves a deflectable catheter to go into the region of interest within the heart where ablation is performed. Although this is the standard, there are many limitations to this approach, which really have led to plateaued success rates and persistent complications. Most often, unlike seen here, images of the heart are shown which make it seem that the heart chambers are nice and smooth and easy to navigate. However, these figures here highlight the realities that what we as clinicians face during these procedures. For instance, here in the left ventricle, this dissection highlights the highly trabeculated nature of the left ventricle, for instance, and the varying degrees of wall thickness that we see. So as you can see on the right, therefore, on the right figure, in clinical practice, a relatively small ablation catheter is often not able to eliminate all these areas of abnormal conduction targeted for ablation as represented by the yellow stars. Further, these areas of interest can occur deep within the ventricular wall where RF ablation depth is often not sufficient. And given the underlying trabecula, the inherent beating motion of the heart, catheter stability is a constant problem which limits our effectiveness. Further, these radiofrequency ablation catheters are irrigated, which means these patients are getting high amounts of fluid during the procedure, which often can lead to complications both peri-procedurally and even post-procedurally.
Wait a minute, so you're telling me as a clinician, Nilesh, that you're facing problems of catheter instability, the need for continuous contact, limited ablation depth, and fluid administration? It sounds like we need something better. Like nanoablation. Specifically, nanoablation works by nanoparticle perfusion in the specific regions of abnormal tissue leading to cardiac arrhythmias. An external source is able to activate the nanoantennae to reach target temperatures above 50 degrees C, where permanent cell death occurs, but less than 100 degrees, where complications such as perforation can occur. In preclinical preliminary testing at the bench, nanoablation was so effective, only one application was necessary to achieve durable tissue ablation.
So therefore, we propose a nanoparticle guided treatment for cardiac arrhythmias. Although this could be potentially applicable to all cardiac arrhythmias, really our index patient population of interest will be those who are suffering from recurrent ventricular tachycardia. This is a patient population that is growing exponentially with more than fourfold increase in catheter ablation procedures just for ventricular arrhythmias, with now more than 3,000 plus ablation procedures performed in this space just in the United States alone. Further, we anticipate that this delivery will be intravenous. Now, obviously, subsequent efficacy and safety studies, both in large animal and early phase 1 clinical trials, may change that to either coronary injection or potentially even catheter-based balloon occlusion techniques, some of which have been pioneered here at Methodist, where we can actually target the region of interest. As the sharks know here, the cardiac arrhythmia rhythm space is one of the most booming sectors within the cardiovascular industry. All projections point to this being a tens of billions of dollar industry just in the short-term future, if you will. Even with our current ablation catheter technology, this accounts for nearly 40% of much of the early expected or near-term revenue based on various projections. Given the potential game-changing technology of nanoablation, all the market leaders in the field, many of the companies you see listed here today, would have a vested interest in pursuing technology such as nanoablation.
With your support, we'll be able to finalize our prototype. We're currently at this step creating a prototype and in vitro testing, which will be followed by scale-up device validation, preclinical testing, GMP manufacturing, clinical testing, and finally, marketing our nanoablation. So, Sharks, who will join us as we begin this journey of nanoablation to change patient lives around the world?
Well, thank you very much. This is an excellent presentation. I would join you, but first you need to answer some questions that I have. First question is, This technology is very promising, but how will you be able to make sure that your particles are delivered precisely where they need to be delivered at the right concentration so that you can perform the ablation?
I'll go ahead and take this. And so really, obviously, this is going to be a stepwise approach. At this stage, based on what Dr. Filgueira mentioned, initial testing and then subsequently even small and subsequently large animal testing will be able to determine, number one, particle concentration, particle retention, not just acutely, but then even for exactly how long, and then even potential other end organ effects, if you will, of particle retention. So these are some of the initial proof of concept steps that we will need to overcome here in the first few steps before we get into ultimately clinical trials.
In order to achieve that, it's going to be a twofold approach. It's going to be a local delivery approach plus a targeted delivery approach. So one of the things that we envision is a balloon procedure where we inflate the balloon and we have the particles localized in that region where the balloon is. And we've actually already done some preclinical testing where we did this in a tube where we inflated the balloon and we saw that the particles did not dissipate, that they stayed within that region where the balloon was inflated for over 20 minutes. The particles still remained in that localized spot. So we think we can overcome this challenge. And if we need to, we can use a double balloon approach as well.
And this is an approach that's been used clinically for other forms of chemical ablation for refractory arrhythmia. So that concept is already there as far as particle delivery, if you will.
So the device, is this piggybacked off an existing device, or is this a brand new device you guys are developing the prototype for?
So as far as the device, that's going to be standard techniques that we currently use to get to the regions of interest. So whether that's through intravenous approaches, whether that's through coronary injection, whether that's through, as we mentioned, balloon occluder catheter-based approaches, these are all standard techniques, off-the-shelf techniques used by multiple vendors for multiple clinical applications. The specific, obviously, goal here would be to get the particles of interest to the region of interest.
So focusing back on the particles then, it sounds like you have somewhat of a platform technology where you could pick the marker you want to go for and then decide how you're going to do the ablation based on the marker. So is there one in particular you're going to focus on first and move down the pathway, or are you doing a pan approach where you're going to try a lot of different markers? How are you going to move toward that GMP process where you're controlling what you're trying to make?
So we mentioned 3 that are particular of interest, but we also have others that we aren't ready to disclose right now, but we do have other targets that we're interested in working with. As a scientist, you always have to have your backup plan or your alternate strategy. So that's why we wanted to have 3 potential candidates that were very, very strong. In fact, you've heard a lot today, earlier this morning, about phosphatidylserine, which is one of our strong candidates. I'd also like to say that we have different alternative ways to activate. I talked about activating those nanoantennae, and that's part of this whole technology. It could be either light-based approach, which is known in the field, using something like the water window, which there's transparency in the near-infrared, or something like an externally applied magnetic field that could be used to activate the particles.
All right, thank you.
Well, you've answered all my questions before I had a chance to ask them. So I just let's get this finished so we can go license this. This is a great idea.
And we'd also like to mention that we do have an invention disclosure filed with your office, John.
Well, then I will ask an additional question because I am really intrigued. So what is the safety of these gold nanoparticles into the cardiac tissue?
That's a great question. In fact, when I first got my ears pierced, the metal that they used to pierce my ears was gold, a gold earring. So it's known that gold doesn't oxidize readily. You don't really have too much of an immune response. It's one of the types of metals that it's really biocompatible. In fact, sometimes you can even get it on a fancy dessert that you order at a restaurant. It does end up going to certain off-target organs when given systemically, such as the liver or kidney, and we can do some different types of quantifying to figure out how much goes there. But it's really not something that is of too concern. In fact, they've given this in clinical trials to humans already, and it's been evaluated safety and efficacy. It's just not been evaluated for something like our technology or proposed for the heart. So we don't anticipate too many hurdles related to safety.
Well, Dr. Filgueira, Dr. Maturia, thank you very much for your presentation. This was excellent. Well, I'm very pleased to present the third contestant from the day, Dr. Longgu Fang. He's an associate professor in the Center of Cardiovascular Regeneration at Houston Methodist, and has partnered with Aifa Shen, who is a full faculty in the Department of Nanomedicine, in this project that is titled Engineer Clearance of Vascular Senescent Cells to Improving PAD, which is Peripheral Arterial Disease. So, Dr. Fang, please.
Okay. First, I would like to thank the committee for giving me an opportunity to participate in this Shark Tank Cardiovascular Nanomedicine competition, and I also would like to thank the generosity of this Kostas family for me to have an opportunity to present my application. So my lab mostly focus on the periphery, focused on angiogenesis, a process of vascular growth. So as some of you may know that PAD is a peripheral arterial disease. So the PAD is caused by atherosclerosis, which is atherosclerotic plaque present on this vascular wall. And PAD affects millions of Americans, 10 millions of patients in the U.S. And showing here is the normal artery. You can see that the blood flow is smoothness in this vasculature. However, in this PAD tissue, the atherosclerotic plaque that is formed on this vascular wall will cause layering of this artery, and this will limit the blood flow to this the blood flow and restrict the tissue oxygen supply. So the PAD mostly occurs in the aged populace. So we hypothesize that vascular agent. So we hypothesize. What is the leg? So we hypothesize that vascular agent, which is mostly caused by the senescent endothelial cells, will contribute to this disease. So if we can selectively eliminate this senescent endothelial cells, we can improve vascular regeneration. So what is the senescent endothelial cells and what they cause to this PAD? The senescent endothelial cells, they will have several important features that contribute to this PAD. So first, they have this SASP symptom, which is a secreted senescence-associated phenotype. They will secrete a lot of cytokines and chemokines that contribute to this inflammatory environment. And also, they have this telomere erosion and also this cell cycle shock, so they don't proliferate. But on the other hand, they are resistant to apoptosis, which means they don't die, but they can cause the harm. And lastly, when you do a staining, you can see that they have increased expression of this beta cyclidase. So the cytokinetics is a selective drugs that can kill this senescent endothelial cells. So when you have, as I mentioned in the previous slides, that if you have senescent endothelial cells, they will contribute to this local inflammation, and therefore might cause this the immune cell aggregation and cause the stop of the blood flow. So we hypothesize that the cytokinetics, when encapsulated by this nanoparticles, they will selectively kill this senescent endothelial cells. Therefore, the healthy endothelium in the nearby will come and cover this released space. And according to Dr. Cooke and Dr. Cooke Alex, the recent review published in Circulation Research, I would imagine that there will be 4 different forms of vascular regeneration, which include angiogenesis, arteriogenesis, and de novo of vascular genesis, and also this transdifferentiation. So this is so this is what we're going to do. So since CD39 is usually highly expressed in the senescent endothelial cells, so we propose to capture this CD39 molecular antibody to these nanoparticles, and then in addition we also put this GAL6 modification on this nanoparticle. Inside this nanoparticle, there's going to be a cytolytics is BCL2 inhibitor. So we think that we hypothesize that when we deliver this this nanoparticles to the in vivo, this nanoparticle will bind the senescent endothelial cells, including this senescent endothelial cells that express high level of CD9, and then this senescent endothelial cells will uptake these nanoparticles, and inside this lysosome, this beta-galactosidase will cleave this GAL6 of this nanoparticle, and which will lead to the disruption of this nanoparticle, and this inside BCL2 inhibitor will be released, and then will lead to this killing of this senescent endothelial cells. Then finally will be removed by the immune cells. So here's what we propose to do in vitro. So we're going to either use white type HUVEX or the endothelial cells that are subject to senescent treatment. And then we mix these two type of cells together, and this type of cells will either be subject to the cyanolytics treatment, unpacked cyanolytics, or these nanoparticles that capture this cyanolytics. Their final molar they're going to be in the same concentration. And then what we expect is that the cyanolytics, this nanoparticle encapsulated cyanolytics will be rescue this in vitro angiogenesis to the level that is comparable to this wild type normal endothelial cells. But it's much better than the naked cyanolytics or the control. Our future studies, of course, will have to assess using the animal models to assess this how the therapeutic effect in the murine hemodynamic ischemia model, and also we will have to see in patients what's the percentage of senescent endothelial cells in ischemic muscle and microvasculature. And if this goes well, we're going to do synthesize the GMP grade senescent endothelial cell targeting nanoparticles that encapsulating this cyanolytics. And finally, hopefully, we can have, if it goes well, we can go for phase 1, phase 2 clinical trial. So, lastly, I would thank our Center for Cardiovascular Regeneration, Dr. Cooke, for the support, and others, other members in my lab. And thank you all for your attention.
Dr. Feng, quick question for you. So, do you have preliminary data that are supporting your hypothesis that this approach may actually work?
Well, I don't have data by myself, but there's the data from published studies showing that this cyanolytics indeed can promote cardiac regeneration after the ischemia and reperfusion. But their mechanism is through the if I remember correctly, is through the reduction of fibrosis. So we propose that it can improve vascular regeneration. So it's different target, but can achieve the same effect through vascular regeneration.
Thank you. And another quick question. We have been notified, we know that Dr. Aifa Shen will be retiring from academia, and he's a partner in your project. So who will you collaborate with in the nanomedicine team for continuing this project, if successful?
That's a good question. I just know. I hope that since Methodist is has strong nanomedicine team, if I was chosen to be funding, I assume it won't be a problem in our institute.
Fair enough. Other sharks?
I have a quick question for you, Dr. Feng. Is there a reason you picked the navitoclax for your senolytics?
No, it's not. It's just whatever is good. I just use that as an example to show a proof of principle. I think whatever senolytics can be, as long as it work well, can be good for it.
Okay, yeah, the reason why I brought that up, because I know Navitoclix has a thrombocytopenia, so it could be quite dose limiting. You know, in a oncology environment, maybe that's not so much of an issue, but maybe in something more chronic.
Right, yeah, that's a very good point, indeed, yeah.
Yeah, just listening to the other sharks ask questions, it sounds like you've got a lot of pathway diversion that can happen, right? So you haven't really nailed down the molecule you want to encapsulate. Navitoclix is good, but it could be something else. So how does that influence your strategy for scale up, for controlling manufacturing, if you're not even sure what molecule you're going to land on?
That's a good point. That's why we need that. That's why you see I only propose an in vitro experiment to do that pilot testing. So it's I think at this stage I can only say this from the theoretical point of view. What we can do. This is a promising therapeutic pathway. To bring anything to the ground, we need pilot studies. That's why I think this the support from the Kostas Foundation is critical for the pilot testing.
Thank you, Dr. Fang. This has been a good presentation. So now this group of bloodthirsty sharks will deliberate and see what project they want to get on board with.
Thank you.
Cool. All right, now.
Well, that was fun. I want to thank the Shark Tank judges for their work and the participants. But don't go away, stick around. We're going to have the video right now, Kostas family tribute. And then following that, we're going to have the awards. We'll hear who won the Shark Tank, and we're also going to hear who won the poster awards and who won the Kostas grants this year. So stick around. We'll be back with you after the video.
So when I say the word Kostas, the first thing that happens is a big smile comes across my face. And honestly, that's the reaction that I had from being around Mr. and Mrs. Kostas. They were just really nice people. The next word that I would think about in describing them is humble. I had the opportunity to travel with Mr. Kostas when we went up to Northeastern. It was amazing to see the pride that he had in his alma mater and to see in what high regard all the people at Northeastern who we met that day actually had for Mr. Kostas. And I'd say the same thing really about the folks down here at Houston Methodist. He used to come to our Heart Center Council and listen intently, and then always asked very in-depth questions. And he was listening. He was listening to everything that was being said. And when he came up with the idea of endowing and linking what were really, at that time, two of the really strong centers of excellence that we had here, nanomedicine and cardiovascular disease, I thought it was just a brilliant idea. And it was set up in a way that kind of drove the interaction between clinicians and the basic scientists, because he recognized that that was a gap, that we had essentially built two parallel towers with not enough interaction taking place. And so I actually think that the grants in the Kostas name, probably the most effective grants here that have fostered that kind of collaboration, because it persists really to this day. Once you get it going, it persists to this day. And you're going to hear from several of the people who have had the benefit of receiving those grants, and they've done incredibly well and presented their work, got subsequent follow-up grants, and I think he would be very proud of what has been achieved, and I think it served exactly what he wanted and what the goals and the directions that he wanted to see this going. Again, I just have this kind of warm feeling every time I really think about the family, because they in return are so warm and embracing, have given so much to Methodist, so much to the community here in Houston, and so much up in Northeastern, and that is truly, truly remarkable. I really want to thank the entire Kostas family for initially creating this and for their subsequent dedication to making sure that we continue this in his memory. Thank you very much indeed.
Hello, my name is Huey Lin, and I'm Director of the Adult Congenital Heart Program here at Houston Methodist, and an adult congenital interventional cardiologist. With Dr. Blanco, I want to thank the Kostas family for supporting our work in interventional cardiovascular cardiac MRI. What this means is the work that the Kostas family supported allowed us to develop and build devices that really advanced the ability to do cardiovascular catheterization and intervention in a radiation-free environment in the cardiovascular MRI suite. In addition, through the support of the Kostas family, we have been able to continue innovating the way we care for adults with congenital heart disease here at Houston Methodist. Thank you so much for all of your support.
My name is Maham Rahimi. I'm a vascular surgeon in Houston Methodist Hospital. When I came to Houston Methodist in 2017, my goal was to establish myself as a surgeon and a physician scientist. I was fortunate to hear about Kostas family and their support in advancement of cardiovascular research and nanomedicine. It was an exceptional opportunity for me due to my background in vascular surgery and nanotechnology, and I applied for a grant. I was fortunate to receive the support of the Kostas family and start my dream, my career in cardiovascular nanomedicine. I was able to present my work in several conferences and able to publish my work last year. I am grateful and appreciative of the Kostas family for their support of my research that has started my career and allow me to participate in an exciting research which has led me to apply for NIH R01 grant and support advancement of cardiovascular nanomedicine. Thank you, Kostas family, for your gift and support.
Hello, my name is Bruna Corradetti. I'm an assistant professor at the Department of Nanomedicine at the Houston Methodist Research Institute. And today I would like to express my greatest gratitude to the Kostas family for supporting my vision about developing RNA therapeutics for the treatment of chronic wounds associated to cardiovascular disorders. Cardiovascular disorders represent a major, with the leading cause of disabilities in the United States, with an enormous impact on patients' quality of life. The numbers associated to chronic disorders has been predicted to further increase as the result of the COVID-19 pandemic. And this will make the impact we can create with the research you kindly supported even bigger. While the promise of RNA therapeutics is still being realized, I am very proud and very honored to contribute to innovation as a part of your team. Thank you very much.
I would like to thank you, Georgia and Cynthia Kostas, and the Kostas family for your vision and support for the Kostas Center for Cardiovascular Nanomedicine. Over the years, the center has provided valuable resources to foster collaborative research between faculty in the Department of Nanomedicine and clinical scientists in the Houston Methodist Cardiovascular Group, with the goal to benefit patients affected by cardiovascular diseases. As of today, the Kostas Center has prompted the largest number of active collaboration among a research and clinical group with HMRI, a clear sign that your vision has always been on point and has already made an incredible impact. I also would like to thank you for your sincere commitment and dedication to this program, which demonstrate your genuine desire of doing your best to help us researchers succeed in our work with the ultimate goal of improving and leading medicine. Thank you.
Hello, I'm Steve Igo, and I welcome this opportunity to pay tribute to the Kostas Family Foundation. My meetings with Dr. George Kostas were always memorable and truly a joy. The annual program dinners hosted by his daughters were delightful. In 2016, the Kostas Foundation funded our project on controlled intrapericardial drug delivery for treating heart failure. Our cardiovascular nanomedicine team included surgeons, scientists, biomedical engineers, and research fellows. We used our novel access device to insert a catheter into the pericardial sac surrounding the heart. These X-ray videos demonstrate that IPC delivery is targeted and heart-specific. We studied the cardiotherapeutic effects of a clinical generic drug using a porcine model of myocardial ischemia reperfusion injury. Our test drug was delivered IPC by continuous infusion or by single injection of controlled release nanoparticles. This is a new use for a clinical generic drug. Compared to IPC saline infusion, IPC delivery of free drug or nanoparticles significantly improved left ventricular ejection fraction following myocardial ischemia reperfusion. In 2019, we published this review article on technologies for intrapericardial delivery of therapeutics and cells. The field continues to expand. Our Kostas project has resulted in intellectual property disclosures to Houston Methodist. These include device platform technologies for IPC access and an implantable pericardial catheter. The IPC drug and delivery methods we investigated are novel for treating myocardial infarction and heart failure, the leading causes of death and disability worldwide. So, to the Kostas Family Foundation, thank you for supporting our cardiovascular nanomedicine project for heart repair.
I'm Dr. John Cooke, Professor and Chair of the Department of Cardiovascular Sciences, and I am so thankful for the support of the Kostas family for our work in cardiovascular nanomedicine. A few years ago, at a holiday party, I talked with the patriarch of the family, George Kostas. I sat down to say hello to George, and soon the din of the party faded into the background during what became a very lively discussion of what it takes to innovate, to really make a difference in a scientific or a medical field. Certainly, George spoke from experience. He was a remarkably productive visionary who pioneered the design and operation of the first synthetic rubber production plant during World War II, when the Allies really needed it. Another very useful invention was his patented corrosion-resistant zinc coating. Both of those innovations were driven by his brilliance and energies, sure enough, but there was something else, his need to give back and to leave this world a better place. He and his family are deeply religious, and George focused his time and his philanthropy on religious institutions, as well as on science and technology. And with respect to that, science and technology, in our nano world, George Kostas was a mega spirit of humanity and innovation. The George and Angelina Kostas Center for Cardiovascular Nanomedicine at Houston Methodist Hospital is a great example of his philanthropy, his vision, and with the help of the Kostas family, will continue to make a difference here at Houston Methodist. Welcome back. We're here in the DeBakey studio. I'm with Cynthia Kostas here on my left, and Georgia is joining us by video, by Zoom. Hi, Georgia. Can you hear me? So Georgia is joining us by video, and what we're going to do first is I'm going to make the awards for the meeting. And then I think you have some remarks.
Yes.
You'd like to make some remarks, and I think Georgia also has some remarks as well. So it's my pleasure to make the awards for the George and Angelina Kostas Research Center for Cardiovascular Nanomedicine. We're going to start with the poster session awards. And for the poster session awards, we had 6 awards. And the first 3 third place winners in our poster contest were Rhonda Holgate with her talk on RNA-enhanced skin repair and rejuvenation, Yarel Carcamo with her talk on intrapericardial treatment in a porcine model of reperfused myocardial infarction, and Abishai Dominic for a surprising role of TNIC in regulating endothelial interferon signaling and activation. So those third place awards garner them $350. Congratulations to Drs. Holgate, Yarcamo, and Dominic. Now our second place winners, there's 2 second place winners, are Troy Hendrickson for disruption of polycystin 1 increases susceptibility to atrial fibrillation through altered DNA damage response, and Rolando Alvarado and Nada Pahar for a circular RNA vaccine for COVID-19. Those second place award winners get $750 for their fine presentations. And now the winner of the George and Angelina Kostas Research Center poster presentations for her talk is Anahita Mojiri for an RNA nanotherapeutic for reversing vascular senescence and extending lifespan, and that's a $1,000 award for Dr. Mojiri. So congratulations to the poster session winners. And thanks to the Kostas sisters for the funding for those awards. And now we'll announce the Shark Tank winner. If you didn't have a chance to see the Shark Tank, it was a lot of fun, even better than last year. And I think it's something that we're going to continue because what that is about actually is about taking an idea and turning it into something useful. And that's what we want to do at Houston Methodist. So Shark Tank, yeah, it's fun, but it also is representative of what we intend to do in terms of taking ideas and translating them into transformative therapies. So let me get the envelope here. Okay, the Shark Tank Award winners this year for their talk and their presentation on the ablation technology is Carly Figuera and Nilesh Maturia. Congratulations, guys, for your wonderful presentation, and thanks to the other Shark Tank presenters and the judges. Every year we have also grants from the Kostas family for projects that are worthy of moving forward toward translation. And we have 3 awardees this year, and each of these award winners will receive $50,000 for their projects to enhance or speed their projects forward. And I should say we had 9 proposals this year, and I thought they were better than ever.
Excellent.
And they were outstanding proposals. And so it was a little bit difficult to choose the top 3, but these did differentiate themselves from the others on the basis of the science and the clinical merit and the likelihood that they're going to keep moving forward to the clinic. So those 3 awardees, each of them get $50,000 for their project, are in no particular order. Roman Sukoverson and Biana Godin with Khurram Nasser on an RNA-based therapy for familial hypercholesterolemia. So their idea is to use RNA encoding LDL receptor to treat familial hypercholesterolemia. That's a great group with Roman, the scientific director of the RNA Corps, Biana Godin, who's a nanomedicine doc, and Khurram Nasser, who's a preventive cardiologist. And then the second grant was to Bruna Corradetti. She led the effort, along with myself and Maham Rahimi, on engineered RNA-based conductive patches for the treatment of endotheliopathy-associated chronic wounds. And then finally, Arvind Bimraj and Keith Euchre, who are leading our heart failure effort for their nanovaccine, HSP60 mRNA loaded into a lipid nanoparticle for heart failure. So 3 exciting projects, and the Kostas funding will help move those forward. So now I'd like to hand the floor to you, Cynthia, for some concluding remarks for this meeting.
We've had a wonderful day today and want to thank everyone, the participants and the observers, for being part of this. I was asked to say a little bit about our grant, and I wanted to just mention that my mom and dad, George and Angelina Kostas, oriented their life towards serving God through serving humanity. Their purpose in beginning this grant was to encourage research that was innovative and that could go quickly to clinical solutions in this new burgeoning field of cardiovascular nanomedicine. We chose Houston Methodist because of the amazing care that they received by doctors here, such as Dr. Torrey, Dr. Lumsden, Dr. Robert Jackson, Dr. Sandra Sessoms. And my parents fostered and witnessed that this kind of collaborative effort between medical industry, clinicians, and researchers could really yield focused ideas and outstanding outcomes. The purpose of this conference, as was envisioned by my parents and funded by my sister and I, was to bring forth cooperative alliances, inspire one another for all of the scientists that came, and to open doors for further collaboration. All of you who participated today are continuing that great tradition of being learners, lifetime learners, and we appreciate that that is what elevates your field and brings good patient care to the clinic. A conference like this doesn't happen without a lot of hard work and thought and planning, and so Georgia and I especially wanted to thank Dr. Dirk Saltzman, Dr. Alessandro Grattoni, Dr. John Cooke, Rebecca Hall, Emily Schultz, and Ann Duhan, all of those who played a pivotal role at different points to bring this gift and this conference to fruition. Georgia and I are so pleased, and I know I would echo Dr. Lumsden's words. We know that my dad and mom would be so proud and pleased at how Methodist has stewarded our gift. Thank you again for being here, and we look forward to seeing you next time.
Thanks, Cynthia. That was wonderful remarks, and I think Georgia has a few words to add as well. Georgia, can you hear me?
I can, and I want to add.
I think you might be on mute. We can't hear you here.
Okay, let's try again.
I can't. We're having trouble hearing her. Sorry, Georgia. Let's try to give that another try because we couldn't hear you here in the studio.
Can you hear me? Is this better? Is it now coming through? Okay. I just want to add my congratulations and thank you to all of you at Methodist and all of you who participated today to create this fantastic conference. You've done an excellent job with all the presentations, and Cynthia and I are so proud of all of you. We thank you for your dedication. We thank you for inspiring us today, informing us today, and invigorating everyone with new ideas that hopefully will propel the movement forward. And as was said today, viva this new era in cardio nanomedicine. You brought it from ground zero to takeoff, and we're excited about the exploration you were doing into inner space. And we wish all of you well and Godspeed. Your products, your processes that are helping to create this pipeline of great promise to people everywhere. So thank you, Methodist.
Thanks, Georgia and Cynthia. Before we close, there's some other people I want to thank as well. Those are Christina Herrera, who helped a lot with the organization of the meeting, as did Mariana Smith. And the guys in the studio here, led by Tyree Horn, Monique Andy, George Tripsis, Fernando Castillo, and Obed McCallop. And then, of course, Alan Lumsden, who developed the DeBakey Education Forum. All of our speakers, poster presenters, panelists, and judges, thank you. And I really want to thank the Kostas family again, because you've done so much for us. You've really brought this cardiovascular nanomedicine to Houston Methodist. And I want to thank you for the knowledge that we gained today. I want to thank you for the passion and enthusiasm that we felt today, and the hope and the promise for the future. Thank you so much.
Thank you, Dr. Cooke.
And so concludes our meeting today. Thank you all for joining us at the Kostas Annual Symposium on Cardiovascular Nanomedicine. Thank you.